Hope you all enjoyed yesterday's session. Let us start today's session. Arogiraj sir?
Yeah.
Am I audible, sir?
Yeah.
Can we start the session, sir?
Yeah, it's okay.
Sir, shall we start the session, sir?
Yeah.
Sir? Shall we start, sir?
Yeah, you can start.
Today's session starts with Dr. S. Arokiraj, Assistant Professor, Department of Food Science and Biotechnology, Sejong University, Republic of Korea. He has published more than 80 research articles and has more than 10 years of teaching and research experience. He has received many awards and grants from Government of India. He has filed an Indian patent. With this, I would like to invite Dr. S. Arokiraj to deliver his lecture. Welcome you, sir.
Okay, is it visible? My screen,
Yes, sir.
okay.
Yes, sir. Yes, sir.
So good morning to everyone. First of all, my apology for the delay. So this is COVID time. So I think you know. So unfortunately, I got some. I need to take some class for my students. So that is a delay. So again, my apology. So today we are going to discuss about the impact of herbal medicine in this COVID-19. So before starting my session, I thank the principal, the professors and the management and staff and students of Sri Vidya Mandir Arts and Science College, Krishnagiri district for organizing this international conference. So in Tirukural, so before I need to start with Tirukural. So in Tirukural there is a poem called Katrilin Ainum Ketka Abdurvarku Urkatin Utrandunai. So it means usually most of the students you can know. So some students, they don't study in the examination, but at the time of examination, so before the exam starts, you can see many students, particularly the last bench students, they will ask the questions or they will ask the answer, the person who already known about that answer. Okay, so it is like that. So this kind of international conference is kind of sharing the knowledge and exchange of knowledge, exchange of information. So appreciate all the students for attending this conference, because you can receive a lot of information, and also appreciate the staff. So in this session, I used to discuss about what are the importance of medicinal plants and use of medicinal plants in viral prevention and treatments, and general techniques. And also, if time permits, I can discuss about the job opportunities in pharma, and also higher education opportunities in South Korea. Okay, so most of the students are life science students. So I used to congratulate, I used to appreciate all the life science students. You need to feel proud, because most of the life science students, they feel like they feel disappointment, or they feel lack of confidence. Oh, I took life science students, I took this life science, I took microbiology, I took botany, I took zoology, like that. So in this corona pandemic period, the corona taught the world, so without science there is no life. Okay, so without science there is no life, because last year, I think in March or April, I don't know. So India declared the lockdown. So during lockdown, there is no car, there is no bus, there is no airport. Still, there is air travel was blocked, and no train. And you can see many people, they are walking from Tamil Nadu to Uttar Pradesh through the highways. So almost everything is stopped. But there is only the one machine that was continuously running. That is a PCR machine. Okay, so by using the PCR machine, they used to detect whether you have corona positive or not. So in this COVID time, in the entire world, the life science workers, professors, and researchers, they played a very important role in controlling this pandemic period. Why I am mentioning means, many people, they start appreciating the police workers, doctors, nurse, but many people, they forget the most of the molecular biologists or virologists or microbiologists. So without you, it is difficult. And also another thing, in entire India, so Tamil Nadu played a very good important role in controlling this pandemic, because Tamil Nadu have a very good health system infrastructure, and also top universities. So this is one of the major reasons for controlling this pandemic. Okay, so I'm from South Korea. So many people know. So next to China, South Korea is the second major country which highly affected with COVID-19 coronavirus. Even though it has been affected with coronavirus, and South Korea is one of the best country which controlled the virus within a short time. So the first case reported in South Korea in January 30. In March, April, it was an epidemic. It was became an outbreak. And June, July, within this 2 months, April, May, June, within 3 months, South Korea completely controlled. At least they have controlled the coronavirus spread, because they followed the 3 techniques: testing, treating, and tracking. So testing means they did large analysis of sample, PCR samples. So they use the testing, and treating once by using the PCR kit, and also normal rapid detection kit. So they identify the patients who have corona positive or not. If the patient is corona positive, so they used to go for treating. And if the infected person is corona positive, they used to track the person. So where the positive patient he went, whether he went to the hotel, he went to the restaurant, he went to the church, or he went to the university, by using like a credit cards, or by using the mobile phones, by using the GPS facilities, they used to track. After tracking, they isolate the person. I think now everyone is common. Most of the people you can understand. So these are the 3 successful techniques South Korea handled. And even in BBC, I think in April, I gave an interview about how South Korea, they control the coronavirus. Okay. So most of the people knows that like South Korea is a well advanced country and also they have very good advanced technology. So it is one part. Okay. So during this COVID time, what I observed. So one part I observed, they have a very good mass sampling, mass testing and treating and tracking facilities, advanced facilities. This is one part. And another part, what I observed is automatically the functional foods like a traditional medicinal food products has been, the consumption of medicinal food products has been increased in another side. So this is the one of the major paper magazine that Korea Herald. So you can see the functional foods like health functional foods see boom in way of virus outbreak. So only during this pandemic period, even in India also you can see. So many people started drinking kapasuba kudini or niloebu or ginger or garlic or curcumin like something. So same thing in Korea also it happened. You can see. So in South Korea, the one of the major herbal is ginseng. You can see this is the ginseng. It is like similar to our ginger. Okay. So this herbal medicinal plant, they have a very good traditional medicinal properties like antioxidant, anti-inflammation, and anti-diabetic and anti-cancer property. Also, it is very good immunostimulant activity. It boosts your immune cells. It boosts your immune system. நம்மளோட நோய் எதிர்ப்பு சக்தி அதிகரிக்கும். And here you can see during this, so I belong to the 80 kids. So most of the 80 kids or 70s or 60s, that time India is economically low. It's a poor country. So that time we don't have very good infrastructure. Particularly in Tamil Nadu is okay. In most of the states, still they don't have the proper medical facilities and also they don't have the proper infrastructure. So since the condition is like that, so countries like India and most of the developing countries, we depend upon the herbal medicine or traditional medicine. Okay, so you can see. So during this chikungunya outbreak and also during the COVID-19 period also, you can see many people, they started drinking the kabasabu kudinir, niloembu. So, and this herbal drink or this kind of medicinal plants like niloembu, it has been reported to inhibit the virus. Okay. So, as I mentioned earlier, so now only during this COVID time and during this lockdown period only, many people, they started learning about our herbal medicine. So is this medicinal plants and this kind of herbal medicine is new? No. And this kind of herbal medicine we have been practiced for 3,000 to 5,000 years before. If you, here I want to mention one poem from the Sangam literature. So, Thingani Iromed Vembu Manasari. So it means in ancient time, our ancestor, so it is a song from Purananuru. Thingani Iromed Vembu Manasari. It means, so prevent the viral infections or any bacterial infections, our ancestors, they have used the neem extract or neem leaf. Okay. So this is a song. And also I need to quote another song from Nattinai. Maramsa Marundam Kollar Mandar. So it means how you can see here I need to point out how our ancestors, they have protected the herbal medicine. If you want to tell in Tamil, so there is a king. Okay, ஒரு அரசர் இருக்கிறாரு. அந்த அரசருக்கு ரொம்ப பயங்கரமான நோய் வந்துருச்சு அவருக்கு. அப்ப அவருக்கு உடனே வைத்தியம் பார்த்தாகணும். அப்ப அங்க இருக்கிற ஒரு மந்திரி என்ன பண்றாரு, மந்திரி கூப்பிட்டு ஒரு வைத்தியரை கூப்பிட்டு சொல்றாரு. அப்ப வைத்தியர் வராரு. வைத்தியர் வந்துட்டு என்ன பண்றாரு, அந்த அரசருடைய நாடியை பிடிச்சு பார்த்துட்டு, இவருக்கு உடனே நீங்க தோட்டத்துல போயிட்டு ஒரு மரம் ஒரு செடி இருக்கும், அந்த செடியை பிடுங்கி அந்த வேர் இருக்கும், அந்த வேரை அந்த வேரை வந்து நல்லா பொடியாக்கி அதை நல்லா பிழிஞ்சு அந்த சாரை எடுத்து நீங்க கொடுங்க. அப்ப அந்த மன்னர் சொல்றாரு, அப்படி அந்த மரத்தை அந்த செடியை அந்த மருத்துவ குணம் உள்ள மூலிகைய அப்படி சாகடிச்சுதான் எனக்கு என்னுடைய உயிரை காப்பாற்றணும்னா அப்படிப்பட்ட உயிர் எனக்கு தேவையில்லைன்றாரு. ஓகே, so this is the simple one of the best example I can say how our ancestors, they have protected our traditional medicine and also the medicine value. Okay, So how medicinal plants can improve our immune system? So you can see, so this kind of medicinal plants, usually plants are very rich in flavonoids and vitamin C. So vitamin C is very is important to enhance our immune system. So it is important. That's why most of the children's kids or most of the adults or many people, we used to take vegetables, fruits, colorful fruits or vegetables like that. So you can see here the king Tondeman Tondenatumanan. So the king Adiyaman. So he gave gooseberry as a Nellikani. So Tondenatumanan Adiyaman Nellikani Petravar. So why he gave this Nellikani to the Abhayar? Because this Nellikani, it contains high vitamin C. It increases the immune system. If the immune system is good, you can extend your life. Okay, so that is the common thing. So apart from this thing, this kind of herbal medicine, they also possess a very good anti-inflammation property, and also it acts as an immune stimulant. It boosts our immune system. It increases our immune system, and also it promotes the activity of lymphocytes and increase the phagocytosis and induce interferon production. And here, this is important, interferon production. So most of the herbal products, most of the medicinal products, they play a very good role in inducing the interferon production. Okay, so what is interferon? You can see. So interferons is nothing but it is important protein molecules. So mainly is to trigger the immune system. Okay, if there's any antigen, viral antigen or bacteria or any infection enters into our body, okay, our body will produce interferon, and this interferon will activate the immune cells. Okay, so what are the immune cells? Like natural killer cells or macrophages, like so those kind of cells, and it will stimulate. Oh, you can see like the interferon will say the interferon will send message to our immune cells. You can check antibody antigen has been entered, bacteria has been entered in your body, virus has been entered in your body, go and attack, like that. Okay, so as I mentioned earlier, so most of the herbal medicinal plants, they activate the interferon productions, and interferon production, apart from the activating the immune cells, they have the very good ability to interfere with the viral replication. It stops the viral replication even if the virus entered. If you take the herbal drink regularly, like Nilayambu Kudinir or Kabasavu Kudinir regularly, it stops the viral replication. Okay, so recently I made a publications on Kabasavu Kudinir. I made two publications. So finally we found out this Kabasavu Kudinir has the ability to stop the viral replication. So I will discuss it later. Okay, and also once the interferon production is high in the body, so what are the general symptoms? You can see fever, muscle pain, and flu-like symptoms. So if that is interferon production, so these are the symptoms. So that is the reason if you have any viral infection or viral attack, if you have any viral attack, maybe a dengue virus or like a COVID-19 virus or any type of SARS virus, our body will produce interferon production. Once the interferon production is activated, these are the symptoms. You have the fever, muscle pain, and flu-like symptoms. So this is the reason. Usually now most of the supermarkets, most of the people they're checking your temperature. Okay, so if your body temperature is increased, it means your body has some type of infection. It means your body is different. So that is the reason. Okay, so coming to the COVID time, so WHO has declared the coronavirus. COVID means coronavirus 2019. Okay, to be pandemic. So it affected more than 200 countries. Around 120 million people has been affected and killed more than 2 million people throughout the globally. And in India, the first case has been reported in January 13, originated from China. So till date, you can see around 11 million people has been affected with COVID-19, and the recovery rate is higher. So usually compared to other countries and India, even the infection rate is higher, you can see the recovery rate is high. So what I feel, because this kind of high recovery is mainly most of the Indians, we practice herbal medicine, traditional medicines. So that is the main reason. Okay. So this COVID-19, okay. So this COVID-19 is caused by the SARS type of virus, severe acute respiratory syndrome type 2 virus. So among this kind of coronavirus, there are some types of human coronavirus, alpha coronavirus and beta coronavirus, and this COVID-19 comes under this beta coronavirus. So compared to alpha, this beta coronavirus, it is more virulent, and more virulent and has the highest mortality rate. So coming to the nucleic acid, so it is a RNA virus. So now we know the sequence. So previously in 2020, from January, that time, so they identified the SARS coronavirus is similar, the genome sequence is similar to the SARS coronavirus. So based upon this sequence only, many countries like China, Korea, they developed the PCR kit. So by using the PCR kit only, RT-PCR kit only, they identified, they used as a diagnostic tool. Okay. So next, the important, this is the structure of coronavirus. So the main thing, the importance, what are the structures of coronavirus? You can see it contains a spike protein and nucleic acid and envelope protein and membrane protein. So usually this spike protein play an important role in viral attachment. Okay. So in viral attachment, the spike protein is important. So this is the spike protein, you can see. So what is the main role for the spike protein? The spike protein, it plays an important role for binding of host cell receptor. Okay. So this spike protein will go and attach to the host receptor. Okay. So இது ஒரு வைரஸ் இருக்கு, அந்த வைரஸ்ல ஸ்பைக் புரோட்டீன்ற ஒரு புரோட்டீன் இருக்கும், வெளியே இருக்கும், அது முள்ளு மாதிரி இருக்கும். அந்த புரோட்டீன் என்ன பண்ணும், நம்ம உடம்புல லங்ஸ்ல, நுரையீரல்ல இருக்கும், கிட்னில இருக்கும், அங்க வந்து ACE2 ரெசெப்டார்னு இருக்கும். அந்த ரெசெப்டார்ல போயிட்டு அட்டாச் பைண்ட் பண்ணிக்கும். பைண்ட் பண்ணிட்டு இதோட நியூக்ளிக் ஆசிட் உள்ள போய் இன்சர்ட் பண்ணிவிடும். So this is the main thing. Okay. So this is the major part, like the structure of proteins. For regarding coming to the COVID-19, so most of the drug and pharma companies are vaccine even for vaccination also. The target protein is 3CL Pro. Okay, 3 chymotrypsin like protease. So what is the main function of this chymotrypsin or 3CL Pro? This 3CL Pro play an important role in controlling the viral replication. So therefore, the pharma companies, the vaccine, drugs or anything, okay. So if you give a drug, if I take a drug, if I'm infected with any type of coronavirus or any virus, okay. So if I take this drug, any drug, any medicine, so that drug, that medicine will stop this 3CL Pro, okay. So once this 3CL Pro, this protein, the 3CL this, once it stops the protease enzyme, so what happened? The virus will not have the ability to multiply. It cannot replicate. Okay, so that is the thing. Okay, so here as I mentioned earlier, you can see this is the spike protein, this is the coronavirus, and these are the spike protein. Okay, and here you can see this is the ACE2 receptor. Okay, so where this ACE2 receptor found? This ACE2 receptor found in lungs, gastrointestinal tract, kidneys and blood vessels. So that is the reason COVID-19 coronavirus highly affects the people who have respiratory problem, who have kidney issues, and who have some blood pressure issues. This is the reason. Okay. So now we understand what is ACE2 receptor. So ACE2 receptor, it plays an important role in RAS pathway. It means renin angiotensin aldosterone system. Okay, so the major function of this ACE2 means mainly it controls a pathway called renin angiotensin aldosterone system pathway. What is the main function So what is the role of this thing? So these renin-angiotensin aldosterone system, they play an important role in regulating the process called blood pressure. It regulates the blood pressure, our blood flow. It regulates the wound healing. If you have any infection, it will heal the wound. An inflammation process. Okay, so this is the thing. And also exchange of oxygen and carbon dioxide between the lungs and blood vessels occur during the epithelial linings of the cells. And also this ACE2 present in the epithelial linings in the lungs. Okay, நம்மளோட நுரையீரல்ல ACE2 receptors நிறைய இருக்கும். இந்த ACE2 receptor எங்க அதிகமா இருக்குன்னு பாத்தீங்கன்னா நம்ம லங்ஸ் நுரையீரல் இருக்கும், குடல், அதுக்கப்புறம் கிட்னி, அப்புறம் blood vessels அது அங்க அதிகமா இருக்கும். So this is the thing. So since if you have infected with coronavirus, the spike protein go and bind to the ACE2 receptor. When this spike protein go and bind, if the virus bind to this receptor, what happens? It will automatically insert RNA and it will multiply the RNA replication. This is the one. This is the one point. And second point, if the number of coronavirus is high, is increased in our body, if it binds to the ACE2 receptor, it will block the function of this ACE2. The first point, what is the function of ACE2? Mainly, it controls the RAS pathway. What is the major function of RAS pathway? It controls the blood pressure. It increases the wound healing and also inflammation. Once this spike protein or this coronaviruses go and bind to the ACE2 receptor and it blocks the function, automatically the patient will experience the breathing problem, breathing difficulty. So that is the reason many COVID patients, they have the breathing difficulties. This is the reason. And also another reason, for normal or healthy person is okay. So those people who already take, who have kidney issues, kidney problem, or blood pressure problem, or heart patient, they used to take some tablets. They used to take some medicine. So if they take the medicine, automatically it activates the ACE2 receptor. So once the ACE2 receptor is activated, the COVID virus enter. Once the COVID virus enter in our body, it go and bind to the receptors. So this is the thing. Okay. So recently I published an article. It is a Korean herbal, Gerania herba. It is a Korean medicinal plant. It has the ability to inhibit the SARS coronavirus 3CL Pro. As I mentioned earlier, what is 3CL Pro? This 3CL Pro, okay, so mainly involved in the viral replication. It controls the viral replication and also spike protein. Spike protein, this is the spike protein. Okay, so here we can see. So this is the life cycle of COVID-19 virus. So this is the coronavirus structure, spike protein, and this is, you can see, this is spike protein, red color, and membrane protein. This is the membrane protein, and here it is envelope protein, and this is a nucleic acid, RNA virus, and here it is ACE2 receptor. And this ACE2 receptor found mostly in lungs, kidney, and other things. So once the virus enter into the body, this is a spike protein. The spike protein will go and attach to the ACE2 receptor. Once it attaches to the ACE2 receptor, this nucleic acid is released into the cell. So once it is the nucleic acid RNA viral genome is released, it undergoes the replication process. Okay. So for this replication process, it requires the 3CL Pro and this thing 3CL Pro. Okay, 3CL Pro. So once it is replicated and it go for translation process, in this translation process, it will produce M protein, spike protein, envelope protein, and nuclear protein. Next, in endoplasmic reticulum, it makes from it go for maturation and for assembly. Next, it will be released. So this is a process. So this is a process if the patient is infected. Okay. So now what happens if you take some herbal drug or if you give some medicine? Okay. So we identified this is the Gerania herba. It is a Korean herbal drug. And this herbal drug, it played a two important role. The first thing, this active compounds present in the Gerania herba, it go and bind to the spike protein. So once the spike protein is attach or block, the virus cannot attach to the ACE2 receptor. This is the point one. And another point, it blocks the 3CL-Pro mechanism. So once the 3CL-Pro mechanism is stopped, even the virus is infected in our body cells, it cannot replicate, it cannot multiply. Okay, so here you can see multiplication, it cannot multiply. So the virus enter into the body, so it cannot multiply. So automatically, what happens? Now, what are the other functions of the medicinal plants? It increases the immune system. It activates the immune cells like macrophages and neutrophils. So those kind of macrophages, it functions of a phagocytosis. It kills the virus. So this is the mechanism. Okay. So this is general viral life cycle thing. Initially it attaches the host cell. It enters the virus. Then it releases the genome and viral replication of viral genome and protein synthesis, processing and assembly. This is the process. Okay. So once the patient is infected, this is the COVID patient. Okay. So he is a COVID positive patient. So, once if you have once if body is infected with COVID-19 or any type of virus, your body automatically activates the interferon production. So as I mentioned earlier, once the interferon production is activated, you have fever and also coughing or sneezing. Okay, so that is the reason most of the people, if you have infected with virus, if you have cough. Once if you cough, what happens? The virus will be circulated in the aerosols. Okay. So here what I mentioned here. So this is the spike protein, and this is the COVID coronavirus positive patient, and he is negative. Okay. So he don't this positive patient, sorry, this COVID-19 negative patient, he didn't use any face mask. So once if he didn't use face mask, the COVID-19 positive patient, he cough or sneeze. So once he cough or sneezing, so the virus particle will be surrounding, it will be dispersed in the air, in the atmosphere as aerosols or droplets. So it can easily enter into the host, any negative patients. Okay. So once if you don't wear the mask, what happens? So usually this coronavirus, SARS-CoV coronavirus, it go and bind to the ACE2 receptor. And where this ACE2 receptor is present, this ACE2 receptor present in the lungs. That is the reason. Okay. So once if you don't use the face mask, this COVID-19 automatically attached to the nose epithelial cells, lungs, and it can easily multiply. So this is the main reason you need to use face mask. Okay. So I explained about the viral mechanism. So most of the drug companies, they target at initial time at like a viral entry, and also during a replication and transcription process, and also for viral release. So these are the drug target. Okay, so like a fusion viral inhibitor. These are the inhibitors. So still, we are fighting with the COVID. Even we have vaccination, but still we are fighting. So as I mentioned earlier, so to stop or to prevent the infections, we need to always wear the face mask. And if you have any infection, you need to use the face mask to prevent the spread of infections. Okay. So what are the major symptoms? So usually, major symptoms like breathing difficulties, fever, head pain, and also for other things. So during that period, so now we have vaccine production. We developed the vaccine. During last year, from March to December, we didn't develop the proper vaccine. Okay, so during that time, how we prevented the COVID vaccine means how we prevented the coronavirus. So it is the use of herbal medicine we protected. So this is the thing. So most of the herbal medicines here you can see for coronavirus. So what are the, so now we have vaccination. So previously, what are the treatment they use? Only symptomatic relief is provided. In Tirukural, they used to say, Noi Nadi, Noi Mudal Nadi, Adutanikum, Vainada, Vainadi, Vaipet Sayal. Okay, for what are the symptoms, and for the symptoms they are giving treatment. If you have fever, they give some like paracetamol. If you have some viral infections, like they give some antiviral drugs like that. Okay, if you have some respiratory problem, they used to keep in ICU and with all breathing facilities like that. Okay, only symptomatic relief is provided to the patient. Okay, and previously, what are the drugs? They use antiviral drugs and also antibodies, like for the antibodies which are used for MERS coronavirus and SARS coronavirus, and also for combination therapy they use for anti-malarial drug also they use, like a hydroxychloroquine and azithromycin. And even these drugs are they can stop they can protect from the COVID infections during that time, but the problem is it made some lot of side effects. Okay, here you can see there are some reports shows that drug hydroxychloroquine is not effective for those infected with coronavirus and shows adverse effect. It shows some, so those people who have already infected with some kidney problem or heart problem, so they have for them this drug is not responsible. Okay, so and these are the symptoms of COVID, like headache, cough, muscle pains, and fever, shortness of difficulties. Okay, so previously, what treatment they give means if you have fever, if you have a headache, if you have cough and you have muscle pain, they give treatment only for the symptomatic. That is the reason I mentioned only symptomatic relief is provided to the patient. Okay, and here now we develop the vaccines. Usually the vaccines like the DNA vaccines, they used to take or RNA, mRNA vaccines they used, and also some protein, some subunit of viral protein. They use a spike protein. They used to take the mRNA of the spike protein and they developed the antibodies like that. So these things. And these are the companies so far they developed the vaccination, like Oxford, AstraZeneca, Moderna, Pfizer. So these are the things, vaccines they developed. In India, we have Covaxin and Covishield. Okay. So now we have vaccination, but even there is some reports, it shows that this vaccine, AstraZeneca, in Netherlands, it does, and also in Germany, it's in some countries, it shows it has been reported this vaccine shows some side effect, so it has been stopped. Okay, but now we have vaccination. During that time, most of the countries, so they come like Korea, India, China, like most of the countries, developing countries, they completely depend upon the alternative medicine that is called herbal medicines. Okay, so there are herbal medicine. There are many types of herbal medicines like we call Siddha, Ayurvedic, Unani, Chinese medicine, and Arabic medicine. So this kind of herbal medicine and traditional medicine, it can differ from country to country, and also it can be influenced by the various culture and history and personal attitude and so. For example, in Tamil Nadu we practice Siddha medicine, and if you go for North India, they use Ayurvedic medicine. So that is the thing. And you can come into the literature, you can see there are several reports. Like this is my publications. This kind of herbal medicine, Marcella marita, ambret seed oil. So this kind of herbal medicine already we have reported to be antibacterial, antioxidants, and so on and so. And in 2020, like last year, you can see there is a report from the Chinese Journal of Natural Medicine. So they use a lot of traditional medicine to protect from the COVID-19. And this is apart from the COVID infections, herbal medicine can be used to control the bacterial infections and other type of infections. Okay. So why this kind of herbal medicine is important? As I mentioned earlier, so if developed countries, okay. So most of the developing countries, they don't have the proper medicinal facilities or proper infrastructure. So they completely depend upon the herbal medicine to protect from the viral infection or any type of infection. And another part, coming to the India, so herbal medicine, it's a very big market. Economically, it contributes high economy to the India. You can see in 2005, around $6 billion. So in 2020 at 50 billion, $55 billion. So most of the pharma companies, they completely depend upon this our traditional medicine. They take the main product, the raw material from the thing. And you can see China already they so now they have vaccination. So before that, during the pandemic, so China they used herbal medicine, and also some people they started the trial. And also you can see in India, you can search for immunity powers of the sales of Ayurvedic products. So most of the Ayurvedic product like the curcumin, cumin, so most of the herbal product, the price also has been increased. So during the pandemic period. So why? So now almost it takes around 1.5 years to develop the vaccine. Even people, they are afraid to take the vaccination because the problem is, this is the thing. So usually if there is for example, so here I reported this plant, Marcella minata, it has a very good antibacterial activity like this. Okay. Like here this plant, Allium sativum, it can prevent or it can stop the dental pathogens. This is my report. Okay. So generally, if the product, if the compound it should come into the market, it will take around 5 to 10 years. Why means initially for screening around 5,000 or 10,000 plant has been screened for any viral infection or any for bacterial infections. Out of 5,000 to 10,000 plants, 250 plants will go for the preclinical testing, like maybe animal trial. Okay, so out of 250, 5 will enter the clinical trial. Clinical trial means like for human trial, phase 1, phase 2, phase 3. Okay, so now what for Covaxin or Covishield for vaccination, even we developed the vaccine from the vaccine for coronavirus. So now many people they are afraid. Why means because lack of clinical testing. Okay, so that is the reason. So for completely out of 5,000 to 10,000 plants, it only one compound will be approved by the FDA drug, and it will take around 0 to around 10 to 15 years. So this is the thing. And what are the different phases of clinical trial, like phase 1 trial, phase 2, and phase 3? Like phase 1 trial, so once if you develop the product or vaccination, they will give the drug to the 20 to 80 volunteers, and they will check whether if they're giving the vaccination or if they're giving any drugs, they will check the control patients and positive patients. Okay, and they will check whether after giving the vaccination or after giving the drug, they will check whether they have headache or they have any fever or they have any adverse side effect, any other allergic problem like that. Okay, so after screening, they will increase the process for 100 to 300. Next for phase 3, it goes for 3,000 to 5,000 period. Okay, so this is the thing. So what is the problem in now currently? Most of the people, they are afraid for going for vaccination or anything. This thing, the safety thing. So this is the reason it took for developing of one single good product. It takes around minimum 10 years. Okay, so during that period, even now for vaccine for COVID time, almost it takes 1 year. Now we have many vaccination, but it takes 1 year. But within 1 year, how people they have protected? People have protected by they have adapted, they have used by using the herbal products. So this is the thing. Okay, so coming to the Tamil Nadu, what is the Siddha medicine? Many people they use the Siddha medicine. So what is the Siddha medicine means? It is a use of specific polyherbal formation. Poly means many, herbal. It is a combination of many herbal things. So already there are several reports of Indian medicinal plant used to treat the SARS coronavirus infections. For example, Andrographis paniculata. So it is called Niloembu. In Tamil, it is called Niloembu. And this Niloembu has been reported to inhibit to suppress the SARS coronavirus and also the SARS coronavirus 2 activity. Okay, so this thing. And also another plant, glycerin, it is this plant compound has been reported to inhibit the viral replications on SARS associated virus. And you can see this Niloembu Kudinir. So during this chikungunya, even that time also we also face the same problem. We don't have any proper medicine or proper vaccination. So our Tamil Nadu government and many people, they voluntarily they intake the Nilavembu Kudineer. Because Nilavembu Kudineer, we don't know that Nilavembu Kudineer doesn't have any medicinal properties. People used to take, but scientifically it has been reported this kind of Nilavembu Kudineer has the ability to prevent kill the chikungunya and dengue virus infections. Okay. Next, apart from antiviral effect, this kind of herbal drug like Nilavembu Kudineer and Kabasubu Kudineer, they have the ability to stimulate the immune cells, particularly macrophage, neutrophils, and dendritic cells. Okay, if any viral antigens or any bacterial antigen enter into the body, this, if you drink the herbal drink like Nilavembu or Kabasubu Kudineer regularly, so your immune system will be increased. That is the reason. Even that is the main reason in India. Even it is high infections, the recovery rate is high. So that is the main reason. And this report, these findings, we made a publication and we published. So okay. So for leave about the Nilavembu Kudineer. So here in COVID-19, so most of the people, particularly in Tamil Nadu, even the Tamil Nadu government, they recommended the Kabasubu Kudineer. Okay. So here, this Kabasubu Kudineer contained 15 compounds, and these 15 compounds are effective for already reported in controlling the fever, cough, sore throat, shortness of breath. Okay, and these fever, cough, sore throat, and shortness of breath, if you infected with SARS coronavirus, COVID-19, these are the symptoms. Okay, so already this Kabasubu, why they use Kabasubu Kudineer means Kabasubu Kudineer is used for preventing the fever, cough, and sore throat and shortness of breath. And these symptoms, if you have COVID infection, it can occur. So this is the main reason. This is the main reason, like government of Tamil Nadu and also for Ayush, Minister of Health, they recommended the Kabasubu Kudineer. This is the main reason. Okay. So next, before going for any research, so you need to go for recent technology or modern technology. We need to check. So if you make a literature search, you can see more than 1,800 research paper has been published based upon antiviral plant. You can see by using the herbal plants. So many research article has been published, and so as I mentioned earlier, you can see here out of 5,000 or 10,000 screening process, at least you can get one or two or three products. So this is the main thing. So people who want to do some research, good quality research, you should have a very good literature survey. So why people go for plant-based drugs? Because they are safe compared to the synthetic drugs. Compared to the synthetic drugs, here I mentioned already for COVID-19 they use hydroxychloroquine and azithromycin, and this it is not effective for the kidney failure patients. Okay, but this kind of herbal drugs it is less compared to the synthetic drugs. It is less toxic, and also it boosts the immune cell. It acts as a very good immunostimulant. This thing, and here in Nilavembu the major compound is anthragliflozin, and here you can see the curcumin, manjal. The major compound is turmeric powder. The major compound is curcumin, and this curcumin has been reported as a very good immunomodulator. It can act as a very good. It can boost the immune cells. So you can say, so just we don't have any report like for herbal medicine. If there is any, you can ask me if there is any product. Yes. For example, Haemophilus influenza. Okay, so now common cold. So the Haemophilus for this influenza virus, there is a drug called Tamiflu. Okay, so this Tamiflu, it acts as a very good. It can control the. It acts as a neuraminidase inhibitor. Okay, so it controls the viral replication. So where this drug comes from? This drug initially it comes from the plant product. It's called Elysium verum. Okay, so this Tamiflu drug has been isolated has been isolated from the plant called Elysium verum. So this is the thing. Okay, so this is the main reason India has a very good big pharma companies, pharma economy. Okay, so how these kind of antiviral, antibacterial mechanisms happen? Because plant contain high like secondary metabolites. So what is the major important role of secondary metabolites? Things you can see there are many type of secondary metabolites: terpenes, phenolic compounds, and nitrogen containing compounds, and sulfur containing compounds. And these special individual metabolites, they have specific role in controlling the virus. For example, terpenes. So these terpenes, they have a very good antiviral mechanism, and it has been used as anti-malarial drugs such as anti-medicines, and also other related compounds, and also mainly particularly in essential oils. Okay, in oils, this kind of terpene compound as we can find, and this kind of essential oils has been already reported anti-H1N1 influenza virus and also HSV virus. It has been reported, and it has been this kind of terpene compound has been reported to control the virus. And phenolic compounds, and this kind of phenolic compounds already reported, they have very good scientific publications to against HIV, HSV, dengue virus influenza, and also the polio virus. And most of the pharma companies and most of the herbal drugs, they take this kind of phenolic compounds because more than 6,000 compounds have been identified. Okay, identified. And here particularly, so these kind of flavonoids, they have a very good, they can stop the viral replication. Okay, so here it has an article, another article. This kind of flavonoids, flavonoid compound, they can prevent the viral attachment. There are several mechanisms of flavonoid mechanism. The first one is it can prevent the viral attachment or viral entry, and it can prevent the viral replication at the early stage. And also, once even if the virus has been inserted into our body, if there is any, it can stop the transcription and translation process. And also, even after escaping from the translation process, the virus can stop the packaging and release of the thing. And also, it can flavonoids can increase the immune cells. So that is the main reason you can see many pharma companies they target the flavonoids. And recently, you can see this SARS coronavirus, it has the ability to stop the 3CL Pro mechanism. So once the 3CL Pro mechanism is stopped, viral replication is stopped. So flavonoids has a very good excellent source. Next, alkaloids. Alkaloids also has been reported to inhibit the virus. So you can see here the curcumin. Okay, so normal manjal. So curcumin plays an important role. So this type of the flavonoid compounds or phenolic compounds, what happens if the viruses enter into the body, it stops the transcription translation process. But the main advantages of curcumin, it will never even, for example, if the viruses enter into the body, it will, the curcumin compound, the curcumin will not allow the virus to attach to the host cell. That is the main function of the curcumin. Okay, so and also it acts a very good agent as a virus. It will kill that virus. Okay. So as I mentioned earlier, in COVID, they used asymptomatic management. Noi nadi, noi mudal nadi, adu danikkum, vai nadi, vai pachayil. Okay. So these are the, for COVID time, for coronavirus, these are the our Siddha medicine and Ayurvedic herbal medicine they used for the prevention of coronavirus. For example, Androcopis planigrata, it is called Nilayambu, it is from Siddha medicine. The one thing is, we need to take only 60 ml. So, but many people, they used to take more than 60 ml. No. Okay. So if you want to have a very good immune system, and also if you want to prevent any viral infection or any type of infection, you need to drink Nilayambu Kudineer and also Kabasabu Kudineer only 60 ml. Only 60 ml is recommended. Okay. I can find many people, they used to drink like liters of liters. Once if you consume high amount of this kind of plant compound, it will affect the kidney and liver also. அளவுக்கு மீறினால் அமுதும் நஞ்சி. So that is the thing. Okay, the recommended dose is நிலவேம்பு. So according to the oils, they recommended anti-herpes splendica, நிலவேம்பு குடிநீர், like it control for fever and thing, and ஆடாதோடா. Okay, it can control the respiratory infection. So best example is we can see now we can have the ஆவி பிடித்தல். நம்ம இப்போ சளி வந்துருச்சு, நெஞ்சில் சளி வந்துச்சுன்னா நம்ம என்ன பண்ணுவோம், போய் ஏன்னா ஒரு தைலம் போட்டு ஆவி பிடிப்போம். ஓகே, அதுதான் மெயின் ரீசன். ஸோ இன்னும் கிராமத்தில் பார்த்தீங்கன்னா அந்த ஆடத்தோட இலையை எடுத்து நல்லா கொதிக்க வச்சு ஆவி பிடிப்பாங்க. எதுக்கு ஆவி பிடிக்கிறாங்கன்னா ரெஸ்பிரேட்டரி இன்ஃபெக்ஷன், நம்ம லங்ஸ்ல எதனா பிளாக்கேஜ், வைரல் பிளாக்கேஜ் இருந்துச்சுன்னா அது கிளீன் பண்ணிவிடும். ஸோ தட் இஸ் த மெயின் ரீசன். Allium sativum, this is garlic. Okay, you can see this, it has ability to inhibit the SARS virus, anti-herpes splendica. So there are several things. So Boveria, diffusa, Coryndium sativum. So most of the herbal drugs, they have the target of ACE2. Okay, what is the main function of ACE2? Okay, so this ACE2 mainly important for controlling the blood pressure, oxygen flow. Okay, so that is the thing. So particularly this coronavirus, what is the binding site? The coronavirus, the spike protein go and bind to the ACE2 receptor. So once if you take this kind of garlic, like Allium sativum, nilavembu, anti-herpes splendica, and this kind of Coryndium sativum. So these kind of herbal plant, if you take as a medicine, it can prevent the viral entry. And Glyceria glabra. So these are the several medicinal plants. These medicinal plants, they have a very good antiviral mechanism. You can see. So, and here this is another compound called Geranium sanguineum. So this plant has been reported for influenza virus, and for SARS virus, you can see Lycoris radiata. The main compound is lycorin. This compound has been an anti-SARS coronavirus. And Phyllanthus amarasis. Okay, this is the common plant. Many people know as. I think Kidanalli, Phyllanthus amaras. It inhibits HIV replications both in vitro and in vivo. And also there are several reports. This kind of herbal medicine, they can prevent the viral replications. Okay, and now we can see many studies. There is no time. Okay, so now at present we don't have any time for developing the drug. So many researchers, they use molecular docking methods for identifying the potential target. The target for the main target for the coronavirus is spike protein, 3CL Pro, and also some other nucleic acid, other thing. So particularly many researchers, they target the two protein like spike protein and 3CL Pro. So based upon the target, so once if you identify some plant like a plant compounds or anything, you can go for docking study. Okay, so even we have very good positive effects on many positive side for herbal medicine. The thing is, the Food and Drug Administration, FDA not approving the herbal medicine because we don't have the proper we don't have the proper data. For example, if I take for any herbal medicine for my body, for my health condition, the Kabasabu Kudini or any herbal medicine is working. But the same concentration, if I take 60 ml, if I give the same 60 ml concentration to another person, it will not work. It depends upon the person's body weight like that. So we need a proper data, and we need to conduct many experiments. Once if you conduct the many experiments only, we can we can identify what is the right dosage and what are the side effects and other things. Okay, so coming to this conference, so what is the main outcome means, and also what during the corona period, what we learned, both the traditional medicine and modern medicine, they need to work together. So traditional medicine during the COVID time, we don't have any proper antiviral drug. We don't have any vaccination, nothing. But we know that Kapasaba Kudinir can prevent the virus. That is the thing. So that is the traditional medicine. And modern medicine. Okay. So what is the modern medicine? Means in Kapasaba Kudinir, there are a lot of compounds there, like more than 145 and 150 compounds there. Out of 145 compounds, not every compound will not be active. Okay. So what happens? Like modern medicine, like universities, like any universities, research scientists, they know they need to work together. So why they need to work together means traditional medicine, for example, these are all common rural people. So they know what type of plants they can identify. Okay, இப்ப ஒரு எடுத்துக்காட்டா பாத்தீங்கன்னா இப்ப கிராமத்துல இருக்கிற வைத்தியருக்கு வந்து எந்த நோய்க்கு எந்த தாவரம் யூஸ் பண்ணா உபயோகிக்கலாம் அவருக்கு நல்லா தெரியும். ஆனா அந்த அந்த தாவரத்துல என்னென்ன மூலக்கூறு இருக்குன்னு அவருக்கு தெரியாது. இது ஒரு பாட்டு. ஆனா பக்கத்துல ஒரு பல்கலைக்கழகம் இருக்கு, பாரதியார் பல்கலைக்கழகம் இருக்கும், பாரதியார் பல்கலைக்கழகம் இருக்கும், மதுரை காமராஜ் ஏதோ ஒரு பல்கலைக்கழகம் இருக்குன்னு வச்சுக்கலாம். இப்போ உங்களை போன்ற பேராசிரியர், உங்களை போன்ற மாணவர்களுக்கு உங்களுக்கு அந்த காம்பவுண்டை பிரித்து எடுக்கிற தெரியும். அது எப்படின்னா HPLC யூஸ் பண்ணுவீங்க, GCMS யூஸ் பண்ணுவீங்க, NMR யூஸ் பண்ணுவீங்க. அதை யூஸ் பண்ணிட்டு அந்த என்னென்ன காம்பவுண்ட் இருக்குன்னு கண்டுபிடிப்பீங்க. அந்த காம்பவுண்ட் எடுத்துட்டீங்கன்னு வச்சுக்கலாம், அது எடுத்து நெக்ஸ்ட் அது மார்க்கெட் போகும். அதுதான். ஸோ இப்போ நம்ம இந்த கொரோனா டைம்ல நம்ம என்ன மரபு வழி மருத்துவம், நம்ம நவீன மருத்துவம் ரெண்டு இருந்து செயல்பட்டாதான் இது போன்ற கொரோனா போன்ற பேரிடர் காலத்தில் நம்ம தப்பிக்க முடியும். ஸோ the outcome is both the traditional medicine and modern medicine, they need to work together for successfully controlling this kind of pandemic thing. So based upon this, so recently I published 2 articles and Kawasaki coordinator, and this is the first article, I think. First paper we used, and our findings has been published in many news channels, okay, in Korea and also in India. You can see this is the Korean newspaper, and this is in India. So I made a collaboration with the Royal College, you can see. So we found that in Kawasaki Kudineer, 145 compounds, out of 145 compounds, 15 compounds, they are highly have the ability to stop the viral replication. இதான் மெயின் ரீசன். ஏன் நம்ம ஊர்ல வந்து குறிப்பா தமிழ்நாட்டுல நோய் வந்தாலும் அவங்களுக்கு இறப்பு விகிதம் ரொம்ப கம்மி, அதே போல சீக்கிரமா குணமாயிடுறாங்க. அதுக்கு என்ன காரணம்னா கபசுப்பு குடிநீர்ல 145 மூலக்கூறுகள் இருக்கு. அந்த 145 மூலக்கூறுல 15 காம்பவுண்ட் பார்க்கும்போது அந்த வைரஸ் பெருக்கத்தை தடுக்குது. அதாவது எனக்கு வந்து கொரோனா வைரஸ் இன்ஃபெக்ட் ஆனாலும் ஒரு 7 நாள்ல அந்த வைரஸ் பெருக்காம தடுத்துருது. வைரஸ் பெருக்காம தடுக்கிறதுனால அந்த இருக்கிற ஒரு 10 வைரஸ் உள்ள வருதுன்னு வச்சுங்களேன், அந்த 10 வைரஸ் உள்ள வந்து ரெப்ளிகேஷன் அது பெருக்கமாச்சுன்னா 100 ஆகணும். அது 100 ஆச்சுன்னா ஆட்டோமேட்டிக்கா என்ன ஆகும், ஏசி ரிசப்டார் மற்றது பிளாக் பண்ணும். பிளாக் பண்ண என்ன ஆகும், உங்களுக்கு நெஞ்சு வலி வரும், அப்ப கிட்னி ஃபெயிலியர், தலைவலி, அந்த மூச்சு விடுறது கஷ்டமா இருக்கும். இப்ப இந்த கபசுப்பு குடிநீர் குடிக்கிறதுல என்ன ஆகுதுன்னா இது வந்து அந்த வைரஸ் பெருக்கத்தை தடுக்குது. 10 வைரஸ் வருதா, அந்த 10 10 வச்சுக்குது. அந்த 10 10 இருக்கிறதுனால நம்ம அடுத்தது என்ன பண்ணணும், மற்ற மூலக்கூறுலாம் என்ன பண்ணுது, அந்த நம்மளோட நோய் எதிர்ப்பு சக்தி அதிகப்படுத்துது. அந்த நோய் எதிர்ப்பு சக்தி அதிகப்படுத்துறதுனால என்ன ஆகுதுன்னா அந்த கிருமியை, வைரஸ் கிருமியை சாகடிச்சு. This is the main reason கபசுப்பு குடிநீர் has the ability to control the coronavirus. So we published the thing. Okay. So finally I end my session. So still the coronavirus is there. So you need to be careful. Okay. Thank you. And so people who want to go for higher studies, you can see this. So South Korea, they have a top ranking universities, okay, like Seoul National University, Sangong University. These are the top ranking universities. Among top 500, more than 20 universities are in South Korea. So people who are interested, they can come. So the main eligibility criteria for doing a master degree and PhD, you should have IELTS and 2 essay publications and 2 conference presentation. If you have this criteria, you can get monthly stipend of 60,000 to 80,000. நீங்க IELTS TOEFL அந்த மாதிரி ஒரு IELTS ஸ்கோர் மினிமம் ஸ்கோர் இருந்தா போதும், 5.5 to 6 அது இருந்தா போதும். அப்புறம் 1 இல்ல 2 essay பப்ளிகேஷன் வச்சிருந்து இது போல 2-3 கான்ஃபரன்ஸ் அட்டெண்ட் பண்ணி சர்டிஃபிகேட் வச்சிருந்தீங்கன்னா நீங்க மாஸ்டர் டிகிரிக்கு PhD நீங்க அப்ளை பண்ணீங்கன்னா உங்களுக்கு மாசத்துக்கு 60,000 to 80,000 ரூபாய்க்கு சம்பளத்துக்கு ஸ்டைஃபண்டோட ஸ்காலர்ஷிப்போட உங்களுக்கு கிடைக்கும். So thank you the Sri Vidya Mandiri Arts and Science College for giving me the opportunity. Thank you. Hello?
Yes, sir.
Yeah, tell me.
Thank you for your informative talk, sir. Sir, some of our students are having some doubts, sir.
Yeah, sure.
Good morning, sir.
Yeah, good morning.
I am M. Naveen Kumar, UG second Botany. I have one doubt, sir.
Yeah, tell me.
What is rule of Kabasara Kudinir against Corona, sir?
What is the?
What is uh, what is rule of Kabasara Kudinir against Corona, sir?
The main thing is, okay, for COVID coronavirus, main thing two protein is there. One is spike protein, and second is 3CL protein. Okay, spike protein, the major role of spike protein, it go and attach to the ACE2 receptor. That is the point one. Okay, so after the attachment, it will insert the viral RNA. Next, the viral RNA will be start replicate. Okay, it goes for transcription translation process. For this transcription and translation process, the virus need an enzyme called 3CL Pro. 3CL Pro. Okay, the protease enzyme. That protease enzyme is important for the viral replication. So in case of cabasabucutinib, what we identify is this cabasabucutinib has the ability to stop the 3CL Pro mechanism. Once the 3CL Pro mechanism is stopped, the viral replication will be stopped. So it's the reason. புரியுதுங்களா?
Thank you, sir.
ஸோ அதனால தான் உங்களுக்கு வந்து அந்த நோயே வந்தாலும் உங்களுக்கு வந்து அதிகமாக இன்ஃபெக்ஷன் ஆகாது.
Okay, sir. Thank you, sir.
Yeah, thank you.
Hi, sir.
Hi.
I have a wonderful presentation, sir.
Yeah, thank you.
Thank you so much. So I have one question.
Tell me.
Many of the plants have been studied and brought to have antiviral activity. So we do some further research in such plants to avoid in corona, sir.
Yeah, we can do. But the thing is, you need to do animal trial. You need to do. You need to go for animal trial. Another issue is particularly what is the major drawbacks in herbal thing is we used to study only the crude extract. We should go for compound identification. Okay, as we study like that, so we know everyone knows that cabasabucutinib can inhibit the coronavirus. But if you enter into the market, okay, if you enter into the pharma industries, we need to identify the compound. இப்போ நம்ம உடம்பு இருக்கு, ஓகே, கண்ணு இருக்கு, மூக்கு இருக்கு, இருதயம் இருக்கு, கிட்னி இருக்கு, ஒருத்துக்கும் ஃபங்க்ஷன் இருக்குல்ல. இப்ப நம்மளோட பிளட் பிரஷர், பிளட் எல்லாம் வந்து இதயத்தை கண்ட்ரோல் பண்ணுது. புரியுதுங்களா? அப்ப பிளட் பிரஷர் எல்லாம் இதயம் கண்ட்ரோல் பண்ணுது. அப்ப இப்ப கபசுப்புக்குட்டினா கொரோனா பொறுத்த வரைக்கும் என்னன்னா அதிகமா வந்து மூச்சு பிரச்சனை. அதுதான் அதுக்கு எப்படி கண்ட்ரோல் பண்ணணும்? இப்ப கபசுப்புக்குட்டில கிட்டத்தட்ட 145 மூலக்கூறு இருக்கு. அது எல்லாம் அதாவது கிட்டத்தட்ட ஒரு 15 ஒரு கலவை தான் அது. ஒரு 15 பிளான்ட். கபசுப்புக்குட்டின்றது ஒரு 15 பிளான்ட். அந்த 15 பிளான்ட் நீங்க ஆய்வு செஞ்சீங்கன்னு பார்த்தீங்கன்னா ஒரு 145 பிளான்ட்ஸ் இருக்கு, காம்பவுண்ட் இருக்கு, மூலக்கூறு இருக்கு. அந்த 145ல எது முக்கியமா இருக்கோ அதை நீங்க கண்டுபிடிச்சுதான் நீங்க வந்து பார்மகோமெடிக்கு போக முடியும். So that is the reason.
Thank you so much, sir.
Yeah, tell me.
Sir, நீங்க தமிழ்ல ரொம்ப நல்லா பேசுறீங்க. அதாவது தமிழ் கல்ச்சரை கரெக்டா ஃபாலோ பண்றீங்கன்னு நினைக்கிறேன்.
இல்ல பசங்களுக்கு புரியணும்ன்றது தான். The main thing is language we need to understand.
எல்லாருக்கும் யூஸ்ஃபுல்லா இருந்துச்சுங்க சார். Thank you.
ஒரு நிமிஷம் இருங்க. I will make another slide. ஒரு 2 minutes கிடைக்குமா பசங்களுக்கு போட்டு வந்து.
Yes, sir, you can proceed, sir.
Sharing வர மாட்டேங்குது. ஸ்லைட் தெரியுதா?
தெரியுதுங்க சார்.
ஸ்லைட் தெரியுதா?
தெரியுதுங்க சார்.
ஷேரிங் தான் இருக்கு. தெரியுதா?
தெரியுதுங்க சார். தெரியுதுங்க சார்.
தெரியலையா? தெரியுது
Sir, it's clear, sir.
இல்ல ஸ்லைட் தெரியுதா?
Yes, sir, visible.
ஓகே. ஜஸ்ட் 5 minutes. பசங்க எத்தனை பேர் இருக்காங்க இதுல?
Sir, more than 100 students are here, sir.
ஓகே. இல்ல யூஜி பசங்களா பிஜி பசங்களா?
Sir, all UG, PG, research scholars எல்லாரும் இருக்காங்க சார்.
ஓகே. ஒரு 5 minutes டக்கு முடிச்சிரேன் நான்.
Okay, sir.
So students kindly make a note. This is the important thing. So நிறைய நான் தமிழ்ல பேசுறேன். ஓகே. Mostly Tamil students only. ஓகே. மாணவர்கள் இப்ப நிறைய பேர் முக்கியமா கவனிங்க. இப்போ நிறைய வந்து யூஜி படிக்கிற மாணவர்கள் இருப்பீங்க, போஸ்ட் கிராஜுவேட் பிஜி படிக்க மாணவர்கள் இருப்பீங்க. என்னுடைய இது என்னன்னா, இது ஒரு நல்ல வாய்ப்பு. இப்பயே நல்லா படிச்சிருங்க. ஏன்னா எது எப்ப நடக்கிறதுன்னு தெரியாது. ஏன்னா மருத்துவத்துக்கு நீட் வர மாதிரி நீட் வந்திருக்கு. அதே போல இப்ப வந்து நர்சிங்க்கு நீட் வர மாதிரி லைஃப் சயின்ஸ் உங்களுக்கும் நீட் வந்துடும். அதனால வாய்ப்பு கிடைக்கும் போது படிச்சிருங்க. நீட் வந்துச்சுன்னா அதுக்கப்புறம் ஒன்றும் எல்லாருக்கும் எல்லா வாய்ப்பும் கிடைக்காது. ஓகேங்களா? சோ இப்ப அடுத்தது யாருக்கெல்லாம் நிறைய பேருக்கு வந்து மாஸ்டர் டிகிரி பிஹெச்டி படிக்கணும்னு ஆர்வம் இருக்கும். சோ அவங்க எல்லாம் என்ன பண்ணணும்னா, இங்க தென்கொரியால வந்து பல உலகம் தரம் வாய்ந்த பல்கலைக்கழகம் நிறைய இருக்கு. இப்ப டாப் 200 இருக்கு உலகத்துல இந்த டைம்ஸ் ஹையர் எஜுகேஷன் அப்படின்னு உலகத்துல தலை சிறந்த ஒரு 100 ஒரு 200 பல்கலைக்கழகம் பாத்தீங்கன்னா, நம்ம இந்தியால என்னென்ன பல்கலைக்கழகம் நீங்க சொல்வீங்க? இந்தியால எதுதான் டாப்னு சொல்வீங்க? ஐஐடி சொல்வீங்களா?
Yes, sir. IIT.
ஆனா ஐஐடி எல்லாம் ஒண்ணுமே இல்லைங்க. வேர்ல்ட் ரேங்கிங்ல ஐஐடி எல்லாம் இல்லவே இல்லை. டாப் 100ல ஒண்ணு கூட இல்லை. சோ அதனால நீங்க அதை வந்து மனசுல இது அந்த ஒரு சில பேருக்கு ஒரு தாழ்வு மனப்பான்மை இருக்கும். இங்க படிச்சாதான் வேலை கிடைக்கும், அங்கதான் படிச்சா வேலை கிடைக்கும், அப்படிதான் ஒன்றும் கிடைக்கிறது. நான் வந்து தமிழ்நாட்டில் தான் படிச்சேன், பாரதியார் மெட்ராஸ் யூனிவர்சிட்டி தான் படிச்சேன், கிராமப்புற பள்ளியில தான் படிச்சுட்டு இங்க வந்தேன். சோ அந்த எண்ணத்தை எடுங்க. இங்க படிக்கிறவங்க ஐஐடி அப்படியே கிடையவே கிடையாது. உலக தரவரிசைத்துல 100ல ஒண்ணு கூட கிடையாது. எவ்வளவு அசிங்கம் தெரியுமா அது? ஆனா தென்கொரியால பார்க்கும்போது டாப் 200ல 10க்கும் மேற்பட்ட பல்கலைக்கழகம் இருக்கு. பாருங்க, சியோல் நேஷனல் யூனிவர்சிட்டி, சன்பியோங் யூனிவர்சிட்டி, கொரியா கேஸ்ட் யூனிவர்சிட்டி, இதெல்லாம் பாருங்க. டைம்ஸ் ஹையர் எஜுகேஷன்ல பாருங்க 63rd பொசிஷன், 82, 100, 142, 199 பாருங்க. சோ இந்த யூனிவர்சிட்டி நோட் பண்ணிக்கோங்க. பசங்க யாருக்கெல்லாம் வந்து ஹையர் எஜுகேஷன் மாஸ்டர் பிஹெச்டி படிக்க போகணும் இருக்குன்னா இந்த யூனிவர்சிட்டி நோட் பண்ணிக்கோங்க. சோ இதெல்லாம் தலை சிறந்த பல்கலைக்கழகம். புரியுதுங்களா? ஓகே. இப்ப நான் இல்ல சார், நான் வந்து மாஸ்டர் டிகிரி படிக்கணும், பிஹெச்டி படிக்கணும், எனக்கு எப்படி படிக்கணும்னு எனக்கு தெரியாது சார். இப்ப இது என்னோட பல்கலைக்கழகம், சேஜாங் யூனிவர்சிட்டி, என் பல்கலைக்கழகம். இங்க போங்க. இங்க போனீங்கன்னா யூனிவர்சிட்டி வெப்சைட் போங்க. வெப்சைட்ல போனீங்கன்னா ஓப்பன் ஆகும். அபவுட் சேஜாங், அகாடமிக்ஸ், அட்மிஷன், இன்டர்நேஷனல் ப்ரோக்ராம்ஸ் ஒண்ணா இருக்கும். அகாடமிக்ஸ் போங்க. அகாடமிக்ஸ் போனீங்கன்னா அதுல வந்து ஃபேக்கல்டி போங்க. நிறைய டிபார்ட்மெண்ட்ஸ் இருக்கும். நம்ம இதுல பாத்தீங்கன்னா பிசிக்ஸ், கெமிஸ்ட்ரி, பாட்டனி டிபார்ட்மெண்ட், ஜுவாலஜி டிபார்ட்மெண்ட், மைக்ரோபாலஜி டிபார்ட்மெண்ட் ஒரு டிபார்ட்மெண்ட் இருக்குல்ல, அது போல இங்க நிறைய டிபார்ட்மெண்ட்ஸ் இருக்கும். அந்த டிபார்ட்மெண்ட்ஸ்ல இங்க பாருங்க, காலேஜ் ஆஃப் ஆர்ட்ஸ், சோசியல், பிசினஸ், அது மாதிரி ஒண்ணா இருக்கு பாருங்க. இப்ப என்னுடைய டிபார்ட்மெண்ட் வந்து ஃபுட் சயின்ஸ் அண்ட் டெக்னாலஜி. ஓகே. சோ இப்ப காலேஜ் ஆஃப் லைஃப் சயின்ஸ் இருக்குங்க. அந்த காலேஜ் ஆஃப் லைஃப் சயின்ஸ்ல நிறைய டிபார்ட்மெண்ட் இருக்கு. ஃபுட் சயின்ஸ், பயோடெக்னாலஜி, மாலிகுலர் பயாலஜி, பயோ இன்ஜினியரிங். நம்ம இதுல லைஃப் சயின்ஸ் பார்க்கும்போது பிசிக்ஸ் இருக்கும், கெமிஸ்ட்ரி இருக்கும், பாட்டனி இருக்கும், ஜுவாலஜி இருக்கும், அது இருக்கும். ஸோ அதே போன்ற இங்கேயும் அப்படிதான். இப்போ என்னோட டிபார்ட்மெண்ட் ஃபுட் சயின்ஸ். இப்போ இந்த ஃபுட் சயின்ஸ் டிபார்ட்மெண்ட்டை கிளிக் பண்ணீங்கன்னா அங்க ஃபேக்கல்டிஸ் வருவாங்க பாருங்க, யாரால ப்ரொஃபசர்ஸ். இப்போ உங்களோட டிபார்ட்மெண்ட் பாட்டனி டிபார்ட்மெண்ட். இப்போ பாட்டனி டிபார்ட்மெண்ட் எடுத்தீங்கன்னா ஒரு 6 ப்ரொஃபசர் இருப்பீங்களா, 6 to 7 ப்ரொஃபசர்ஸ் இருப்பாங்க. அதே போல எல்லா டிபார்ட்மெண்ட்லயும் ஒரு 7 ப்ரொஃபசர்ஸ் இருப்பாங்க. அந்த ப்ரொஃபசர்ஸ் பாத்தீங்கன்னா பாருங்க, அவங்களுடைய ஃபோன் நம்பர் இருக்கும், அவங்களுடைய அகாடமிக் அவங்க எங்க படிச்சாங்கன்னு இருக்கும், அவங்களுடைய மேஜர், அவங்க ஆய்வு எதுன்னு பாருங்க. ஸோ இவருடைய மேஜர் இப்போ ஹாஸ்ஓகி, இவர் வந்து பார்த்தீங்கன்னா நேனோ ஃபுட், இது ஹாங், இவருடைய மேஜர் பார்த்தீங்கன்னா மைக்ரோபாலஜி, இது இவருடைய மேஜர் ஃபுட் கெமிஸ்ட்ரி, அந்த கெமிஸ்ட்ரி, இது வந்து கிம் மைக்ரோபல் டிரான்ஸ்ஃபர்மேஷன். அப்ப நீங்க ஒரு ப்ரொஃபசர்ஸ் நீங்க பார்த்துட்டு, பயோடெக்ல போயிட்டு ஒரு 7-8 ப்ரொஃபசர்ஸ் இருப்பாங்க. அந்த ப்ரொஃபசர்ஸ்ல அவங்க என்ன ஆய்வு பண்றாங்களோ அவங்களோட ஆராய்ச்சியும் உங்களோட ஆராய்ச்சியும் ஒரே மாதிரி இருக்குன்னு பாருங்க. அப்ப அவரோட இமெயில் ஐடி பாத்தீங்கன்னா அவருடைய இதை பாருங்க, மெயில் பாக்ஸ் பார்த்தீங்கன்னா இமெயில் ஐடி இருக்கும். அந்த இமெயில் ஐடிக்கு நீங்க தொடர்பு கொள்ளுங்க. இது போல வந்து என்னோட பேர் ஆரோக்கியராஜ், நான் வந்து இந்த காலேஜ்ல படிக்கிறேன், எனக்கு வந்து மைக்ரோபயோலஜில ரொம்ப ஆர்வமா இருக்கு, இந்த மைக்ரோபயோலஜி சம்பந்தப்பட்டவா நான் வந்து ஏற்கனவே நான் ஒரு 2 ஆய்வு கட்டுரை நான் பப்ளிஷ் பண்ணிருக்கிறேன், 2 தேசிய கருத்தரங்குல நான் வந்து பேசியிருக்கிறேன், அப்படி நீங்க ரிட்டர்ன் கொடுங்க. கொடுத்தீங்கன்னா அவரு ஓகே பண்ணுவார். ஓகே பண்ணீங்கன்னா உங்களை மாசத்துக்கு ஸ்டைபண்ட் நீங்க ஒரு பைசா நீங்க கட்ட தேவையில்ல. மாசத்துக்கு 60,000 முதல் 80,000 வரை உங்களுக்கு மாசம் ஸ்டைபண்ட் வரும். புரியுதுங்களா? சோ அதுக்கு என்ன தேவை? IELTS ஸ்கோர் ஜெனரலா 5.5 to 6 நீங்க ரொம்ப பயப்பட தேவை. நம்ம பசங்க அது தமிழ்நாடு கல்வி தரம் நல்ல தரம். அதுல நீங்க வந்து ஜஸ்ட் நீங்க அட்டென்ட் பண்ணாலே 55% 65% எடுப்பீங்க, அது போதும். அப்புறம் இந்த SCA பப்ளிகேஷன், இந்த பேப்பருக்கு முக்கியமா நெக்ஸ்ட் வந்து நீங்க அது இதெல்லாம் எப்படி கிடைக்கும்னு பார்த்தனா, உங்களுக்கு ஃபைனல் இயர் ப்ராஜெக்ட் இருக்கும்ல, அந்த ஃபைனல் இயர் ப்ராஜெக்ட் BSCO MSCO அந்த ஃபைனல் இயர் ப்ராஜெக்ட் அது ஒழுங்கான ஒரு நல்ல ப்ரொஃபசர் கிட்ட ஒரு நல்ல ரிசர்ச் லேப்ல ஒழுங்கா பண்ணுங்க. அங்க பண்ணும்போது குறைஞ்சது இவ்வளவு டெக்னிக்ஸ் எல்லாம் தெரிஞ்சு கத்துக்குங்க. ஏன்னா நிறைய பேர் வந்து நிறைய நினைச்சிட்டு படிச்ச உடனே வேலை கிடைக்கணும். அதாவது படிப்புன்றது ஒரு டிகிரி தான், அது வந்து ஜஸ்ட் ஒரு ஏணி தான், ஒரு என்ட்ரி தான். அது நீங்க சக்சஸ் ஆகிறதுக்கு நீங்க அடுத்தது நீங்க உங்களோட தொழில்நுட்பத்தை நீங்க கத்துக்கணும். இப்ப நீங்க இது ஒரு ப்ராஜெக்ட் பண்றீங்க, இப்ப ஆன்டி டெங்கு இல்ல ஆன்டி பாக்டீரியல் ஏதோ ஒரு ஒர்க் பண்றீங்க, அந்த ப்ராஜெக்ட்ஸ்ல நீங்க இது போல டெக்னிக்ஸ் எல்லாம் 2-3 டெக்னிக்ஸ் கத்துக்குங்க. GCMS, LCM, HPLC, செம், டெம், PCR இது போல எல்லாம் ஒரு 4-5 தொழில்நுட்பத்தை கத்துக்கினீங்கன்னா உங்களுக்கு நீங்க பப்ளிகேஷன் பண்ணுங்க. கத்துக்கிட்டு நீங்க ஆய்வு கட்டுரை பப்ளிஷ் பண்ணிட்டீங்கன்னா ஒண்ணு உங்களுக்கு ஹையர் எஜுகேஷன் உங்களுக்கு ஈஸியா கிடைச்சிடும். இது ஒரு வாய்ப்பு. புரியுதுங்களா? சோ என்னென்ன டெக்னிக்ஸ் இதெல்லாம் டெக்னிக்ஸ்ல கத்துக்குங்க. இது ஒண்ணு. அடுத்தது இப்போ வந்து மோஸ்ட்லி நிறைய பேர் PhD போறீங்க, போஸ்ட் டாக் போறீங்கன்னா சோ பாருங்க, சோ நீங்க ஜெனரலா நெட்ல நீங்க சர்ச் பண்ணாலே கிடைக்கும். இது போல இமெயில் ஐடி, ப்ரொஃபசர் இமெயில் ஐடிக்கு நீங்க தொடர்பு பண்ணாலே உங்களுக்கு கிடைக்கும். இது ஒண்ணு. அடுத்தது கம்பெனிஸ் பாருங்க. இந்தியாவில் நிறைய ஃபார்மா கம்பெனிஸ் இருக்கு. நீங்க மார்க்கெட்டிங்க்கு வேலைக்கு போகலாம், ரிசர்ச்சரா வேலைக்கு போகலாம், வாத்தியாரா வேலைக்கு போகலாம். வாத்தியாருக்கே ரொம்ப கஷ்டம் தான். இப்ப நிறைய பேர் யாரும் இப்ப இந்த கோவிட் டைம்ல அநியாயம் பண்றாங்க, யாரும் சம்பளமும் முடியறது கிடையாது. சோ ஃபார்மா கம்பெனிஸ் போகலாம், இது போல கம்பெனிஸ்க்கும் நீங்க போகலாம் நிறைய இதுல. ஓகே, சோ அங்கெல்லாம் பாத்தீங்கன்னா சோ இவ்வளவு கம்பெனிஸ் இருக்கு பாருங்க. பயோகான், சீரம் இன்ஸ்டிடியூட் ஆஃப் இந்தியா, பேனசிபர்டு, இவ்வளவு கம்பெனிஸ் இருக்கு. அஸ்ட்ராஜெனகா, இவ்வளவு கம்பெனிஸ் இருக்கு. இந்த எல்லா கம்பெனிஸ்க்கும் முக்கியமான தேவை என்னது? நீங்க ஃபார்மா கம்பெனிஸ், மருந்து கம்பெனிக்கு போறீங்கன்னா கண்டிப்பா வந்து இது போல தொழில்நுட்பம் தேவை. இதுல அட்லீஸ்ட் ஒரு 2 இல்ல 3 நீங்க கத்துங்க, எல்லாமே தேவையில்ல. ஒரு பயோடெக் கம்பெனி போறீங்கன்னா PCR, சீக்வன்சிங், செல் கல்ச்சர் பத்தி தெரியணும். மருந்து கம்பெனிக்கு போறீங்கன்னா GCMS, LCMS, HPLC அது போல இதுக்கெல்லாம் போகணும். இதுதான் முக்கியமான விஷயம். புரியுதுங்களா? சோ இதெல்லாம் முக்கியமான கம்பெனிஸ். இப்ப வேலை நீங்க எப்படி தேடுறதுன்னா ஒரு ஈஸியான சிம்பிளான ஒரு சுலபமான வழி சொல்ல பாருங்க. இது ஒரு கம்பெனி. இந்த கம்பெனில பார்க்கும்போது கரண்ட் ஜாப் ஓப்பனிங்ஸ் போங்க. இப்ப நீங்க வேலைக்கு போகணும், படிச்சு முடிச்சுட்டு வேலைக்கு போகணும், அதுக்கு ஏற்ற நீங்க இப்பயே நீங்க தயார் பண்ணிக்கணும். இப்போ ஜாப் ஓப்பனிங்ஸ் போகும். இது ஒரு கம்பெனி. இந்த கம்பெனில ஜாப் ஓப்பனிங்ஸ் இதுல என்னென்னலாம் போட்டுருக்கோம்? GR ஆஃப் இந்தியா இந்த கம்பெனி. ஸோ அதுல ஹெட், பிசினஸ் மேனேஜர், ரிசர்ச் ஆஃபீஸர். இப்ப ரிசர்ச் இப்ப பயோ அனலிட்டிக்ஸ்ன்ட்டு ஒரு இது, அதுல அவங்க என்னென்ன டெக்னிக்ஸ் யூஸ் பண்றாங்க? LCMS, HPLC, ஸோ இதெல்லாம் இருக்கு. அப்புறம் அனிமல் ஹேண்ட்லிங். ஸோ இதெல்லாம் எப்ப நீங்க கத்துக்கணும்னா நீங்க வந்து ஃபைனல் ப்ராஜெக்ட் பண்ணும்போதோ இல்ல PhD பண்ணும்போதோ நீங்க அப்படியே கத்துக்கணும். இங்க பாருங்க நிறைய கம்பெனிஸ்ல பாருங்க M.Pharm இல்ல அவங்களுடைய என்ன தெரிஞ்சுக்கிறாங்க. இன்ஸ்ட்ருமெண்டேஷன் முக்கியமா இருக்கணும், செல் கல்ச்சர் தெரியணும், இதுதான் மெயின். இது அப்புறம் சில பேர் வந்து ஐடிஸ் போகணும்னு ஆசைப்படுவீங்க. நீங்க நிறைய மெடிக்கல் கோடிங் ஜாப்ஸ் இருக்கு, அதுக்கப்புறம் ஹெல்த் கேர் ஜாப்ஸ் இருக்கு, கால் சென்டர் ஜாப்ஸ் இருக்கு. நீங்க அதுக்கும் அப்படியே போகலாம். ஸோ சில பேர் ட்ரைனிங்ஸ் எல்லாம் கொடுப்பாங்க. சில பேர் நிறைய பேர் வந்து இந்த பயோ ஸ்டாட்டிஸ்டிக்ஸ் நல்லா பண்ணுவீங்க, அந்த ஸ்டாட்டிஸ்டிக்ஸ் ஒர்க்லாம் நல்லா கத்துக்குங்க. அதே மாதிரி சில பேர் கம்ப்யூட்டர்ல வந்து சாஃப்ட்வேர்ஸும் நல்லா ஹேண்டில் பண்ணுவீங்க, அவங்களுக்குலாம் நல்ல ஸ்கோப் இருக்கு. இது போல இந்த ப்ரோக்ராமிங் இதெல்லாம் நல்லா இருக்கு. ஓகே, ஸோ அதே மாதிரி மெடிக்கல் ரெப்ரஸன்டேட்டிவ்ஸ், சில பேர் நல்லா பசங்க நல்லா பேசுவீங்க, நல்லா ஆக்டிவா இருப்பீங்க, அவங்களுக்கு இப்ப நல்லா 30,000 to 50,000 60,000 நல்லா சேலரி கொடுப்பாங்க. ஒரு 3 வருஷம் 4 வருஷம் நல்லா உழைப்பு போட்டீங்கன்னா உங்களுக்கு நல்லா இது வரும். ஸோ முக்கியமான விஷயம் நுண்ணிய நூல் பல கற்பின். அதாவது நீங்க என்னதான் நீங்க படிச்சாலும் இது பண்ணாலும் அந்த படிப்புலாம் உங்களுக்கு வராது. உங்களுக்கான ஒரு தனித்தன்மையை நீங்க வளர்த்துக்கணும். ஸோ அதுதான் அது முக்கியம். புரியுதுங்களா? ஓகே, தேங்க்யூப்பா.
Sir, hello sir.
சொல்லுங்க.
Sir, thank you sir.
தேங்க்யூ.
Thank you very much for your informative lecture. நீங்கள் ஸ்டூடெண்ட்ஸ்க்கு நிறைய அதர் தென் சப்ஜெக்டும் நிறைய பண்ணிட்டு இருந்தீங்க. இன்ஃபர்மேஷன் கொடுத்துட்டு இருக்கீங்க. This function I should thank பேரின்பம் மேம் ஃபார் இன்ட்ரடியூசிங் யூ டு அஸ். ஆக்சுவலி மேம்க்கு தான் நாங்கள் ரொம்ப தேங்க்ஸ் பண்ணோம். ரொம்ப இன்டெப்த்தாக எடுத்திருந்தீங்க. ரொம்ப ரொம்ப நன்றி. தமிழ்லேயே நான் உங்களுக்கு நன்றியை தெரிவிக்கிறேன். I hope we'll be in good touch in future also. And some of our students as well as research scholars have doubt. நான் உங்களுக்கு பர்சனலாக இன்கேஸ் எந்த டவுட் இருந்தாலும் நான் உங்களுக்கு மெயில் பண்ண சொல்கிறேன். தேங்க்யூ வெரி மச் சார்.
தேங்க்யூப்பா.
Okay sir, thank you sir. So shall we wind up the session sir?
Yeah, you can wind up.
Okay sir, thank you sir.
Let us start the next section. It is Dr. Matthew Dunn, Principal Scientist and Head, Plant Genetic Resource Division, Jawaharlal Nehru Tropical Botanic Garden and Research Institute, Palod, Thiruvananthapuram, Kerala. He has research experience for more than 34 years. He has described 7 new species of flowering plants, has published more than 80 research articles, has authored 11 books and 4 booklets. He has produced 5 PhDs and is now guiding 4 PhDs. To his credit, he has completed 9 projects and has ongoing 5 projects. He is a life member in Indian Association for Angiosperm Taxonomy and Indian Association of Spices and Aromatic Plants. With this, I request Dr. Matthew Dunn, sir, to deliver the lecture. Welcome, sir. Sir, you are not audible, sir. Sir, you are not audible, sir.
Hello.
Yes, sir, you're audible, sir.
Okay, thank you. So thank you for the nice introduction, and I appreciate the organizers of this webinar, and I congratulate all the authorities of Vidyanagar Arts and Science College, and thank you for inviting me for this. So directly I am going for my presentation. Hope is it visible now? Hello.
Yes, sir, the presentation is visible, sir.
Okay. So today the topic which I am going to deal with is a very simple topic, in very much related to your syllabus of PG as well as UG students. It is nothing but three terminologies: classification, taxonomy, and systematics. So I would like to connect these three terminologies as it's a progression from classification, then to taxonomy and to systematics. So probably this was the first question raised by a human being. So in the beginning, people want to know which they can eat and what they cannot eat. So they were searching around for food in the beginning and found some are edible and some are not good to eat, or even some are toxic. So actually the first classification began there in terms of searching the suitable food material. So that was the beginning of classification, I think so, especially the plant classification. The purpose was to find a suitable food. So this means primitive classification was an applied science with extremely practical aims, as I told now, with extremely practical aims to find which is the edible plant or which is the non-edible plant, and also benefits as for simple categorization of plants as medicinal, edible, or toxic. So this was the beginning of classification. There we can classify things, not only plants, any things in different ways. For example, you can see this bundle of books. So when we are classifying books, we can classify according to the size of the book, or the color of the binding, or the nature of its binding, like that. But all of them are unscientific and not basically good for a classification of books. So all of you may know that in libraries, the books are classified based on the subject. So if the books are classified on subject, we are not considering the color or the size of the book. So in this example, I would like to show that when we are classifying the plants also, there are certain rules on what basis we are going to classify the plants. So here in the case of books, in every library, the classification is based on the subject of the book. Like that, when we are going for plant classification, it has its own ways and rules. So as you may be knowing, D. Kendall was the first man who proposed the terminology taxonomy. The word, you may be knowing that it is coined by from two words, taxis and nomos, which means taxis means order, or nomos means law, whatever it may be. It is basically a science of arrangement or classification with certain laws. So we cannot simply classify the things. So there should be some laws or some basics for that.
Excuse me, sir.
Yes.
Sir, can you share the screen in full screen, sir?
Is it okay?
Yes, sir. Thank you, sir.
Okay, thank you. So again, I'm coming to the three terminologies: classification, taxonomy, and systematics. Of course, classification, the arrangement of organisms into groups based on trait similarity. So there, the consideration to classify an organism is mainly the similarity within that organism. But coming to taxonomy, it is further advanced that it contains the naming of the organisms and their categorization, while systematics is clustering of groups or organisms based on a unifying set of principles, including interrelationships, evolutionary traits, etc. So see the difference between the three. So classification is simple grouping of some organisms, but while taxonomy is a bit more scientific with the names and all, while systematics is more elaborate through the clustering based on certain principles. And from systematics, we can find the interrelationship between these organisms or the evolutionary traits in them. Thus, the progression of classification to the present day systematics was not through a simple, smooth and straight trail. So it is not at all a simple thing or a comfortable thing. Certain efforts has to be there to come into the systematics of a particular group. So this terminology may also be familiar to you, the alpha taxonomy and omega taxonomy. Actually, this term was proposed by Tyrrell in 1935. Of course, the alpha taxonomy, alpha, you know, the beginning, the first letter of Greek alphabet, and omega the last. So thus alpha taxonomy is the holistic study based solely upon more or less obvious morphological characters. In alpha taxonomy, we solely depend on the morphological characters of a particular plant, while in omega taxonomy is a more complex study based upon all available characters aimed at the synthesis of a good and predictive phenetic classification. Of course, Carl Linnaeus, the father of the taxonomy, was basically a physician. So initially he worked on medicinal plants which are used for various treatment systems for curing the diseases. And later on he got interested in various type of organisms, not only plants, animals and all. So interestingly, he has described 7,700 species of plants and 4,400 of species of animals. So this is the first page of his famous book, The Species Plantarum. And there he used the sexual classification system. So it means he Linnaeus made the classification based on characters, especially he considered the excuse me. Hello. Hello.
Sir, we can hear you, sir.
There's some disturbance there.
One second. Sir Balamurugan, sir, can you please mute your audio, sir?
Okay.
Yes, sir, you can carry on, sir.
Yeah, yes. So we are talking about Linnaeus and his classification system. So for Linnaeus, considered the sexual characters, especially the structure of stamens, as the fundamental character to classify plants. So he considered the number of stamens or the pattern of arrangement of stamens or the attachment of stamens, like that. So he gave this terminology. Monandria means one stamen and diandria means two, like that. And monaceae means plants are monoecious, dioecious means plants are dioecious. So this terminologies was given by him. So based on the structure or the number or the arrangement of stamen, he classified the plants. So this is the diagrammatic representation how the stamens are different with respect to number, structure, shape, size, etc. And coming to different schools of classification, this may be also in the syllabus, I think. So there is artificial classification, natural classification, phonetic, phylogenetic, and evolutionary classification. So of course, artificial classification is based on easily observable or qualitative and quantitative characters such as the color, shape, size, etc. of plant parts, the flower color, flower shape, leaf shape, like that. So, so simply saying, when we are considering these qualitative and quantitative characters, which are easily notable, we can call that type of classification as artificial classification. While natural classification is a bit more advanced, we can define it as based on overall similarity in grouping taxa. So based after seeing the quantitative or qualitative morphological features, if we are grouping them based on these characters, we can call it a natural classification. And the famous classification by Bentham and Hooker was a typical example of natural classification. Coming to phonetic classification, and it is an integrated approach on the relationship based on data from all available sources, not only morphology, but also anatomy, embryology, cytology, palynology, and chemical data. So all these data are mutually correlated, and it can be effectively used for classification. So we can call it as omega taxonomy. So the phonetic classification is almost the same as omega taxonomy. They are mutually very much close to each other, because in this type of classification, we are using not only the morphological characters, but other supporting evidences from anatomy, embryology, cytology, chemistry, etc. So this is the diagrammatic representation, how they are correlated. So based on fundamental data, so the floral biology, breeding systems, the chemical data, morphology, anatomy, cytology, so and even numerical taxonomy, all these are different aspects which can be jointly form a data pool and resulting in an improved classification. So with all supporting evidences and depending on other disciplines, a taxonomist can elaborate his studies into an improved way of classification or a systematic status. So this is a typical example from horticulture. So you can see both the bonsais. So bonsais are a very famous art based on reducing the tree size, you know. So it's a famous art originated in China and Japan. So this is a Chinese decorative plant famous for bonsais and used in Japan and China for centuries. So they look exactly similar. And here you can see the leaves also. So usually, since they are keeping as bonsais, they will not produce any flowers normally. And thus, taxonomically, both of them are considered as same. But later, a horticulturist who used to regularly prune these plants found that one plant is different from the other just because its smell is different. He simply noted that the smell of this particular plant is different from the smell of this plant. He noticed this by pruning the plants to make his bonsais. And later on, he planted a twig from this in the field and allow it to grow like a normal shrub, which later on flowered. And interestingly, it was noted that the first plant is Acer Dirganum of family Aceraceae, while the second one, this one is Premna microphylla. So many years, the bonsai collectors named these two as the same plant, But later on, by a simple curiosity in the variation of the odor or smell of the plant, later on it was confirmed that the second plant is Pramna. Pramna has a characteristic smell just because of the presence of flavonoids. So this was confirmed in 1985 that this flavonoid profile is different, and this is how a chemical data helped to confirm the taxonomic difference. This is another example. You can see both plants. In horticulture and in gardening, many people are collecting and maintaining succulents and cacti like this. And even in flower competitions, it is appearing that both are coming under cacti. But astonishingly, we can see that all things displayed in succulents are not belongs to the family Cactaceae. So we are thinking in a taxonomic point of view. And so it is when we are making an injury on the stem, if we can see a white latex oozing out here, you can see a white latex, definitely it is a Euphorbiaceae member, not a Cactaceae member. So Cactaceae members never have any white latex, only watery juice inside that. But Euphorbiaceae members are with white latex. So very easily we can distinguish such things. So all these are observations which help us to distinguish different things. So this is one of our new species proposed in 2005. The Acrotrima belongs to the family Dilleniaceae. So while describing the new species also, many taxonomists are using not only morphological characters, but other characters. See, here you can see characters such as stomata. See, it is different in both Anisocyatic in Arnotianum, while this is the new species proposed, which as it is collected from Agasthyamala, it is named, we named it as Acrotrima agasthyamalianum. See, when we observe the stomata, it is different in both. And trichomes. Trichomes are also very minute and microscopic, and unicellular in the Arnotianum, and also shorter trichomes, but while it is very long, narrow, and tapering in Agasthyamalianum. And see the structure of epidermal cells, the shape of epidermal cells is angular in one while it is sinuate in the other. So along with the morphological characters, which is visible or which can be detected qualitatively or quantitatively, we can rely on microscopic or anatomical characters also. So this is the species proposed, and see, you can see the structure of stomata, so and the trichome and all. So even in protologue, we can include these type of characters to support the morphological characters. This is another example from another new species proposed as Spilanthes tetralobata from Kerala itself. And this Spilanthes tetralobata was very similar with Spilanthes ciliata, but here again some microscopic characters, like this is the cross section of petiole. See the difference. So anatomical characters are clearly supporting it, and see the structure of pollen grain of both. So again, difference is there, and also akene. Akene means minute seeds. In composite, you may be knowing that the seeds are very minute, and microscopic observation clearly give distinct difference between these two. So my point is simply we can use these type of characters, especially microscopic characters, also to support the morphological characterization. So now coming to phylogenetic classification, it is based on the evolutionary descent of a group of organisms where the relationship depicted through dendrogram, cladogram, etc. You may be knowing these terms such as dendrogram, etc. A dendrogram is a broad term for the diagrammatic representation of a phylogenetic tree. And while a cladogram is a phylogenetic tree formed using cladistic methods. So depending upon the branching pattern of this cladistic tree, we can establish the interrelationship between different class of plants. This is a simple diagrammatic representation of different genera of Zingiberaceae in South India. And this was a part of my PhD program 20 years back. And when I did my PhD program, I relied on, it was a chemotaxonomic thesis. So I studied the chemical constituents of the 42 taxa of Zingiberaceae from South India. But astonishingly, it was so interesting that the cytological data was very much correlated to these things. So the basic number of genus Zingiber is 11 or 12, and while these Elteria, Alpinia, Amomum, and Globa all are 12 alone, and only Zingiber shows some aneuploidy, ploidy like 12 and 11, and all others more than 12, 12 or more than that. While the costus, which is very much different from all others, has only 9 set of chromosomes. So this was very much matching with the morphological as well as chemical data, and the cytological data supported much to find the differences in different generic constituents. So evolutionary taxonomic classification means it differs from phylogenetic classification in such a way that the gaps in the variation pattern of phylogenetically adjacent groups are considered as more important. Once again, I will tell the gaps of variation. So here in evolutionary taxonomic classification, the variation is considered as an important criteria to assess the phylogeny or the resemblance between other taxa which we are studying. So we can say biosystematics is at the same time most elementary and most inclusive part of biology. Most elementary because organisms cannot be discussed or treated in a scientific way until some taxonomy has been achieved. That's very, very important. So it is an elementary, it's very basic, because we can name a plant just because of its morphological characters. And most inclusive because systematics in its various branches gathers together, utilizes, summarizes, and implements everything that is known about organisms, whether morphological, physiological, or ecological. So this definition is made by G.G. Simpson in 1961. And this clearly indicates simple morphological characters are helpful for, which are helpful for classification, will lead to a elaborate taxonomic form, and later on it will move to the systematics. So today's evolutionary biologists use comparison of various parameters such as morphology, biochemistry and physiology, chromosome structure or cytological data, cell ultrastructure, including the cellular inclusions, the type of tyloses or Golgi bodies or vacuoles, all can be used as such supporting evidences, and cellular metabolism and pathways, and nucleic acid sequences and protein composition, of course, the DNA barcoding and all those things, and geographical distribution, the biogeography, and also ontogeny, the embryo structures and the development of embryo. So all these can be used to build a complete catalog of evidence showing that two species were derived from a single common ancestor or not. So coming to the systematics, provide what are the functions of systematics, we can see. So it will provide classifications, both all classifications such as natural, phylogenetic, evolutionary, etc. And systematics are very much relevant to understand intraspecific variability. So there are variability within species or level also. So systematics is the major tool to determine the intraspecies variability and allow prediction of attributes of taxa not yet studied. That are another aspects of systematics. I will come to it with examples and understanding evolutionary process and including biogeography, phylogeography, etc., and give relevant data for conservation biology. So when we are doing systematics, all these benefits are there. And see, assessment of intraspecific variability. I told about it. So variability within a species. See, this is news, bananas. See how much variation is there. There are great examples such as in mango, coconut, even sweet potato. Much variation is there with respect to the shape, color, taste, all these things. So they are best examples of genetic variance. So this is a study conducted at our institute by one of my students to assess the intraspecific variability within Clitoria terminaceae. You may be knowing the plant Shankupushpi. Shankupushpi is a medicinal plant. So this blue variety and this white variety are the most common varieties of Clitoria terminaceae. So variability is very much there with respect to color, shape, etc. And see, in this case, another Fabaceae member of Mucuna Nicurna, it has in this plant, flowers are almost same, but here the fruits are different. See, the size, shape, and color of the fruits are different. And this also a study conducted by one of our former students. So, so reasons for natural variability is very important. And diversity occurs at molecular level due to ecological impacts, ploidy, mutation, or various other unidentified reasons. So the reasons we cannot predict actually, but the molecular studies may evolve, may give some clue why this much variability is found within a species. So even an ecological impact can create a variation, and a morphotype can be evolved with much variation. So even cytological differences of ploidy. So several reasons are there. So and another important point is natural crossbreeding between variants, multitudes, genetic variability. So therefore, molecular diversity and genetic diversity are very much linked to each other, because just because of molecular diversity, this genetic diversity is happening. And coming to another aspect, molecular diversity, the diversity at molecular level occurs within a species. Example is the sandalwood from Marayur region. You may be knowing that sandalwood are growing in different parts of our country, but the sandalwood harvesting from Marayur has the best quality of fragrance. And so it is because of that geographical influence on that particular species. So it is Sandalam album, but whenever Sandalam album, the sandalwood is growing in different regions. If it is grown in Trivandrum or in Coimbatore, it may not be of good quality as in the Sandalam album grown in Marayur. Same as the case of turmeric from Alleppey. It's also a geographical indicator. Status is get to that, because the quality of this particular turmeric, the curcuma longa grown in that particular region, has very significant importance with respect to curcumin content and all. So we have studied some other variations also with respect to Centrella asiatica from different. We have collected 62 accessions from different parts of South India, and found that chemically they are different. The chemical active principles used in this medicinal plant is metacosticide and asiaticide. Both percentage of these compounds are varying from different accessions collected from different regions. So this is a very clear evidence of intraspecific variability or genetic variability within Centrella asiatica. This plant also, this is a Singiber serum, but a wild relative of our Inji, the ginger. And the active principle is Rambon. So the ceremony content is very much varying in different accessions just because of this genetic variability. And values of biosystematics is another example is to analyze the potential of certain plants which we have not studied. So here we compared the inflorescence of different species of Musaceae, wild species such as this is Musa acuminata, Musa balduscana, and this is Encita superba, another genus of family Musaceae. Both all are wild bananas, and usually the banana flowers are used used as a vegetable. And a comparative study showed that literally, so this is the typical banana which we are using. The Palangodan variety is taken as the standard, which is widely used as a vegetable. But you can see that the wild bananas are also found more nutritive with respect to various constants. So this is a good example of a biosystematic study. And you may be knowing the compound camptothecin. Camptothecin is an anti-cancer compound, and it was first detected from the plant Camptotheca acuminata. It is belongs to Nicaceae family, but in this plant is in high altitudes of northern India, and the yield is also very poor. So the camptothecin is a potential compound, as I told you, it is anti-cancer. And later on, in another plant, Nothopodactes pneumoniana family Icosinaceae, see how different. So the same compound was detected in another plant, and much later, the same compound was found in Ophioraceae mangos of Rubiaceae. So here, a chemical which is very potential chemical is detected in members of different genera, different family. So in this aspect, we cannot use it for a chemotaxonomy purpose, but the advantage is this plant is very difficult to cultivate and multiply. And this also prefers high altitude hilly regions than Nothopodactes. While Ophioraceae can be much more easily cultivated and produce more amount of camptothecin from this. So this type of study is the result of biosystematic studies conducted by various researchers. And one more example I will tell you, these are the 4 Sida species. Sida, the first one Sida alnifolia. Sida, you may be knowing the Kurundote, a precious medicinal plant. The first one is Sida alnifolia, second Sida rhomboidea, third rhombifolia, and fourth one scabrida. All these look very much similar with respect to morphology. See, even the flower color, leaf shape, habit, everything. So that's why it is creating a lot of confusion. So many people are mistakenly connecting these together, and even for taxonomies it is a headache. So all these 4 are put under the name Sida rhombifolia complex, and it is 1996. It is named. These 4 were put together under Sida rhombifolia complex. So these are the 4 different Sida species coming under that complex. See the roots. So as you are knowing, the roots are the medicinally important plant. But while we are checking the anatomical characters, we can distinguish these very easily. So you can see the crystals, even see the structure, distribution, size, and shape of crystals in 4 different species are different, and they can be used as a clue to detect the root of which species is taken for the study. See here the arrangements of vast region, especially the phloem pattern, arrangement phloem pattern with respect to its shape, density, etc. So here all the 4 species are very distinct with respect to the vast region in root anatomy. So my point is, all supporting evidences such as anatomy, cytology, chemistry, or many data generated can be used effectively for to resolve taxonomic problems, and thus systematics has an important role. So my message is, look around the enchanting world of plants, enjoy the diversity, enhance your knowledge, endeavor to understand the interrelationship and ensure its conservation, and proud to be a botanist. So this image you may be familiar. This is from Hortus Malabaricus, the prestigious voluminous treatise published by Dutch people, the first book published on the flora of a South Indian region in the 18th century. So that's all. Thank you.
Thank you so much for a wonderful lecture, sir. In spite of the delay, you gave a very interesting talk, sir. Thank you so much, sir.
Okay, thank you.
Sir, some of our students are having questions, sir.
Yes, please.
Good afternoon, sir.
Good afternoon.
I am Sindhu, sir, final year student.
Okay, please.
I have a question, sir.
Yes, please.
What are the difference between variety, form, ecotypes?
Varieties, forms,
Ecotypes.
and ecotypes. Variety is mainly as below the species level, and like the cultivars, we can see the mango varieties, you know, isn't it? I shown these varieties are mainly based on the genetic diversity very much, and especially for horticultural plants or agricultural crops, we are relying on varieties to select better food to get better food material. Like in the case of coconut, mango, banana, definitely varieties are there. But forma is a taxon. So for forma, it is also a subspecies. It's beyond the level of species, but it cannot be considered as a subspecies. That much difference may not be there. Forma means just a minor variation with respect to the leaf shape or the flower color, or not much difference with respect to the reproductive characters. Only if there are certain differences with respect to the reproductive characters, we can consider it as a subspecies or a species. So it is lesser. It is, if the difference is very lesser, we can consider as a forma. And which was the other one you asked? Hello, could you hear me?
Form,
Could you hear me?
form.
Yes, sir. Yes, sir.
Yes, sir.
Okay. So what was the third one you asked?
Ecotypes, sir.
Okay, ecotypes. So when during my presentation, I told you that ecological conditions has a main role in creating morphological variations. For some example, if a same plant is grown in a low altitude and a high altitude conditions, a high altitude plant may produce more trichomes. Even in the case of bamboos or Oplandra, the genus Oplandra, the reed bamboo, we have observed that the bamboo collected from a high altitude is very dwarf and narrow stemmed one. When we have planted the same plant in our garden, it is growing as a normal Oplandra plant. So this type of variation, which is not genetical, only based on the ecological pressures or environmental stress. If some plants show some morphological variations, they can be called ecotypes.
Thank you, sir.
Okay.
Sir, excuse me, sir.
Yes.
So some of our participants asked many questions like Karupusamy. Nowadays, which type of classification we are following?
We can follow any suitable classification. We cannot say one is wrong or one is right. And the expert is coming in the next session.
Okay.
My Guru, Professor Ramachandran VS is there for taking the next session. He's the most expert person to deal with different type of classification and all. Very vast experience. Sir, Ramachandran sir.
So one more person asking, Balamurugan, he's asking, is there molecular tool to classify the plants?
Yes, there are molecular tools. Yeah, DNA barcoding, RFLP, all this can be used for classification.
Okay.
Even some families are merged. The Asclepiadaceae and Apocynaceae are merged just because of this molecular characterization.
Okay, sir. So another one question my student will ask, sir.
Rahul sir.
Yeah, please.
I am Innocent, third UG Botany.
Okay, proceed.
Sir, someone want to ask question, sir.
Yeah, please.
How to describe your new species?
That's we cannot simply reply this. So due to time limit, I have to go for a COVID vaccine now. So it's an elaborate answer is required. So could you please contact your new teacher there, Murugation? My student Murugation is there in your department. So he has published a new species. So he will give you elaborate information on how we can publish a new species.
Okay, sir.
Simply, we cannot answer that. That's the problem.
Thank you, sir.
Okay, thank you.
Thank you, sir. Once again, I thank you to spend your time in the busy schedule. Once again, I thank you, and students are very inspiring about your presentation, sir. Thank you, sir.
Okay, thank you. Thank you so much.
And also, sorry for the delay, sir. Because
That's
of
okay, no problem. No problem. No problem.
Thank you, sir.
Thank you.
Sir, shall we start the session, sir? Am I audible, sir? Sir, you are not audible, sir.
Unmute. Please unmute your
Sir, one second, sir. One second, sir. Sir, please unmute your audio, sir.
Hello.
Yes, sir,
Hello?
you're audible, sir. Yes, sir, you're audible, sir.
Fine. Thank you. Thank you. Okay.
Sir, shall we start, sir?
Yes, you can start. Don't too much. Say VS will talk. That's all.
Okay, sir. Okay, sir. Thank you so much, sir. Next session is by Dr. V.S. Ramachandran, sir, Professor retired, Department of Botany, Badir University. He has more than 30 years of research and teaching experience. He has produced about 15 PhDs and 30 MPhil students. To his credit, he has published more than 100 research articles. I'm very happy in welcoming you, sir. Sir, you can start your session, sir.
Thank you. Thank you very much. Download. இன்னைக்கு டேட் போட்டதுல இனிதான் தேடிட்டு முடியும். நிறைய வந்து ஒரு 2 மட்டும் சொல்லிருக்கோம். அங்கெல்லாம் இருக்குது. இல்ல நம்ம எங்க அம்மா கிராமத்தை விட்டு வந்திருக்க எல்லாம் எங்க மனுஷன் ஜாலியா பேசி இல்ல நீங்க நான் சமையல் பண்ணீங்களேன் லேட் ஆயிதான் நீங்க இருங்க இது இமெயில் அது உள்ள அது கேளுமா ஷேர் ஆகுதுங்களா? ஹலோ ஹலோ
ஹலோ கேக்குதுங்களா?
ஸ்கிரீன் ஷேர் ஆகுதுங்களா?
ஸ்கிரீன் ஷேர் ஆகுது.
ஓகே 1 மினிட்.
ஓகே. ஃபுல் ஸ்கிரீன் செட் பண்ணிருங்க.
இல்ல அது போன்ல
ஓகேங்களா பாருங்க.
இல்ல இது
ஓகே டன்.
ஓகே थैंक यू. நெக்ஸ்ட் டைப் பண்ணிக்கோ. ஹலோ
Sir, you are audible, sir.
Okay.
Slide is also visible, sir.
Okay, thank you. I wish to thank the secretary, principal, convener, organizing secretary, and Dharani for inviting me to this international webinar. I'm going to talk the relevance and importance of biodiversity conservation with reference to climate change, a case study from Western Ghats. As you know, India is one of the 12 megadiversity countries in the world, and we have roughly 45,000 species of plants, out of which we have 17,500 species of angiosperms. Many may think what do you mean by angiosperms? What we eat, rice, what we take, tea, coffee, or what we use, different type of edible fruits, all constitute a single species. For example, rice, Oryza sativa, it is a single species. Like that, we have 17,500 species from India. You should know there are few terminologies with reference to biodiversity. One is Earth Summit. It is held in 1990 Rio de Janeiro conference. The first word biodiversity came into the existence only from 1992. Earlier, it was referred for plants, systematic botany, whereas for insects, it is systematic entomology. So now, subsequent to the Earth Summit, the another first word has come into the existence. It's called hotspot. What do you mean by hotspot? It is introduced by the entomologist from US, Norman Meadows, introduced the hotspot. The meaning is where the endemic organisms are more, at the same time, the biotic and anthropogenic pressures are more. So based on that, the hotspot introduced to conserve the organisms which live on the earth. Initially, it was 18, later it was 24, 25 in the year 2000, and 34. Now we have 36 hotspots today. With reference to Earth Summit, one more you should know the culmination Earth Summit leading to CBD, Convention on Biological Diversity. What do you mean by Convention on Biological Diversity? The animal wealth or plant wealth or micro wealth, whatever you use, the user group should give a sovereign rights to the country of origin. That is called CBD. I can give an example that the turmeric, there was a battle in US to get the patent for turmeric. Finally, Pativaidiyam in Tamil, that is, it is origin from Tamil Nadu, has helped to get the patent for our wild turmeric. CSR own. Then IPR, intellectual property rights. If any discovery leads to a new product, who will be the owner? In order to take right credit, you should go for intellectual property rights. So many agencies, especially CSIR, is giving money to get your product, to get patent. So that way, all the four concepts are very important with reference to biodiversity conservation. You have any queries, specific queries, you can ask me at the end. As I told that there are 36 hotspots in the world, and today it is very interesting to know that 4 hotspots in India, that is Western Ghats, it is shared with adjoining Sri Lanka, whereas the Eastern Himalayas is joined with Myanmar, earlier Burma, then the Western Himalayas and Sundaland. So just I want to give, many people may not know what is Sundaland. You know, Andaman Nicobar, there are 36, 37 group of islands are there. The southern end, tip end that is called Car Nicobar Island. So in order to conserve the biodiversity of islands, the Sundaland was included. The other islands available in Southeast Asia also included in Sundaland. So as you know, the Western Ghats is a chain of hills, runs 1,600 kilometer stretch starting from River Tapti to Cape Comorin, covering 6 states, that is Gujarat, Maharashtra, Goa, Karnataka, Kerala, and Tamil Nadu. We have a gap near Palkad, is called Palkad Gap, that is 15 kilometer width. That is why Coimbatore is enjoying the salubrious climate around the year. So Western Ghats is very important. Many rivers, more than 35 rivers, some are east flowing, some are west flowing, all are originated from the Western Ghats. Until we conserve the Western Ghats, we may not be able to get any crops, you will not get any cash crops, you will not get the hydroelectric power. So if you want to enjoy your life, we have to conserve the Western Ghats. That is why conservation is always asked for conservation of any hills with reference to Western Ghats. So coming to why Western Ghats is very important, as I just now I told, it is one of the hotspots in our country, and it has a number of endemics. Roughly, we can say 5,750 species, out of which 33%, 1,800 to 1,850 species are endemic to Western Ghats. That means these plants are not available elsewhere. That is why the biotechnologists, botanists, or any people who involved in conservation, we would like to always call for the conservation of the Western Ghats. The next important is, it is the storehouse of wild relatives of cultivated plants. So what do you mean by wild relatives of cultivated plants? For example, you take pepper. We use every day-to-day life, we use pepper in making rasam. As COVID, in order to compare to the COVID-19, if you take pepper in a way, your body immunity will boost up. So that way the pepper is very important in our diet. Like that, we have 26 wild species are available in pepper. So if you want to improve the pepper, we have to cross the wild variety, then you may get a new aroma or a new flavor, new even improvement, the increase in oil content of the pepper. Like this, we have wild sugarcane, we have wild mango, we have wild jackfruit, like that. Many wild plants are available. So for our taste, for a comfortable life, all these wild plants are helpful. So we have to conserve these wild of cultivated crop. As you know, either the biology students, botany or zoology or biotechnology, it harbors various type of habitats. What do you mean by habitats? We will discuss very elaborately the next slide, and it forms one of the important phytogeographic region. When you divide India, it is divided into 10 phytogeographic regions: Eastern Himalayas, Western Himalayas, Thar Desert, Central India, Northeast India, Deccan, Gangetic Plain, Coromandel Coast, then Western Ghats. So this is one of the important phyto region. That way also the Western Ghats are very important. As I told, the vegetation types of Western Ghats, if you want to go either from Coimbatore, that is from Kallar, foothills of Nilgiris, Kallar, where starts the Ghats section, it will reach Nilgiris. Similarly in Kodaikanal, similarly in Valparai, on the way to Valparai, you get all the 6 types, that is scrub jungle, followed by dry deciduous forest. The third one is master deciduous forest. The fourth is evergreen forest. The fifth one is evergreen forest. Then the shola, that is mountain, temperate forest, and grassland. So, what do you mean by master deciduous forest? You get now, when you see the teak trees shed their leaves, marsupium, they use leaves, amla trees shed their leaves. So now you can only see the stem, the leaves have fallen, that is shed their leaves. It's called master deciduous forest. So what is the evergreen forest? Round the year, you feel as such you are the AC room. So 3 tier, emergent, middle, and the lower canopy. You get 3 canopy, come out with a lot of grasses. Ground layer is very thick. The other way, if you enter into any evergreen forest during monsoon, the leeches will come and suck your blood. That is the indication that you are in the evergreen forest. Then the mountain temperate forest or solar forest. These forests are available only in Nilgiris, Kodaikanal, and Shervarais and Anamalais with reference to Tamil Nadu, whereas it is available in Munnar, in Kerala. Okay, so they are available in sheltered valleys where the water will be running. Trees are very stunted. It is infected with epiphytes, lot of orchids, lot of ferns, and come to grasslands. That is always the shola forest accompanied by grassland. They, when you see the slide, you may able to understand what it is. Grasslands are very important. They absorb the rainwater and store. They let out during the rainy season. It acts like a sponge to absorb the rainy water and let out during the lean season. Unfortunately, without realizing the importance of grassland, Forest Department in 1950s, they converted into man-made plantations like pinus, battle, and eucalyptus. So these are the important types of vegetation with a reference to Western Ghats of Tamil Nadu. Now, you should know as a students of biology, what do you mean by biodiversity? Since I handled few classes for UPSC, that is civil service entrance examination, this is one subject they introduced, biodiversity, the UPSC syllabus. Okay, so ecosystem diversity, species diversity, and genetic diversity. What do you mean by ecosystem diversity? You happen to go to Marudamalai in Coimbatore, or you are in Uttangare, nearby, you get all scrub jungle, that is ecosystem. Then you happen to go to Rameswaram, it is an island ecosystem. And you go to nearby field, agriculture field, it may be a sugarcane field or it may be a rice field. It is a man-made ecosystem. You could able to distinguish what is the ecosystem. So different ecosystems are available, and species diversity. Already I spoke at length. We have about 70,500 to 70,600 species in India. One species, as I told, Mangifera indica, mango botanical name is Mangifera indica. Cotton, arboreum. So it indicates a single species. Like that, 70,600 species right now we have. So much rich we have to exploit for medicinal plants, or edible, or for construction, or for any miscellaneous uses. These plants, so species maintaining the species diversity is very important. Then the last is genetic diversity. So what do you mean by genetic diversity? Very simple example, you take rice, as I told, Varisa Sativa. But how many rice varieties we have? We say Gundu or ADT, we say Punni, we say IR8, we say IR20. All are varieties of rice. That is Varisa Sativa varieties. That is called genetic diversity. Take human beings, even though we are Homo sapiens, all we look alike? No. Why? There is genetic diversity among ourselves. There are 5 groups are there among the human being. That is why the Dravidian race, we live in the tropics, we live near the sun, near the seashore. That is why people little black in color because of continuous work. We work in the sunlight. Many of them we are agriculturists. That is why our skin color is little dark in color. Okay. So this is the 3 levels of diversity. Then you should know the services rendered by the ecosystem. So you know many people fail to understand where do you get water. You get water from the river. From the river, we are getting water to our home. If you don't get water at hills, you will not get water at your river or where the plains, you will not get any water. Air, you know, whatever the pollution, various oxides is available in the air. So it is reduced, continuous release of oxygen in the atmosphere by photosynthesis by the plants. Then the plants serve as a sink. What do you mean by sink? The dust particles, small minute things, it will settle down in the leaves, in the city, in the forest. So if they are not settled down, what will happen? You will get lot of that is air, due to air pollution, many environmental induced diseases. So the ecosystem is helping just to take the dirt which is flowing in the air. Then productivity is very important. As you know, the students of biology, photosynthesis is an important process through which you get the products. It may be a cereal, it may be a protein, it may be a flower, fruit, vegetable, or beverages, whatever it may be. Only because of the ecosystem you are getting the products. So you should remember, we are not given due recognition the service rendered by the ecosystem so far. Now people have started thinking how carbon sequestration, how the carbon dioxide conversion, based on that the World Bank now evaluating the countries which helping in reducing the emission is one. The other one is carbon, how the carbon dioxide is converted is another. So now, as I told, the productivity, now we come to products. See how many products you get from plants. It is innumerable. It is innumerable. However, to discuss few points, food, medicinal plants, gums and resins, spices and condiments, canes and bamboos, then minor forest produce. So I would like to touch upon at least 1 or 2 examples, at least from 1 or 2. Since because of the COVID, now people started giving utmost importance to the utility of medicinal plants. Now our Honorable Prime Minister has agreed that Ayush, that is Ayurveda, Siddha, Unani medicines are much handy to beat the COVID-19 disease. So our Ayurvedic and Siddha plants are helpful. So that way, more medicinal plants are available in the forest. We are yet to exploit some of them. So at this juncture, it is suggested that the conserved medicinal plants are very important. Due recognition has been given by starting a new ministry in 2000 when late Prime Minister Vajpayee was there. One NMPB, National Medicinal Plant Board, was started. So anybody wants to know more on medicinal plants, please visit NMPB site in Google. You may able to get more information. Then I would like to tell what do you mean by canes and bamboos. Canes means a small bamboo like bamboo. So I would like to give one example, that big shopper bag. The big shopper bag, the handle, it is made up of canes and small bamboos. So imagine if the handle is not there, you may not be able to carry the goods. If the handle is with the help of small like cane or bamboo, it is easy to carry. So where do you get the canes and bamboos? Canes and bamboos are more in Northeast India. So we get the raw material from Northeast, and where the jute bag, like how it is being manufactured, mostly it is manufactured in Kerala, and the end users, the metropolitan cities and other two-tier cities of Tamil Nadu and other states. So a small handle is very important to carry the goods. So like that, all products are very important. And finally, to discuss minor forest produce. What do you mean by minor forest produce? Other than timber is called a minor forest produce. Take honey. How many companies are selling honey? How many crores are involved in the sale of honey and honey products? Then amla, that is our emblica, that is Pellanthus emblica, Nillikai. It is one of the product minor. Then tamarind, Tamarus indica, is another minor for products. And our avare, Channa auriculata, like that number of products. I don't want to add more, just to give an example that these are all the products called minor forest produce, which are collected by the tribals and for their own use and to sell outside the world. Then coming to raw materials for the industry. So is again a very big food industry, paper industry, biopesticide, silk, textiles, medicines, so on and so forth. So I don't want to go, I would like to indicate biopesticide. So what do you mean by biopesticide? The well-known example is neem, Azardictin, Azardicta indica, like that. Many biopesticides are in pipeline. Many wild plants are being utilized. Now people talk of organic farming. You see, the ordinary rice you buy, it costs 60 to 70 rupees. So the organic rice, if you want to buy, it costs 3 times higher. So it is free from any your fertilizer, inorganic fertilizer. So neem oil is used, neem oil cake is used, and biopesticides are used. So now it is going to higher and higher in the future years. So the biopesticide industry is going to gain in a much bigger fashion. So that way, many industries will come forward. I have just cited one example. Energy plantation. Why you need energy? You may think what way? Without energy, we may not able to do anything. For example, the villages, now cooking gases are available. However, in the remote area and for some other purpose, they use local trees. The most important tree is Prosopis juliflora, that is introduced in South America due to climate change spreading much faster. The next one is Cachirona ecositifolia. These are another quick growing tree in coastal area. They cultivate in a large scale. Then eucalyptus, as you know, it helps getting pulp, one of the quick growing tree, and biomass is very rich. So for all our requirements of paper making pulp, you can use also for making viscose rayon. So then bamboo, similarly, bamboo also used for making pulp. Then for other domestic consumption, bamboo is finally used. So these plants are really helpful in order to meet the human requirements. These plants are very important. Even we can go for a small forestation. Already in few places it has been tried. Habitat for wildlife. So the forests are very intact. The elephants are there, tigers are there, line-tailed macaque, that is single korang in Tamil, it is there, and other wild animals. So you know, in Coimbatore Nilgiris, even in your district Arur nearby, the elephants quite often enter into the cultivable area or human inhabitation. Always human animal conflict is there because human being, we encroach their habitat. It is not vice versa. So it is a problem, especially in Krishnagiri district of Usur and Nilgiris and in Coimbatore. So we have to give way for their living. The next potential, if you conserve these wild plants, you can export the medicinal, and it is already going on. Then wood, you know, your red sander, how many of them know? I don't know. It is Sterocarpus tantalinus, available in that is Tirupati Hills, Simachalam Hills, where our friends from Tamil Nadu, they go for cutting these trees. Every day you can read in the newspaper. So this tree, the wood, is being exported to foreign countries illegally, clandestinely. It is similarly many earlier rosewood, many such wood is being exported. Ebony, that is Diospyros ebanum, the Dalbergia latifolia, like that many type of wood they are exporting. Then cut flowers. What do you mean by cut flowers? It's orchids. Orchids are a group of flowers, long lasting 4 to 6 weeks. We can cultivate, we can export. Then anthurium is one we can export. Similarly, we have number of other orchids, number of plants, zerbera, many such. I don't go much in detail. These plants, when you export, you will get a foreign exchange. You get also employment opportunity. So that way, how the plants are helpful to the humankind, that one has to think. And tourism. See, after all, the human being, we are not satisfied where we live. Now, last 1 year due to COVID, it is being curtailed. Otherwise, what will happen? People go weekend, quarterly vacation, half-yearly, Pongal holidays, summer holidays. So what will happen when we go to the different forest area? We throw unwanted polythene, the rubbish materials, when the even bottles, it will deleterious to wildlife. So there is an urgent need to educate the tourist. There is increase in ecotourism. So there should be a ban on film shooting in the fragile area. When people go, there is a possibility buying endemic plants, especially for orchids from Kodaikanal and Nilgiris. So due to this tourism, we have lost the movement of animals. the movements animals is curtailed. We also, the endemic plants, some are we lost, and when we throw many things, it is deleterious to. So there should be a restricted tourism in the forest area. Then only we may be able to save whatever the left out plants and animals. And man-made changes. For our comfort, what we did in the forest area, especially Western Ghats, monoculture. As I said already, that raising a single crop called a monoculture. It may be eucalyptus, it may be Pinus petula, it may be Acacia wattle. There are 4 species: Acacia delaptila, Acacia auriculiformis, that is black wattle, yellow wattle, like that 4 species. Then eucalyptus, that is blue gum, Eucalyptus grandis, like that. We converted forest into monoculture. Then agriculture crops. Over the period of time, the agriculture for horticulture crops, we increased at the cost of forest. That way we reduced the forest area in the Ghats. Then conversion of more forest land for non-forest activity. So you want a collector office, or you want a college, you want a school, anything, convert the forest land into non-forest activity. This I studied in Idiki, especially in Idiki district, after construction of the Arch Dam. So we have a very clear set records are there. 2 years back there was a severe flood because the conservation related issues they didn't follow. That is why we faced a heavy loss in Idiki. Then this leads to habitat changes. That means earlier the forest is a continuous patch through laying road or through construction of some new building. So the continuous habitat get altered, so leads to habitat changes. So these are all, it is man-made changes. And finally, see the invasive alien plants. What do you mean by invasive alien plants? We say exotics. In many plants, what we eat, there are introduced plants mostly, but they have been naturalized. Here and there, but these invasive plants spread much faster in the forest, and they never allow the native plants to grow. So these are all the plants with reference to Nilgiris and Kodaikanal. So the first is probably discuss the Citis coparius, Ulex europaeus, Irageron, Corvinchianus, Helichrysum bracteatum, that is everlasting sunflower, Chromulina odorata, that is communist pacha, then Adenophora gelatina. So these are all the species which occupy more open forest area and disturbed forest lands, a severe competition to the native plants, and pose a grave threat to the native vegetation. So it is high time that these plants are to be removed in a war-footing manner in order to save our native forest. So habit presentation. Just I want to give only one point here. Primary cause of species extinction globally and in most countries. So when all the 3 are there, what will happen? We have to lose the species extinction. So that's been very clearly stated. 83% of surface transformed by humans, 60% ecosystems are considered degraded or unsustainable use. So I don't want to tell much. These two important points you may be able to understand. Then, as I said, to go a little further, so agriculture, vertical expansion, man-made, mining extraction, I didn't tell, wood extraction, no longer in our state, fire, summer, forest fire. See, last week I had been to Srikakulam in Andhra Pradesh. Then when I took the participants forest area for yesterday, we have seen fire, that is on Saturday, we have seen fire in Eastern Ghats. So like that, Western Ghats also, you get fire. Then the most important one is construction of multi-purpose river valley projects. So in order to get hydroelectric power, you need power projects. Imagine Nilgiri is a small district, but you have 9 dams. Like that, we have lost the forest. Then construction of EV towers. When the electricity is produced, it has to be transferred to ground, that is plains, for that huge towers are being constructed. So for line, the entire area, forests are removed, then laying roads. As you know, people, urbanization leads to laying roads. When urbanization leads to more urban dwelling, so that way, these are all forms of habit degradation and industry, human settlement, then human water transportation, tourism, pollution, atmospheric pollution. There are land pollution, water pollution, already told invasive non-native species. So importance of forest, I don't want to go much into detail. Water filtration, cycling, flow rates, flood control. You get, these are finally climate regulation at various spatial scales. So 2/3 of the all water supplies originate from or of course through or influenced by forest. So if you don't conserve the forest, you will not get the water. Finally, deforestation. Imagine for the last 300 years, we have lost 50% of the forest. 29 countries, it is 90% loss. 25 countries already loss is complete. And Europe is the least forested country. Africa is the highest current rate loss. And human practice, that is slash and burn forest, and 60% loss is taking place in tropics. At current rate of loss, Amazon rainforest will vanish by 2030, and all tropical rainforests will be gone by 2090. It's a severe warning. See, until you conserve, you will lost everything. Finally, you have to go for a gene transfer plants only. Okay, so grasslands, again, this is very important. Nilgiris, some grassland, converted into wattle, eucalyptus and pine plantations. Just to give one example, grasslands ecosystem that are dominated by herbaceous vegetation, primarily of grasses, but often we have some minor shrub and tree components that do not grow into forest or woodland as a result of climatic other factors. So it is very, very important to maintain the grassland. Then habitat fragmentation, that is with scattered bits and pieces, finally break into small pieces. So habitat fragmentation versus habitat shredding. So I don't want to go much to the detail. I will go into the slides. You can see the slide shows every type of vegetation. See, this is the grassland. This is the forest. You see small upper top. This is shallow forest, mountain temperate forest. This is the evergreen forest. This month of November, October, November was taken. See, the water is gushes out. All the 3 type one can see in Valparai, probably 7th bend or 8th bend. One can see this. So this is the forest. All the 3 one can see in this area. See, this is Silent Valley. You can see the evergreen forest, you can see the grassland, you can see the top. See, you should know what do you mean by Silent Valley. Silent Valley is the national park. It is in Palakkad district. It is only the pristine forest, not disturbed in the evergreen forest. In 1980s, a dam was supposed to be constructed with the intervention of late Prime Minister Indira Gandhi. It was stopped. Now, Nilgiri Biosphere Reserve, it forms part of Nilgiri Biosphere Reserve. So, it's one of the important area, well protected area. Anybody wants to visit, you can visit the Silent Valley National Park online. Can booking is available, but due to Corona, I am not sure. So one should visit. You should, in order to know what is a thick forest, how forest being conserved in Western Ghats. This is the place one can visit and understand. This is Paikara. This is important one of the river in Nilgiri on the way to 9th mile, where the film shooting made. More films are being taken. That is 9th mile, where after 30-40 years, only this year this area got filled. You just see, this is on the way to the top area. This area should not be cultivated, but people started cultivation. We are not supposed to do, but how they are doing, just to indicate this side plantation, can say, without following any conservation, they started cultivating in the hilltops. This is another well down. This is called well down near the All India Radio in Nilgiris. You see the finest plantation here. They make even that games they play here. This is a beautiful area, well down in Nilgiris. This is the 9th mile. This is taken during summer. People go here shooting. They take many places, many films. They used to take. Now they made tourism center by paying 10 rupees or 5 rupees. Now people being allowed in important area for tourist on the way to go to in this. This is a famous plantation. This is 7th mile, where the duet songs are enacted. One hour they charge 5,000 rupees. Even in the man-made plantation, how people make use of the particular habitat is a typical example. Finest plantation in 7th mile, again on the way to Budulur. This is an interesting area. They happen to go, you can see this place. See, this is not supposed to be taken. This is our Bhavani Sagar. This is Bhavani Sagar Dam. How you get the water from river Bhavani and other. So if the dams, today I read in the news that river Amaravati was full last 100 days. It's a record break. Here also 70 feet height, if I remember correctly, the Bhavani Sagar. So people this year will yield a bumper crop or 2-3 crops they can make because of the continuous availability of water. So we thank God for giving copious rain this season. So this is Bhavani Sagar Dam. A typical near Ooty, how the valley lake area is being converted for raising carrot and tomato, cauliflower and cabbage. See, this is the area. This is near on the way to again Avalanche. This was taken. See the tea, tea is very, very important foreign exchange. And Assam is the leading state in the country, and followed by Kerala and Tamil Nadu. So within Tamil Nadu, Nilgiris is the first, second is Coimbatore, third is Tirunelveli district, now it is Tenkasi district. So this is a monoculture. You see how tea is employment, you get foreign exchange. This is a part. You see the deer. This is in Mudumalai. You have taken how the Parthenium is spread, how it entire area. So until we remove these too difficult, other animals can go. It will cause a lot of heating due to pollen grains. Okay, so this is Stachytarpata indica. It's another introduced plant in the open wasteland. So why we want to show this? Attract a lot of butterflies, because whether nectar is more or honey is more, but it's a bit for pollinators, competition for. So native plants will not have attractive flower, size will be small. So because of the butterflies are attracted. See, this is Mimosa diplotrica. It is in Kerala and Gudalur district of Tamil Nadu. So it's spreading much faster in the marsh deciduous forest. Normally it is grown as a cover crop in the rubber estate. So it spreads, and they reported for the state of Tamil Nadu in 2010. So this crop has to be removed. This is the Eupatorium odoratum. Now the correct name is Chromalina odorata. Is called siam weed in English. In Malayalam, it is called communist pacha. Tamil, we don't have. Since it is introduced plant, we can say language translation, communist city. Only that much we can tell. So when during Second World War, our Malayali brothers, when they went for war, Second World War, attached to the this fellow came from Siam, he simply thrown. So one seed came, then it got multiplied. Now it is quite common in Western Ghats, Eastern Ghats, giving difficulty to the native plants. So this plant has to be removed at the earliest. Then this is another Mecania micrantha. This is creeper. Through water course, it spread, and 1976, this is reported in Idukki district of Kerala. In Alais and Nilgiris, now it's spreading near the tea estates. So this also spreading due to climate change. All these plants spread much faster than it was. That is why we always insist that exact has to be removed. This is Eulux europaeus. Tamil it is called Pandrimul. So it is so common on the way to Doddapata, on the way to Avalanche. It spread much faster. It will not allow any wild nearby plants to grow. So forest department has taken some, but the effort is very, very less. It is not enough to control this. So NGOs, even the tourists, when they go, anybody interested can remove this to involve yourself in the process of conservation. This is from Aranticum on the way to roadside Gudalur. When you go from Nilgiris, on both the sides of the road, you can see in Kodaikanal also this plant is so common. So Nilgiri, Ooty town, wherever you walk, you can see this. So these plants, just like other the spread, so these are to be removed. This is everybody know. In Tamil we say Velikathan, Delhi Mul, whatever you say. This is an important tree with a reference to fuel wood. If you remove all, we ended up with the removal of forest. So one way it helps, it serves as a good fuel wood tree. Only the concern is it should be removed in the wildlife sanctuary area. And it should be removed in water bodies, lakes, river, pools, or channels, canals, water body, lakes, that should be removed. So it gives a lot of employment opportunity in Ramanathapuram district, in Sivaganga and Virudhunagar district. So personally, I feel only in the water bodies and wildlife sanctuaries, this has to be removed. Again, due to climate change, this also spread much faster. So we have to make a balance where to remove, where not to be removed. Then only we may be able to get more fuel wood for industry. For example, tea. I told if you take as it is green tea, but you want to make black tea, the tea has to get fermented. So it has the boilers. In order to heat, we need fuel wood. So only this wood is used wherever tea industry. So that way this plant is very important. This is antenna camera. In Tamil it is called Unnichedi. In plains also it is plenty. So this has to be removed. It will occupy just like that. So gradually these trees are to be removed. It curtails the movements of animals in wildlife sanctuary, especially animalize and in Nilgiris. So people have started with only 10% success. Males are long to go. 90% they are yet to remove. This is our wild state, Tamil Nadu wild state animal, Nilgiri tar, and this is peacock in Botany University campus. This national bird is a national animal, tiger. I have seen two locations in the animals where I worked. So see why I want to show this. Now population has come down. This also, the animals have a fashion. Any movement is there, LTM will make a noise that making a caution that somebody is coming. So like that, they speak themselves. Until you stay in the forest, you may not be able to know the signal, how they make, how they talk to each other. So it is a long way to understand the movements of animals. So I normally take classes for the Tamil Nadu Forest Fund personnel, guards, forester, ranger, like that. So this was taken in Gudulur. There they have a one gene pool garden in Odgani, where I have taken classes in 2009, 2010, and the conclusion: if you want to conserve, there should be intensive, extensive exploration. Unexplored area has to be explored thoroughly. You may able to understand short-lived animals. From that, you get new distribution range and the need for change in the threat status. Many times it is located single locality. No, it's available in many places. People never used to go earlier. Now youngsters go and collect, and there is an urgent need, as I suggested earlier, remove the invasive alien plants, natural vegetation, in order to save our native species, that is endemic plants. Otherwise, the pollinating agents are under severe threat. So I wish to thank once again the organizers for giving me a wonderful opportunity to share my experience with you. If you have any specific queries, I will be very glad to answer your queries. So hand over to the organizers. Thank you.
Sir, can you hear me, sir? 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 Thanks to the organizers. எல்லாரும் அனுப்பிச்சு விட்ருங்க. இல்ல நான் அனுப்பல, நீங்க தான் அனுப்பிச்சு விடணும்.
வாட்ஸ்அப்ல
சார் சார் क्वेश्चंस இருக்குங்க சார்.
அன்மியூட் பண்ண சொல்லுங்க அவரை.
சரிங்க சார். நீங்க அன்மியூட் மட்டும் பண்ணுங்க சார். சரிங்க. ஸ்டூடண்ட்ஸ் எல்லாம் நிறைய क्वेश्चन கேட்டுட்டு இருக்காங்கங்க சார்.
சரி. பஞ்சாப்ல இருந்து ஒருத்தர் தேங்க்ஸ் சொல்லிருக்காங்க.
ஓகேங்க சார். ஓகேங்க சார். Due to some technical error, sir has been disconnected. Sir will be in online soon. So participants, if you have any queries, you can ask in the chat box. Sir, am I audible, sir? Sir, unmute பண்ணிருங்க sir.
அன்மியூட் பண்ணிங்க சும்மா.
Okay sir. Those students இருக்காங்க sir question கேக்குறதுக்கு.
Please sir, please.
Okay sir.
Good afternoon, sir.
Hello, good morning. Tell me.
Sir, my name is Arun Pranav.
Excellent.
I am studying third year UG Botany.
Very nice.
Very excellent, sir. Useful data.
Okay.
I have one question.
Please.
How to categorize place as a hotspot?
No, that's why there are two characters. One is endemic nature. Plants, animals which are endemic to particular location is 1 point, 1 parameter. The second parameter is anthropogenic pressure. Anthropogenic pressure means man-induced changes. In India, what we do, we make quarrying, we make dam construction, we make road. All these man-made induced changes. That is why this hotspot, these two are I explained under hotspot. Okay.
Okay, thank you, sir.
Fine.
Sir, good afternoon, sir.
Good afternoon. Very nice.
My name is Sanjay Kumar, third Botany.
Yeah. Good.
I had some question.
Please.
What is Sundaland? Sundaland.
See, Sundaland is a group of islands in Andaman Nicobar Island. We have 36 to 37 islands are there. The last southernmost island called Car Nicobar Island. The name is called Car Nicobar Island, and other islands, that is Malaysia, this side, whatever the islands are there in the ocean, in order to conserve this marine biomes, the Sundaland is being included. So Sundaland means a group of islands which are located near Car Nicobar Island in Andaman Nicobar. Okay.
Okay, sir. Thank you, sir.
Then.
Sir, very good afternoon, sir.
Good afternoon. Nice.
My name is N. Samraj from UG 3rd Botany, sir.
Excellent.
Sir, I have one question, sir. Can I ask with you?
Please, please.
Sir, what is an innovation species, sir?
What is?
Innovation species.
Okay.
Invasive. Sorry, sir.
Invasive. Invasive means which are introduced from other country and spreading much faster.
Okay, sir.
Exotic means introduced from other country and spread much faster. That is called invasive species. Like our Prosopis juliflora. I don't know. Do you know Velikathan? Do you know? Tamil Velikathan?
Sir, Velik?
Velikathan.
Yes, sir. I don't, sir.
That is one of the invasive alien species.
Okay,
it
sir.
spread much faster. ரொம்ப வேகமா பரவுது.
Yes,
That
sir.
is we called invasive.
Yes, sir. Land சுத்தி கூட எல்லாம் போட்டு வச்சிருப்பாங்க. இப்போ கூட எல்லாம் பார்த்துட்டோம்.
அது வேகமா ஸ்ப்ரெட் ஆகிறதுனால invasive.
ஓகேங்க சார். Thank you, sir.
Fine.
Thanks for giving a responsible presentation and my questions answer பண்ணதுக்கு ரொம்ப thanks, sir.
Very nice. Glad. Very glad. Please.
Sir, thank you so much, sir.
Nice, really. I'm very glad. You please convey my thanks to one and all. Your convener, your secretary, or your college secretary, army secretary, all. Okay? All the best. God bless you. God bless you. Bye.
இந்த இது எங்கே கடை தேடணும் குழந்தை கொண்டு போய் போடணும் தெரியாது.
The next session will start by 1:45. The participants are requested to join the next session by 1:45. Thank you.
ஒன்றை
வீடியோ வரல, வீடியோ வருதான்னு பாருங்க. Good afternoon, ma'am. Shall we start the session, ma'am? Ma'am, you're not audible, ma'am. Hello?
Okay.
Yes, ma'am, you're audible, ma'am. Shall we start the session?
Yeah. Okay.
Good afternoon, ma'am.
Good afternoon.
Now the session starts with Dr. Manju C. Nair, Assistant Professor, PG Department and Research Center of Botany, the Zamorans Guruvayurappan College, Kozhikode, Kerala. She has more than 15 years of research and teaching experience. To her credit, she has published 14 new species in bryophytes, has completed more than 5 major projects funded by DST and KSCSTE, and has completed 6 student projects. She has produced 5 PhDs and has 4 ongoing PhDs. She has published more than 100 research articles and 6 books. With this, I'm very glad to invite Dr. Manju C. Nair ma'am to deliver her lecture. Welcome, ma'am.
Thank you very much for introducing me. Very good afternoon to one and all. First of all, I would like to thank the principal, then organizing convener, Dr. Vinod Kumar sir, then Sadish Kumar sir, and Murugeshan sir, who has welcomed or invited me to participate or to give a talk on bryophytes, diversity of bryophytes in Kerala. Thank you very much for giving me a chance to attend this seminar. Thank you. So now I will share my screen. Is it shared?
Yes, ma'am.
Yeah. Can everyone see this? It's okay?
Yes, ma'am. Yes, ma'am.
Okay. Very good afternoon. So my topic is diversity and distribution of bryophytes in Kerala and its major challenges in conservation. So it is very difficult to conclude the diversity and distribution of bryophytes, especially in Kerala, within 45 minutes. But I will try my level best to, I think there is the students from UG and PG. So I am starting with the basics. Some basics I will say about bryophytes. So the bryophytes are divided into 3 major groups. That is liverbirds, hornbirds, and mosses that you all know. So, but scientifically it is termed as Markanthophyta, Anthocerotophyta, Bryophyta. So from the figures, you can see that what all differences you can see in liverbirds, hornbirds, and mosses. Morphologically, it is dissimilar. And very peculiar character is that they are non-flowering. So in the earlier section by Mathudan sir and Ramayandran sir, you have seen that all flowering plants, several beautiful flowers you have seen. But the bryophytes are without flowers and they are non-fruiting categories. But here, the peculiar character is that the gametophyte is prominent and the sporophyte is parasitic on the gametophyte. So at least sometimes, maybe some species shows 1 hour time, 2 months gap. So the sporophyte may mature within 1 hour to 2 months time. So it depends upon the species, this may vary. But the gametophyte is very prominent in this group. And you have all know that what is the difference between bryophytes and pteridophytes. In pteridophytes, the sporophyte is very prominent and gametophyte is very less prominent. But instead, in bryophytes, the gametophyte is prominent and the sporophyte is parasitic on the gametophyte. And you all know that they are the amphibians of the plant kingdom. Why it is called the amphibians of plant kingdom? Because they need, at least at the time of reproduction, they need water for reproduction. And I would say that the bryophytes are non-tracheophytes. So why it is called non-tracheophytes? You all seen that in your textbooks, it is non-vascular plants. So there is vasculature. So I can say that there are vasculature in bryophytes because, so you can see like this, when you take a section of stem or a leaf of bryophyte, you can see like this. So you can see in the center, you can see a specialized cells. So it is called as hydroids. And in the marginal cells, you can see that there are some other cells which are like phloem-like cells. They are called the leptoids. So in the place of xylem, it is replaced with hydroids. And in the place of phloem, it is replaced with the leptoids. So there are vasculature like hydroids and leptoids instead of xylem and phloem to absorb the minerals and nutrients or water absorption. So you have seen that there are several species from 2 mm size to 6 centimeter long. So here you can see, these are all some of the section through some stems. So here all you can see that you can see vasculature here. Here you can see vasculature. This photographs also, there is vasculature. This is the section of leaf, lower one. Here also you can see that there is vasculature. So the absorption of minerals and water is done by xylem. Instead of xylem, it is hydroids. Instead of phloem, it is leptoids in bryophytes. So you can see that this is a small plant. Here you can see the scale is 0.1 mm. It's a very small plant, but this sporophyte is very long. Up to the seta is very long here. And here also you can see very small plants. These are all microscopic, but you can see some plants like Fondinidis antipyretica and some other pleurocarpic mosses. It comes up to 50 to 60 centimeters long. So it is nearly 2 scale length. So how the water and minerals reach the top of the plant? So there should be some mechanisms. So now it is the bryophytes are termed as, then it is termed as non-tracheophytes because the vessels are absent in bryophytes. But the hydroids and leptoids instead of xylem and phloem is present in bryophytes. So we can say that they are non-tracheophytes instead of non-vascular plants. So these are some of the photographs. You can see that this is hanging. So up to this area, there should be some minerals and water absorption up to the tip of the plant. So coming to the Macrantophyta, that is liverworts, it is again divided into 2 types, that is thalloid and leafy type. Thalloid means thallus-bearing plants. It is normally Erixia, very common in Tamil and Kerala. So some mechanisms from thalloid form to leafy form or erect form, there should be some intermediate zones. So in between thalloid and erect leafy plants, there should be some prostrate thalloid form. Here you can see that, like this plant shows some prostrate manner. So this is a thalloid form, Rexia, very common one. So this is Lularia. It's also very common and high altitude areas of Tamil Nadu, especially in Kodaikanal and Ooty. Then Ribolia, it's also very common species in the high altitude region. Thallus is prostrate, dorsiventral, and it is greenish in color. So this is a leafy form. So it's also high altitude species. That's why it looks like a dark blackish brown plant. Then Leucologeni, these are all epiphytic leafy forms. So just I am showing the categories of bryophytes. So coming to mosses, mosses are also divided into 2 types, that is acrocarpic and pleurocarpic mosses. Acrocarpic means the plant is erect. You can see here. This is only a single erect plant with a sporophyte at the tip of the gametophyte. So here in the lower side, you can see that it is highly branched. So there is a main branch and it is highly branched, and each branch bears sporophyte. Here you can see a number of sporophytes. That is called a pleurocarpic. So here it's a diagrammatic form, that is the main branch, and several lateral branches will develop from that. And each lateral branch possesses sporophyte. That is mosses, acrocarpic and pleurocarpic mosses. So here, this is a common one. You all know that. You all say it is polytrichum, but I would say pogonatum. So polytrichum is not found in South India. It is only pogonatum. So this is an acrocarpic moss, and the sporophyte is produced at the tip of the gametophyte. There is no branching. So this also, it's a common low altitude species, Bryum. So this is a pleurocarpic one, Macrometrium. So this is a single plant. It's highly branched. So then Floribundaria, this is also pleurocarpic one. So here you can see on rocky patch, the Thuidium pleurocarpic one. So coming to the classification details, say, so the term bryophyte was coined by Alexander Braun, a German scientist. So these are all from the original manuscript, Braun in 1864. That is first, the bryophyte is given the name bryophyta by Alexander Braun in 1864, and in Schimper, it has been elevated as a division. He's also from German, that is in 1879, and this division bryophyta is divided into Hepatica and Musae in 1883 by Eichler, and then Eichler in 1892 followed Eichler's classification and divided the two classes into three orders, that is the Hepaticae as Marcantials, Gengamanials, and Anthocerotes, Musae, and Sphagnales, Andrials, and Bryals. But the anomaly is that the Anthoceros is included under Gengamanials by all these scientists. That is, Eichler also included like this in 1892. And Howe in 1897 noticed the anomalous position of the order Anthocerotes and changed the class status to Anthocerote. That is, Bryophyta is now given three classes, that is Hepaticae, Anthocerote, and Musae. After 1897, we have three classes. But there is, after that, this is the base. This is the base of classification. That is, in 1897, Howe classified Bryophyta into Hepaticae, Anthocerote, and Musae. So after that, there are several classifications by Scherber, Cavers, Smith, etc., etc. In 1951, Rothmeiler suggested changes in the name of classes. That is, Hepatica into Hepaticopsida, Anthocerote into Anthoceropsida, and Musae into Bryopsida. And in 1957, Proskier again changed the name of Anthoceropsida to Anthocerotopsida. And the Proskier's classification, that is in 1957, is the base of Bryophyte classification. That is, it includes Hepaticopsida, Anthocerotopsida, and Bryopsida. After that, there are several classifications, and recently some changes have been given, and the Bryophytes have been given the subkingdom status, and the name Bryophytes have been changed to Bryophytina or Bryobiophyetina. It is now the research is going on the molecular level to change the status to subkingdom level or to retain the Bryophyta as a division. It is going on. But there are some books are following the Bryophytina into three divisions, that is Bryophyta, Marcantiophyta, and Anthocerotophyta. So we can say the common names, the mosses, liverworts, and hornworts. It is there, but the scientific status is changed. And this is the recent trend, that is Bryophyta, Marcantiophyta, and Anthocerotophyta. So coming to the bryosphere, you have all heard about the spheres, that is hydrosphere, lithosphere, like that. But I would say it's a bryosphere, that is, it's a vibrant world of small, very small plants of high impact on the ecosystem. So why it is said like that? Have you heard about the Robin Wall Kimmerer? So she is an American plant taxonomist and belongs to First Native people of Potawatomi. That is, it is a native aborigine of America. She's a plant taxonomist in one of the institute in America, but she is a native aborigine. And she was written a good book that is Gathering Moss, a natural and cultural history of mosses. So in that she says that the mosses were successfully survived in the earth for more than 3,500 million years. And she asked how it happened. So because there are for centuries floods, tsunamis, earthquakes, then several pandemics. We have faced several pandemics. Now also we are facing pandemics, and several species of animals and plants have been vanished from the earth, but only mosses have been survived. How it happened? So she is asking a question, well, how it happens? And she simply answered that it is a very small, beautiful plants on the earth, but very delicate plants. They take very little from the earth only for their survival. And in turn, mosses give back to a lot to the nature, like giving a wonderful climate to the forest ecosystem. And most of these species, small species, survive with the aid of these bryophytes. So, and there are several economic importance for this small plants, and they play a very important role in the ecosystem. But mosses are these species who have utmost forgiveness, because during drought or scarcity of minerals or water, they wait so calmly for the next season to come. So we all know that after a rainy season, there is no bryophytes. When we water only, it will thrive. Otherwise, normal condition, there won't be any bryophytes in the nature. So in forest, it may be there, but in a normal low altitude area, there is no bryophytes. At that time, during this wonderful season, they start their healing process to survive for the next season. So when it comes, the next season will come, they will sprout like a new baby. So Robin simply explains a very simple manner by citing a moss as an example, how beautiful the earth is, and nature has enough resources for all the creatures to live, but we should use it wisely, better way for sometimes or for better ways, better days. And she has explained all this by understanding the nature only, not because she is a scientist. So that much is bryophyte. How vibrant the world is, we can say that. So like that, you can see that here is a wonderful tree. So I am taking a bryophyte class. So you can say, you just see this. So there are some bryophytes. You can say that there are some bryophytes. But I can say that in that plant, it's a very small tree, and it is not seen in the mountain forest, and it is this tree is found on the semi-evergreen tree, and you can see that it is open. But I can see 1 to 15 species here. So there are several species growing in a single tree. That's the diversity of bryophytes in Western Ghats. So I can say not just in Kerala, in Tamil Nadu, Karnataka, all Western Ghats, this type of trees are seen. So we are just seeing a tree like a tree only, a flowering plant only. But I can see this plant like a host for several other mosses. So when we take a bunch of plants from this tree, so we can see that there are more than 15 species in a single tree. So this is only a diagrammatic representation. I have taken this from one entomology book by Willimer et al. In 2000. So here you can see that the light is penetrating into the plant, this very small plant. So here you can see that the sunlight within this area, within this small square meter area, there are differences by the plant accumulate light in a different source, different manner. So 32 degrees, 27 degree, 31 degree, 33 degree. So this is a small area. So also in this case, you can see that there are several species of insects growing or residing in this plant. And depending upon the availability of the light, the species may change from the leaf tip to the base of the plant. So the physiology may change. Like that, in a single tree, there are several species of bryophytes host, and there are different types of microhabitats for each bryophyte. So the habitats of bryophytes, we can say there are epiphytes, terrestrial one, and aquatic one. So epiphytes, only a single tree you can see from base to top. So from base of trees means buttresses. This area may different in lower part of tree trunk, upper part of tree trunk, and branches and twigs. The diversity may vary in all these zones. And epiphyllous forms, living leaves, then dead leaves, all these are epiphyllous. So we can say the diversity of, if the leaf is seen in this plant, the diversity we have seen in the buttresses or the lower part may vary in epiphyllous forms. The rhizome of ferns also may vary. The fern rhizome is a very good host or abode for several species of bryophytes. So in a single tree itself, we can see different types of bryophytes. That would be better you will understand by seeing these photographs. So this is the buttresses of forest trees. So in an evergreen forest, we can see like this. Buttresses means the rooting region of the trees. It is exposed in the area. So the species growing in this area may vary in this area. This is the same tree. So in the bark upper portion, you can see different species. So in the bark, the availability of the light, the availability of the water, the availability of the substratum, it may vary, and depends upon this variousness, the species may vary. So you can see the bark, there are different types of species growing. This is the leafy forms. So it is very difficult to see, is this a bryophyte or a lichen? It is very difficult to, it is confusing. So here also, the diversity may vary. It's a tea plantation. So the bark may vary. So here also, here you can see the branches and twigs. The branches and twigs are good abode of a good number of species in high altitude forest. That is mostly in the shola forest and evergreen forest. So here also you can see several species. So in a bunch of, I had said that in a bunch you can see a number of species. So this is also another photograph from Ambalapara of Kannur district. So here you can see several species of bryophytes growing along with the ferns, and some orchids also see. So this is the branch of, this is another branch of this same plant. So here you can see another species. So this is not from internet, it is a live plant from Anamudi Shola National Park. Okay, from base to top, you can see a good number of species is hanging on branches. This is one of my new species hanging, Aerobryopsis cerevigolomensis. So it's also myristica root. Higher roots also possess some species. It's also aquatic. So on logs, logs means dead plants. So here in logs also, there are several species which may not grow on the live plants. Okay. So this is epiphyllous. Here you can see an angiosperm leaf. So there are several species growing. This leaf itself may contain 2 or 3 species. So here also you can see. So here, this is the zoom photograph. So this is the photograph of epiphyllous species on leaves. Mostly Legionnaceae members are seen on this plant, except some species like the Ricardia. It is mostly grows on the rhizome of ferns, or this is a Pyrosia fern. So the bryophytes are growing on the fern surface also. So this is Cyatodium. It's a fern. And this is another species, sorry, Cyatodium is a bryophyte, a thalloid liverwort, and another leafy liverwort growing on the surface. It's a Cololegionum. So coming to the terrestrial form, the soil and rocks are divided into wet soil, dry soil, and rocks also divided into big rocks, small rocks, etc. The diversity of all these substrata may vary in each species. So here you can see the cuttings. So all these are bryophytes. Astrella growing vigorously. Here also you can see line cuttings. This is for Sombronia. This is Physidans. So line cuttings. This is Anthoceros growing on the cuttings. This is Astrella, the male thallus of ants, Astrella. Male and female are separate, dioecious species. The male thallus you can see like this. So this is another Brayum argentium. It is a whitish moss. So this substratum may vary, the terrestrial forms, it's banks of river. It may vary. It looks like as a betch. The species we can take it as a betch. So this terrestrial one, Rixia, common one, and normally the Rixia grows on the terrestrial habitat. It won't see on the land cuttings or rocky patches. It's very rare. So this is also Australia growing luxuriantly on the bank of river. So the creeks and crises of rocks, only very few species are seen on the creeks and crises of rocks. Anthosaurus you can see. So there's some species of fissurants also we can see. So some amount of light is available for the growth of bryophytes. It shows that like that. So on termite mound you can see here, it's very few species are seen on the termite mound. The substrate, I mean the nature of the soil also vary in termite mound. That's why it holds only very few species. So you see, this is a rocky patch. It is not grass. This is grass, but it is a species, Campylopus species. It grows like grass in high altitude regions. So this is another species growing in the rocky patch. It's also high altitude species. This is Macrometrium growing on the rocky patch. You can see that this is calcareous species, mostly seen on the rocky patch. So coming to the aquatic also, we have studied that one aquatic species, that is Rixia fluitans. Not a single species. There are several species growing as aquatic one in stagnant water and running water. We can see Rixia fluitans only on the running water. We can see stagnant water. This is Rixiocarpus. It is seen on the pond. So in stagnant water, it may float or submerged one. In running water also, it may float or submerged one. This is Rixia fluitans floating on the running water. So that's two species of Rixiaceae, that is Rixia fluitans and Rixiocarpus natans. Rixiocarpus is another genus. It looks like Rixia only, but the nature is very different. There are a good growth of scales you can see on the side. And there are several species of mosses which grows in aquatic regions. So coming to the herbarium preparation of bryophytes, it's very simple. We can take bryophytes in blotting paper or newsprint packets. So the terrestrial or lithophytes, we can scrap it out from the substrate with a knife and remove the bulk of the soil, leave a thin layer, because some species possess, the rhizoids possess tubers. So tuber is an identifying character. So you can keep some sort of soil in the substrata. So, bryophytes in aquatic species, we can collect as such, no problem. In epiphytes also, we can take it, a chip of bark we can take, and small ticks we can take it as such. Leaves also, fresh or fallen, we can take it as such. And we give the bulk samples were numbered from the field like 9462. And coming to the lab and give, I have told you that in a bunch of collection, there are more than 5 or 6 species. So then we can say A, B, C, D. That can be done when we come back to the lab. So the field, this is the herbarium, very small packets, 5 into 4 inches size packet, and we can keep it as such. There is no twining or tagging is needed for the herbarium. And the field data includes the collection number, locality, date of collection, altitude, microhabitat, macrohabitat, substratum, associated species, collector's name, and ultimately who have done the identification, determine which. Okay. So the liverbirds, the dry preparation and liverbird wet preparation also we can opt. For liverbirds, wet preservation, 70% alcohol or 4% formalin is enough. In dry preservation, it is air dried packet. We can dry the specimen by keeping or in the lab itself. So there is no need for oven dry. So and store the paper packets in 5 into 4 inch size, 12.7 into 10 centimeter like that, and keep all the packets in like this. This is this especially is meant for the herbarium packets of bryophytes. So coming to the bryophytes of Kerala, there are 50 species. I have started with the 230 species in 2000 and now it come to up to 750 species, around Moses 505 species, 15 species of hornbirds and 232 species of liverbirds. And there are several new taxa published, 12 taxa of bryophytes, and 2 are angiosperms, and new distribution records: Asia 3, India 18, South India 36, and Kerala 85. This also shows that there is lack of the survey in the case of bryophytes. So coming to the history of the bryophytes, that you have all heard that Hothus malabaricus, that is written in 1693. In that book, one bryophyte is represented. That is, it is written as Puempeda, Puempeda, and it looks like the back of hen's back. So Puempeda, and it is written in Latin also, and later it is interpreted by Dr. K.S. Manlal by Brian Bicoller. And this is the in a single slide, we can represent the bryophyte diversity in Kerala, because very few workers are working on the aspects of bryophytes of Kerala and Tamil Nadu itself. And Tamil Nadu only Sahai Sadish sir and Dr. Dulid Daniels, both of them are doing some works. And Scott Christian College, Nagargoil, and Sahai Sadish sir from Trichy. And 1983, only 83 taxa is there. And up to 2027, 750 species we have published. And up to this 2020, 11 PhDs are reported from Kerala and published from Kerala. So among these 5 are from our area, and 1 is my thesis, and another 6 PhDs by 2 of them by Daniel sir and 2 from Delhi University. So some of the workers from outside Kerala also doing bryophyte studies in Kerala, but that is not that much rigorous work is going on bryophytes of Kerala, and the estimated diversity is nearly 1,500 species. This is the graph, that is from 83 to 750 species, is huge amount. And we have published 2 books, that is Bryophytes of Wayanad in Western Ghats and Bryophytes of Kerala, Liverbirds Volume 1, and this book, that is Bryophytes of Agastmale Biosphere Reserve. It's part of Kerala, and it is published by Daniel sir. These are 12 new taxa published by our team. These are some of the species: Trichostomum wayanadense, then Amphidium ganguli. Ganguli is the father of Indian Moses, and named after his name. Then Fissurans camadensis, camadum, it's a Seka Grove in Kasaragod district. It's an aquatic species, that especially you can see that it is an aquatic species. And Symphysodonilla Madhusudanane, the species also named after my professor Madhusudanan sir. This is Aerobryopsis irregulomensis. You heard about Irregulam National Park in Idukki district, the first national park in Kerala. So it is named after this area. Then Chilocyphus chinnarensis, it's also from Chinna Wildlife Sanctuary. And this is Cololegina manilaliana. So you know that case Manila, he has written the translated the Hothus malabaricus to Malayalam and English, and recently he has been awarded with the Padma Shri Manila, case Manila. This species is named after him, Cololegina manilaliana. And this is Fissurans manilaliana, another Fissurans species, and another species of Rixia sahyadrica. It's very interesting. So you have all studied that Rixia has 2 regions, that is photosynthetic, upper photosynthetic region and lower story region. But in this case, we can see that the lower region is with the photosynthetic region, upper region is with the story region. So the further studies, we have published it as a new species along with Dr. Cargill from Australia. And they have also collected 1 species similar to this Sahyadrica. So the further studies are going on. What was happened in this species? So this is Micrometrium varanicum. So here you can see the this Protonema. So this is very small microscopic plant. So here you can see it's very small plant, and the sporophyte, here you can see the sporophyte after this post mature, and it is dispersed and it produces the Protonema. From the field itself, we can see the Protonema. So this is the Protonema live specimen. So another new species, Ficedens crispus, variety Nelyambadiya. It is named after Nelyambadi Hills. And this is Brachychromia malabarica, is from Malabar Wildlife Sanctuary, and this is about to come. And coming to the altitude and bryophyte distribution, the altitude is very influential for the dispersal of the bryophytes. So we have seen several species. In degree classes, we heard about only Rexia, Markantia, Funaria, like that. And PG class, we also heard Rexia, Markantia, Funaria, Australia, Targionia, or up to 8 to 10 species. So I have shown you several species which is distributed in the high altitude and low altitude region. So in low altitude regions of Kerala and Tamil Nadu, we can see nearly 30 species. It's very common in low altitude areas. But in higher altitude, all other species are found in the higher altitude areas. And the species growing at the high altitude and low altitude is very less. So here you can see some lower altitude species, that is Octoblepharum albidum. It is known as whitish moss. So here you can see it is whitish in color. When we give some water to this plant, it will become greenish in color because it shows the leucocyst cells. That is why it looks like whitish color. So Rexia, it is Rexia billardii. It's very common species. Rexia frosti. It's also seen on the low altitude area. Sometimes it also seen on the medium altitude also. So very common fluorescent species, that is Iathodium cavernarum. It's very interesting to see that this sporophyte is produced on the ventral surface of this plant. It's very common in the low altitude area. This is Ricardia multifida. It is seen on the secagrues, low altitude secagrued areas, and it is not seen on the other areas of open land. It's seen on the secagrues only. This is also Palavicina lily. It is seen on the low altitude and high altitude area. It's seen as a ribbon-like thallus. So this is Anthosaurus ancustus. Anthosaurus is found on the low altitude and high altitude also. Some species are seen on low altitude. This Anthosaurus and Notothylus also seen on the low altitude area. We can see here in our campus itself the Anthosaurus, the species of Anthosaurus. This is a high altitude species. This is found on the low altitude and high altitude region. This is Anthosaurus crispulus. This is Notothylus leveri. It's found on the, it's a species of Notothyladales of Anthosaurotopsida, that is hornbird. And this is Plagiocasma appendiculum. The species also, it is seen on the low altitude and high altitude regions. So this is very common species, Braham coronatum. It's a low altitude species, and this is also a low altitude species, Hyophila involuta, very common in Tamil Nadu and Karnataka and Kerala. Ficedens ceylonensis. So this is also very common in low altitude. It's seen on the rocky patches, calcareous soil. It is seen, Gymnostomela vernicosa variety tenerum. So this is also seen on the cuttings, wall cuttings and rocky patches and several tiled areas. So here you can see some of the species growing in the high altitude area. This is Brachymenium. It's seen in the family Bryaceae. So this is a shola species, that's why it looks like brownish black in color. So this is another species. This is Funaria. Funaria hygrometrica is found only on the high altitude region. We cannot see Funaria on the low altitude. So earlier in +2 degree classes, we have studied Funaria is growing on the low altitude region, but it's a misidentification. That is, we have studied only Bryum and it is named as Funaria. In colleges also, we have studied only Bryum, but named as Funaria hygrometrica. This Funaria is found only in the high altitude region. This is Macrometria. The species is seen on the rocky patches and also on the high altitude trees. That is Gotchalia. It's also very interesting species. Looks like it is a leafy liverwort and it looks like a brownish blackish in color. This is Imbribrium alpinum. The species is found only in the alpine region, that is mountain region the species is in. That's why it has a reddish color in the plant body. This is Astrella leptophila along with the Pogonatum. There are several species. Here you can see some other species also. So this is Campylopus. It looks like a grass. So we, without knowing that it is a bryophyte, we will ignore this plant as a grass. Earlier, when I have started my studies on the bryophytes of high altitude region, so I have first misidentified as it as a grass. So when we took the plant to the lab only, we came to that it is also a moss. So this is another species of Campylopus. It is a dioecious species. And this is Pogonatum microstomum, a Leucobryum. This species also, we can say that it's a whitish moss. It is seen the leucocyst cells we can see here. So this is Bryum. This is Markantia polymorpha. Here you can see the gemme here. So this is the sporophyte also you can see, and radiophore you can see. So this is the largest liverwort in our area. That is Dumortira hirsuta. Here also you can see the sporophyte. So this is Plagiocasma appendiculatum. Here you can see the male plant and female plant. This is male plant and this is female. You can see the sporophyte. In male plant, you can see it's a dome-shaped structure with a blackish antheridia structure. This is Lunularia. Here you can see the crescent-shaped gemme here. Here you can see that. This is Australac hasiana. This is also high altitude species, Rebolia hemisphaerica. Then Tardione hyperphylla. The interesting is that this is the sporophyte of Tardione hyperphylla. Tardione hyperphylla, the antheridia and archegonia produce at the tip, top of the plant, that is dorsal surface. At the time of reproduction, the archegonia will move back to ventral side of the plant and it possesses the sporophyte there only. So it's only a protective mechanism. So this is Pallavicina ambigua. Pallavicina lily is found on the low and high altitude area, but this species is found only in the high altitude region. So this is Frulenia. Here you can see the additional leaf here, underleaf. This is the ventral surface. This leaf also functions as an absorptive organ. So this is Porella. Porella is a very large species. Then Rhodobryum giganteum. This is the largest moss. The largest liverwort is Dumortira hirsuta and largest moss is Rhodobryum giganteum. So this is Hypopterygium tamarisi, Pogonatum microstomum, Bryum argentium. This is also known as silver moss. So Trematodon longicolis. So what is the difference between Bryum, Hyophylla and Funaria? Most of the people may ask what is the difference between Bryum, Funaria, etc. So this is Bryum. So you can see the gametophyte and sporophyte. So this is the sporophyte young capsule of Bryum and this is the mature capsule. This is the difference from Funaria and Hyophylla and Bryum of in capsular nature. That is, it possesses a cup inside a cup. In Bryum, you can see the capsule as cup inside a cup. But in Hyophylla, it's a very common low altitude species. It possesses an elongated sporophyte. In Funaria, we can see a hanging sporophyte. This is this sporophyte here. Here you can see there is no peristome at all. That is the difference between Funaria, Hyophylla and Bryum. But all the gametophyte looks alike. That is the difference. The Funaria, Bryum and this Hyophylla possess similar type of morphological nature. So only difference we can see when the sporophytes only. So coming to the importance of bryophytes, there are several horticultural impulses. The moss garden at Sanchico, Ito, Japan, we can see a moss garden, moss cultivated stone garden. And there are several species which is used in bold cultivation and landscape trees, ground cover for bonsai, mulching, road packing materials. The sphagnum, hypnum, impertinence, these all species are used in the horticultural. Yeah, you can. This is a photograph from Kyoto, Japan. That says a moss garden. Say they have made several sculptures, and in that sculpture they have given several species of bryophytes spores. So in that area, several species of bryophytes are grown. So this is not a grass; it is a bryophyte. So similar kind of moss garden is in our area in Lucknow, that is in NBRA, National Botanical Research Institute. This photograph I have taken during 2009. They have started in 2009 only. At that time, this is like this. And 2015, it looks like this. So almost all areas, the bryophytes are spreading. So by giving the conditions in Lucknow, it is almost a temperate condition most of the time. That is in 6 months, it is very calm. And all other time, that is next to 6 months, they give watering, misting, etc. So we can make most match. So it's also useful like this. So ground cover for bonsai you can use. So in nurseries they use, so these are resurrection plants, they can use like that. So some birds use bryophytes to prepare a nest. So substratum for higher plants, this is sphagnum. So this is Nepenthes. These spores may get sprouted in the sphagnum. So several species of higher plants may grow and they use bryophytes as substratum for higher plants. So this is also an orchid, Chirostylis, it's growing along with the macrometrium. This is also a method for growing this orchids. So, and ecologically, it is very important. That is soil binding, rock binding, nitrogen fixation. That is all Anthus rotales members possess this noster colonies. Then substratum for higher plants are bioindicators, but very less studied in our area. So several species have been used in medicine. That is Philonottis, Brayum, Niam, etc. are used for relief in pain of burn. Then Polytrichum commune, it is boiled, it is known as hair cap nose. It looks, the calyptra looks like a hair of ladies. So it is used for boiled to made a tea for treating common cold, and extracted oil also used for beautifying the hair. So this is happening in China. So sphagnum in hemorrhages, it is used as a better absorbent than cotton. So when we can keep the sphagnum or any bryophyte in dry condition for hundreds of years. So when we keep it in water, it regains its original shape. And during the World War time, the scarcity of cotton has occurred. And at that time, the sphagnum is used instead of cotton. So it's also used in surgery. So peat moss, you all heard about peat moss. It's a miracle grow. It can be used as a packing material and also for mulching and road packing material, and also used as a fertilizer. So there are several antimicrobial and antifungal properties, and several active constants are there, but the studies are ongoing. So some insects use or eat the capsule of some species of bryophytes. So this is an elementary canal of one insect. So you can see a cut end of leaf inside the insect. So this is a Rexia species. Here you can see a pupa stage of insects. So several this kind of interactions are going on in atmosphere or nature, but the studies are very less regarding this. And the bryophytes are also used as index of atmospheric purity. Some species may treated as pure in the area. So most of the studies are regarded with the lichens, but the studies on bryophytes are very less. And some studies shows that Atricum and Rulatum is one of the of air pollution. So damage to ecosystem, then indicators of climate change, and thereby study its effects on ecosystem. There are several studies are going on in this line. And so there are some species are specific growers, that is, it is found only on the mineral rich areas. Some species are copper moss, it is Melicophora elongata, Scopulophylla ligulata, it's Scopulophylla catracta, these are all known as copper mosses. And due to these properties, these species are used in geological prospecting. This type of studies are also going on in the area. And coming to the conservation issues, I have told you that the bryophytes play a very important role in the ecosystem dynamics. But who says one should conserve the bryophytes or pteridophytes or lower plant group? Nobody will say. So we can say that some charismatic species like elephant, tiger, then lion-tailed macaque, like that, we can say that. But no one says Physidens camadensis, then Aerobryopsis, irregularmensis is there, so we should conserve them. So bryophytes are not considered by the stakeholders or even the naturalists due to its smaller size and not so much direct economic importance. That's the very important case here. The trained workforce is very limited in Kerala for conducting studies on bryophytes. In Kerala, not only in Kerala, in Western Ghats itself, there is no trained workforce areas. So in Kerala, only in Guruvayurappan College it is happening. So in Tamil Nadu, it is happening only in two areas, Scott Christian College and one in Trichy. And there is no proper funding to maintain these aspects. So there is no publications, especially books on identification manuals. It is lacking in this line. And students and research scholars may be prompted to undertake studies on these groups. Biodiversity manners and conservation planners may consider to include these groups or plans also in their programs. So for that, we have to raise our voice. So I will says a few words about the Bradford researchers, that is major research centers in the world and in India. So you can search in the net, that is New York Botanical Garden, Kew Botanic Garden, Missouri Botanic Garden, Singapore Botanic Garden. In Missouri Botanic Garden, you can see that tropicos.org, you can see all the species of the world are revised there. So we can, they see the nomenclature changes in that Missouri Botanic Garden site. So in India, National Botanical Research Institute, very few research centers are there. NBRI Lucknow, then Lucknow University, Lucknow, Botanical Survey of India in Calcutta and Dehradun, Scott Christian College, Nagarkoil, and Tiruchirappalli, St. Joseph's College, then Samurin Zurarippa College in Kozhikode, so in Kerala. So these are only the research centers in India. So altogether in world itself, their research centers are very few concerned with the bryophyte studies. And these are some of the books. That is, these are Ganguly's book, that is Moses of Eastern India and Adjacent Regions. That is in 3 volumes, and it is written, they have described nearly 2,793 species. So these are some other books on Moses illustration, liverbirds Moses of Europe. And these are all identification manuals. So these are some Indian books, that is current trends in bryology. It's on genus Plagiochyla, on Hepatica of Eastern Himalayas, Potassium of India. And coming to the last slide, this is our small team. So 5 of them have completed their PhD and 4 are ongoing. Thank you. Thank you very much. I think I have taken 5 more minutes. Sorry for that. So any doubt you can ask, time is running. Any doubts? So nobody is responding. Chat കാണുന്നില്ല. Chat അവിടെ എന്താ മെസ്സേജ്? അല്ല ഇതിൻ്റെ അകത്ത് ആരും ഒന്നും റെസ്പോണ്ട് ചെയ്യുന്നില്ല. മെസ്സേജ് ഉണ്ട്, ഓക്കെ. എനിക്കിതിന് എന്താ ചെയ്യാം? അല്ല ഇതെന്താ ചെയ്യാം? എന്താ സംഭവിക്കുന്നത് മനസ്സിലാവുന്നില്ല. സാറ് വന്നോ? എവിടെയത്? ഇതെനിക്ക് ഓപ്പൺ ആകുന്നില്ല, അതാണ്.
Ma'am, is it audible?
Ma'am,
Participants போயிட்டு ask to unmute.
Sir.
Ma'am, is it audible?
Yeah, it's audible.
Yes, ma'am. Yes, ma'am. Thank you.
No problem. I have answered some questions. Some of them have asked about the secondary metabolites. So for against COVID-19.
Okay, ma'am.
Bryophytes, someone is asking. So Sadish sir is asking something. Sadish sir. Hi Sadish sir. Please share your PPT.
Sir, ma'am is on line, sir. Please unmute your audio, sir. Sir, Sagai, sir.
Anybody wants to ask any questions? We can ask now.
Okay.
So, sir. So, how is you, sir?
Oh? Sir, unmute yours, sir.
Yeah. Now. Okay. Congratulations, ma'am.
Thank you.
Excellent presentation. I myself with my scholars attending the whole presentation. Presentation was very nice and inclusive. Very nice photographs.
So within 45 minutes, it's very.
But you're starting from the unique features up to the conservation strategies. You took everything with nice photographs, not like other speakers. Words and more photographs.
Thank you, sir.
You proved yourself as a having expertise in this bryophyte. So we are all the witness. We are all the witness. Myself, like me and many bryologists, nowadays doing research because of you, your help.
Thank you very much.
I consider yourself as a goddess of bryophyte. Very number of new species you explored. Our wishes and prayer for your future endeavor in this noble work that is the neglected group bryophyte.
Thank you, sir. Thank you for
very few people. So for the courtesy, I don't have any doubt in your presentation, but I want to ask from away from your presentation.
Okay, sir.
The general question.
Yes, sir.
Because nowadays many bryologists or other researchers working on antioxidant, antimicrobial work in mosses and other bryophytes.
Yes, sir.
These mosses, these bryophytes are comfortably or only growing in the comfort zone. The comfort zone means the climatic condition, other moisture, everything will be comfort. That place only it will flourish. So what is the necessity to they adapt, biochemical adapt or produce new phytocompound? So those plants in the angiosperms, they are producing new phytoconstituents because they have to adapt or acclimatize the environmental stress or any other stress. But these plants, these bryophytes also started producing new phytoconstituents. They are exploiting in nanosynthesis, antioxidant. So why this?
So that's the answer, sir. It's maybe due to the acclimatization characters. So the species growing in the high altitude area, which we taken from that area, and we give a very similar condition in the low altitude area. So it may start adapt to the area. Sometimes it may thrive. So in the high altitude species, it is very difficult to thrive in the low altitude areas. But sometimes it may thrive by developing certain secondary metabolites or some other phytochemical constituents like that. So that may be due to the character. Maybe like that. I think so. That's a thing.
Yes, ma'am. Thank you. Thank you very much, ma'am. Thank you.
So I think you are better than me to answer that.
No, just to share.
You are doing such works.
I just ask for the betterment of the audience.
Yeah.
So no,
It's very
sharing only. Thank
so
you
in the
very much.
high altitude species, there are when we consider the high altitude species and low altitude species, the comparison is very difficult. So in bryophytes itself, it needs such condition. So high altitude bryophyte species, it's very difficult to thrive in low altitude area. When we go into the deep forest, we can see that the gametophyte, it never dies. It never dies. The gametophyte thrive for long. And it produces sporophyte only in a certain period. That's the case. But when we take that plant to the lower altitude area and we give the same climatic condition, it may grow for a certain period. After that, it will die. And the next season only it will come.
Yeah.
So that's why I had told you earlier, that is Robin has written in Gathering Moses, Natural and Cultural History of Moses. In that she has explained very well in that the characters of the Moses, like that. Okay,
Thank you very much.
Thank you.
Thank you, the organizers, for having this wonderful occasion.
Thank you, sir. Thank you, ma'am.
Yes.
Ma'am, our students asked some doubts.
Okay.
I will call them. Thank you.
Okay.
Good evening, ma'am.
Good evening.
Ma'am, I'm SP Rashmi from 2nd PG, ma'am.
So I can't hear you. I can't hear you properly.
I'm SP Rashmi from 2nd PG, ma'am.
Oh.
I have one queries, ma'am.
Just tell me.
Does any insect attack?
So there are several species of. So earlier it is known that the insects attacking is very less in bryophytes due to the occurrence of the secondary metabolites in that. But now it is identified that some species of, I have shown you one species, Evalia and Philota of insects that attack the Rhodobryum giganteum, capsule of Rhodobryum giganteum, that eats that species. And another elementary canal I have shown, one elementary canal, in that we can see a moss leaf. So the insects use bryophytes, some bryophytes, but that much studies are very less. That's maybe they are using, the insects are using bryophytes for eating purposes or some other purposes, and some insects may take as a host. So it's an abode for several insects. So that type of works are very less here in our area, and in worldwide also. This type of works are very less.
Thank you.
So that is why we are unaware of this type of activities.
Good afternoon,
Ma'am.
Ma'am, my name is Nidita. I'm a student of Botany. I have one question, ma'am. Ma'am, which classification is suitable for bryophytes in identification?
So there are several classifications, but recently Goffinet et al. 2009 and Soderstrom et al. 2016. So these two classifications we are following. That includes the combined action of molecular and morphological characters. They have included the molecular and morphological characters to classify the mosses, liverworts, and hornworts. So that is available in the net itself. We can Soderstrom et al. 2016 available in internet, and Goffinet et al. 2009 also available in the internet. So we are following that classification.
Thank you.
Thank you.
Ma'am, thank you, ma'am. Thank you so much. I'm Dr. R. Vinod Kumar, convener of this conference. First of all, your presentation is very nice. I'm so admired.
Thank you very much.
Okay, ma'am. Ma'am, first girl asking one question regarding any insects or pests are
I can't
attacked.
hear you properly, sir.
Ma'am?
Hello?
Hello? Audible, ma'am? Hello?
Hello?
Ma'am, it is
Hello?
audible?
No, it's breaking. The sound is breaking.
Okay, ma'am.
Now it is okay.
Now it is
okay.
audible, ma'am?
Hello?
Now it is audible?
Now it's okay. Now it's okay.
Ma'am,
Yeah, I can hear.
First girl asking one question regarding any insects or pests are attacking the bryophytes. So my question is also
Yeah,
same.
the earlier student asked me like
Yes.
that. It's that.
My question is on the same line, which means in fungal disease, many diseases we studied, like Zika disease, Vaccinia disease in the leaf. Like that, particular diseases are found in the bryophytes.
There's not that much studies in bryophytes.
Okay, ma'am.
No.
Now our
studies are not gone, and we have not noticed any disease in bryophytes. That's the thing. With my field experience, it is seen that the bryophytes are intactly. We can see there is no insect attack or breaking of leaves or anything. We couldn't see like that. We have not seen such an incident. So recently only one of the entomologists from that is Trivandrum Agriculture University, he has shown that some insects, he is an entomologist, and that Ivali and Philota, I have shown you that that is a new species to science, and they have reported that species is eaten by, that Rhodobrium giganteum capsule is eaten by these insects. So very little studies are going on. So you can take that.
Yeah, sure, ma'am. And
there are several scope for studies. Yeah.
Ma'am, now our PG students are studied classification by Watson. Watson classification, they are studied. Which classification? Bryophyte classification. Which one is best, ma'am?
Proscure 1957 basic classification. Proscure 1957.
In our syllabus, we mentioned only Watson.
Watson is outdated. You just tell them to revise the syllabus and include Proscure 1957 and latest Soderstrom et al. 2016 and Goffinet et al. 2009. So most of them are following that classification only.
Okay, ma'am.
So there are several changes in angiosperm taxonomy also. We are using APG classification. So we have to change all classification. In all types of plant groups, the classification has been changed.
Okay. Ma'am, some participants have many doubts. They are in the chat box.
Yeah, sir. ID is with the Murugesan. No problem. Murugesan sir has my email ID and number.
Okay, ma'am. Sure, ma'am.
So I think there is one
last one doubt, ma'am. How to differentiate the Tarjonia and Ribolia, ma'am? How to differentiate the Tarjonia and
Tarjonia? It's very easy to recognize. Ribolia, the morphologically, the thallus is very large. The tip of the thallus is very rounded in shape. In Tarjonia, it is very smooth. It is both dichotomously branched, and the Targionia possesses sporophyte at the lower surface. But the Ribolia possesses sporophyte at the dorsal surface. That is the main difference. Morphologically, it's very differentiating. But Astrella and Targionia, it is very difficult to distinguish.
From here,
so when we take this section only, we can say it is Targionia and it is Astrella.
Ma'am, then how could differentiate the Pogonatum and Polytrichum? Pogonatum and Polytrichum.
In Pogonatum, the sporophyte has 32 peristomatids. In Polytrichum, there is 60.
அவங்க அழகா சொன்னாங்க அந்த சாலிட் ஐட்டம்க்கும் கோகனட் ஐட்டம்க்கு 32 teeth are
The
present.
madam disconnect
Yeah, there's 32 teeth and
Yes, ma'am. Thank you,
so I
ma'am. Thank
think
you so much.
internet problem.
Ma'am, thank you, ma'am.
Okay, thank you.
Yes, very good evening, ma'am. Once again, I
Yeah,
thank
good
you.
evening.
And students are very inspired and very happy to hear your lecture. Once again, I
Thank you,
thank
sir.
you
Thank
ma'am,
you
on
very
behalf of organizing committee. Thank you, ma'am.
Thank you very much for giving me a chance to talk to you or interact with you. So I think in the near future we can see once more again, once again.
Sure, ma'am. Thank you.
Thank you. Okay, may I leave?
Yes, ma'am.
Okay, okay. Thank you very much. So hi, Sadish sir. I am leaving. I think he is not there.
Dr. Ramesh, sir. Dr. Ramesh,
Yes,
sir,
yes,
audible?
yes, yes, Murugesan.
Yes,
Am
sir.
I audible?
Yes, sir.
All right.
Shall we start the session, sir?
Yes, 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 sure.
Okay, sir. Good evening, one and all. Dr. V. Ramesh, Assistant Professor, Head, Department of Botany, Vivekananda College, Thiruvedagam, Madurai. He has published more than 40 research articles in national and international journals and has participated in many conferences and workshops. He has also organized many workshops and has received a grant from Science Academic Education Program, Bangalore. He has also received many awards and honors from Government of Tamil Nadu. With this, I invite Dr. V. Ramesh to deliver his lecture. Thank you.
Thank you, Murugesan.
Yes, sir.
My screen is visible. Mr. Murugesan, can you see my screen?
No, sir.
No?
No.
Still? 1 minute, 1 minute.
Sure, sir.
Now it's visible. Mr. Murugesan, can you see my screen?
No, sir.
But was said. Murugesan, my screen is past your side.
No, sir. No, it is not visible actually.
1 minute, 1 minute.
Sure, sir.
But it was already scattered. Already share my screen. Once again, let me share. Murugesan.
Yes, sir. Not, sir. Your slides are not visible.
Your side, my screen is possible. Check. Murugesan. ஆல்ரெடி அதில் ஷேர் பண்ணிட்டேனே. இல்லை ஷேர் பண்ணியிருக்கேன், ஸ்க்ரீன் ஷேர் பண்ணிட்டேன். பட் இங்கே பாஸ் பண்ண மாதிரி இருக்கு. யுவர் ஸ்க்ரீன் இஸ் ஷேரிங் இஸ் பாஸ்ன்னு வந்து. இல்லை என்னுடைய ஸ்க்ரீன் நீங்கள் ஷேர் பண்ணுறீங்களா? என்ன தெரியுது? 1 மினிட். already, but one minute. Murugesan not able to share my screen, but just you have to share my screen and share my file. You have to share?
Okay, sir. Okay, sir. Over to 2 minutes, sir.
Okay, no problem. Mr. Murugesan, now it's okay. Murugesan.
Yes, sir, visible. Yes, sir, visible. Yeah, yeah,
Right.
it's okay, sir.
Mr. Murugesan.
Yes, sir, it's
audible?
it's visible, sir,
Right,
visible.
right.
Okay, sir. Thank you, sir.
Okay. Thanks.
Kindly start,
Thanks.
sir.
Thank you. Right. At the outset, I would like to extend my thanks to the of Sri Vidya Mandir Arts and Science College and the head of the PG Department of Botany and other faculty members opportunities to share my knowledge on techniques of mushroom cultivations. And my special thanks to Mr. Murugesan because he only invited me to be part of this ICPSB-2021. For that, I also thank Mr. Murugesan. Thank you, Murugesan. All right. Am I audible?
Yes, sir, audible.
Right.
Kindly start the session, sir. Okay.
Okay. Yes. My topic is glimpses of mushroom cultivation techniques. As you know, I hope you all are familiar with these mushrooms. So what is mushroom? My lecture consists of two major parts. One is introduction about mushrooms, and the another one is cultivation techniques. So I hope you all are familiar with these mushrooms. So what is mushroom? Mushroom is nothing but a macrofungus. The mushrooms belongs to the eukaryotic heterotrophic organism. As you know, a lack of chlorophyll, they lived as a and mycorrhiza also. Even though the fungal groups are smaller one, but these mushrooms can in our own eye and they can also be picked by our hand. So this group of fungal mushrooms and structure is called stipe and the pot ring is called known as mycelium. Mycelium in the different structures of kidney or wood another plant or animal sources. Regarding the mushroom varieties, based on the literature survey, till now Murugesan, sorry for the technical issues. Murugesan, Mr. Murugesan.
It's okay, sir. No problem. You continue, sir.
But my screen is not shared right now. Please, you can share my screen file.
Okay, sir. 1 minute.
Okay.
And Dr. Ramesh Kumar, sir.
Yes, yes.
Yeah, you start the. I have shared my screen.
Okay, okay.
Have you visible?
No, no.
Share. Stop sharing. The share level. Share level.
Yes, yes. I can see your screen, Mr. Murugesan. Murugesan.
Okay, sir. You continue your session.
Right,
sir.
right. Next one, please. Mr. Murugesan, next one. Next slide. Next slide. Next. Right. This is right. Last one. Previous one. Right. This one is. Right. Am I audible clearly?
Yes, sir. You are audible.
All right. Based on the literature survey, till now there are 75,000 mushroom species are identified. Among these 75,000 species, only 20,000 species are safely edible, and 1,000 species are poisonous or non-edible. In worldwide, there are 20 species are only commercially cultivated. Regarding in India, only 4 species are grown like button mushrooms, milky mushrooms, oyster mushrooms, and bedstraw mushrooms. Next one, please. Next one, please. All right. There are 2 types of mushrooms based on the edibility. The first one is edible mushroom. The edible mushrooms are most of the edible mushrooms are non-cultivable and some are cultivable. The non-cultivable mushrooms are grown only in wild species. The edible mushrooms are desirable taste and aroma, so that nowadays the mushroom biryani is so famous in all the functions. The edible mushrooms have good nutritive values and no poisonous or side effects. Regarding poisonous mushrooms, the poisonous mushrooms looks very beautiful, colored, brightly colored. The presence of a ring or annulus in the stipe and the presence of vulva inside the soil, and also it's oozing out some milky or colored latex at the damaged portions. Moreover, it produces some unpleasant odor. Right, next one. So how do we identify these poisonous mushrooms from edible mushrooms? Right. Generally, we thought all the colored mushrooms are non-edible, but the colorless mushrooms are edible. That's not a true. But some of the colored mushrooms are also edible, and some of the colorless mushrooms are non-edible, right? Till now, there are some methods to differentiate these spiceless mushrooms from edible mushrooms. The first one is a spore print method. The spore print method is nothing but if you put the umbrella like these 5 layers on the white paper, you can see a lot of spores like that umbrella like spore print. We can't see that spore print in edible mushrooms. That is the first method. The another method is if you break any part of these fruiting bodies, you can see a latex or colored substance from that injured portions. That is edible, not poisonous mushrooms, but we cannot see these latex or colored substance from edible mushrooms. This is the two identification methods for poisonous mushroom identification. Next one, please. Regarding the nutritional and medicinal values of mushrooms, the edible mushroom, edible mushrooms contains a lot of proteins, fibers, vitamins, some minerals like copper, selenium, potassium, phosphorus, zinc, and molybdenum. And moreover, it has low level of fat, sodium, carbohydrates, and no cholesterol. So that these mushrooms are good diet for diabetic patients and heart patients also. The next one. So this is the world largest living giant mushrooms called Armillaria ostoyae. It covers 2.2 acres, that equals to 1,665 football grounds, and also equal to 9.6 square kilometer. Can you believe this giant mushroom? Is it possible? It is reported in the Mulgare National Park in eastern Oregon state of North America. It holds the world largest fungus till now. This is the phytopathogens. Next one. This is the wild mushrooms, poisonous mushrooms. It looks very beautiful and colored with red with white pages and yellow with white pages, called amanita. Our ancient peoples prepare an alcoholic beverages like somapanam. You know somapanam. Somapanam is prepared these mushrooms only. If you touch or smell this mushroom, you can unconscious, or even a death will occur within 1 or 2 days, or small, or you can eat. Right. Next one. This is the bioluminescence mushrooms. We can see these bioluminescence mushrooms in forest only, not in our domestic places. Next one, please. Right. Right. This is the king of herbs called Canotherma species. Till now, there are more than 80 species of Canotherma were isolated and identified. There are so many bioactive compounds like canothermic acid, canothermin, lanostine, glucan were isolated from this mushroom only, so that this medicinal mushroom is known as Canotherma species. The next one, please. This is the Ramaria species. That is not edible one. It has various colors like yellow, white, orange, red. Next one, please. That is the medicinal and also edible mushrooms called citric mushrooms. Called as Lendinus edodus, the first anti-tumor drug from mushrooms are obtained or isolated. This mushroom species only. The price of this species are very high. For instance, 1 kg of these mushrooms amount is 3,500 to 4,000, because it has a high medicinal value and also edible mushrooms. Next one. Right, this is the winter mushrooms. These winter mushrooms are also edible. We can grow these winter mushrooms only at 10 to 14 degrees Celsius. We cannot grow in our Tamil Nadu region, that the winter mushrooms. Next one. Right, this is the bedi straw mushrooms. If you look at these fruiting bodies, you can see the fruiting bodies like bedi straw. In Tamil, we call the Vaikol colon. This is also edible mushrooms. We can grow these mushrooms even 30 to 38 degrees Celsius. This is suitable mushrooms for our Tamil Nadu region. Next one. This is the I already told you some of the colored mushrooms are edible, right? That one is the colored mushrooms, yellow colored mushrooms, all the Phlerotus species. This is edible. I have collected this mushroom species in our Virudhunagar VHMSN college during 2018 to 2010. We also cultivate these edible mushrooms in our laboratory also. Next one. Next one. All right, this is the button mushrooms. Button mushrooms. If you look at these fruiting bodies, you can see the button-shaped mushrooms, button-shaped fruiting bodies. Next one. Right, this is the milky mushroom. In our market, this type of mushrooms are available most common because of the taste and dark white colored. This is also grow in our regions. It can grow 30 to 35 degrees Celsius also. This is also best suitable for our Tamil Nadu region. Next one. Right, this is the medicinal mushrooms called Gileria. During my PhD, I have collected 3 species of Gileria from Western Ghats of Kutralam Hills during rainy season. I have also cultivated these medicinal mushrooms in our PDA artificial medium like PDA potato textures agar medium. Next one. Next one. This is the second type of the Gileria, medicinal Gileria species. And next one. This is the third species of the Gileria. I have cultivated these 3 species of Gileria in my laboratory. I have also tested this culture filtrate as well as fruiting body extract, partial purified crude extract against multi-drug resistant bacterial pathogens and fungal pathogens. I have also tested some human cancer cell lines. They have put significant antimicrobial against multi-drug resistant and also cancer cell lines. Next one. Right. Now on, I go to discuss the cultivation part, cultivation techniques part. Regarding this mushroom cultivation, the wood air mushrooms called Auricularia species was first cultivated in China during 800 AD. Then the first commercial cultivation of these edible mushrooms was reported in France during 18th century. That mushrooms is known as Agaricus species, that is button mushrooms. The next one. Murugesan, next one, please. Right. Why we need this mushroom cultivation? Because the mushroom cultivation practice is good for both health and wealth. Regarding this health side, as I already told you, the mushroom has a lot of proteins, then vitamins, then some minerals. So this is very useful, very useful diet for heart patients and the diabetic patients. So that the mushroom cultivation mushrooms are very good for health. And the another side, it is very good for wealth also, because it is a low cost production techniques, a less investment technique, and we have more income with less investment. And also nowadays the mushrooms are the demand for mushrooms are very high. Moreover, it gives self-employment also, right? So that nowadays, if you want to become a small scale entrepreneur, you have to choose these mushroom cultivation techniques. Next one. Right. If you search in online a digital platform for value ordered mushroom products, you can see a lot of products like dried mushrooms, mushrooms soup powder, mushrooms pickles, mushroom chips, mushroom tea, mushroom juice, and mushroom charges, and some of the cosmeceuticals like face apply creams, and also we can see the immune booster capsule in Amazon like Flipkart also. So we need nowadays the demand of mushrooms are very high. Next one. Right. Regarding the world mushroom production, if you see this graphical representation, you can see the production of mushroom are increasing day by day. For example, in 1960, you can see the very minimum amount of mushroom productions are there, but in 2010 we can see the mushroom productions increasing at million tons, right? Among, I already told you, in India, 4 types of mushrooms are cultivated. Among these 4 types, world also, the button mushrooms holds the first place. Of course, the US produce a lot of button mushrooms in the world. Next one. Regarding in India, the mushroom productions are also increasing year by year. If you see this slide, you can see the productivity and the productions are increasing from 1970 to 2010. In India, there is a research center like DMR, Directorate of Mushroom Research, that is located Sulan of Himachal Pradesh, doing a wonderful research activities regarding these mushrooms. Next one. This slide shows the mushroom production map of Tamil Nadu. If you see this map, you can see the brown colored districts like Coimbatore and Erode produce a little bit high productions when compared to other districts, but still we Tamil Nadu and India the mushroom productions are very low. So we can choose these mushroom productions for become an entrepreneur, right? Next one. Right. These 4 kinds of mushrooms like button mushrooms, milky mushrooms, oyster mushrooms, and bedestar mushrooms are under cultivations in India. Next one. Now I want to discuss only 2 types of mushroom cultivation techniques. The first one is oyster mushroom, and the second one is button mushrooms. Right? In oyster mushrooms, there are 5 major steps are there. The first one is preparation of mushroom farm. Second one is spawn production, and third one is preparation of mushroom bed, and fourth one is spawning and spawn running. Right? And the last one is cropping. All right. In the genus oyster that is called Pleurotus, there are so many species are available like Pleurotus gaju, Pleurotus florida, Pleurotus sapidus, Astiatus, Jammer, Membranaceus, Stichinopliatus, and Kypsicycus ulmarius. Nowadays, people, that means cultivator, mushroom cultivator, prepare more concentrate on this Kypsicycus ulmarius because the size, texture, and shape, flavor, taste of this Kypsicycus ulmarius is so superior, far superior as compared to other species of this Pleurotus. So that nowadays people choose these Kypsicycus ulmarius for oyster mushroom cultivation. Next one. These are some different species of Pleurotus genus. Next one. Look at this Kypsicycus ulmarius. The left side corner, that one is the Kypsicycus ulmarius species. It looks like a gray colored fruiting bodies. When compared to other species, these fruiting bodies are a little bit larger than other species. This species, that is Kypsicycus ulmarius, was developed recently by Meera Pandey and Meera Pandey from IIKCHR, Indian Institute of Horticultural Research, Bangalore. Next one. Next one, please. Right. If you want to do this mushroom cultivation in small scale industry or commercial purpose, we need these are the requirements like spawn bedi straw or other agricultural waste like cotton straw or maize cobs and sugarcane bagaces and formalin or any other disinfecting agents like Dettol, ethanol, and so on. So then soaking tank, chop cutter, thermohydrometer, humidifier, polythene bag, or coconut teeth. These requirements are only for commercial purpose of cultivation, not for our home cultivations. Next one. These are the graphical representation of these requirements. Next one. Right. The first major step is the preparation of mushroom house or farm. Right. If you want to cultivate oyster mushrooms for commercial purpose, you have to choose shady places for the preparation of that mushroom house. This mushroom farm are located inside the coconut farm in our Vivekananda College, Madurai. Then only we have to maintain the environmental conditions like temperature, moisture, right? Then the mushroom farm is made up of coconut teeth or any other palm leaf. When compared to other concrete floor or asbestos sheets, this is the best suitable mushroom house. The size of the mushroom house is depends upon the cultivator or place, but the ideal form of mushroom house, the height of the central region, the height of the central portion is a little bit higher than the peripheral or sides region. Then only we have to maintain the ventilation, air, and light and other humidity, so that you can construct a little bit higher height in central when compared to the peripheral regions. Then inside the mushroom house, you have to cover a sandy soil in and the floor at the height of 5 to 10 inch for maintaining moisture content. Inside the mushroom house, you have to kept gunny bags like arisisaku for maintaining moisture content inside the mushroom house. If you want to put fence around the mushroom house, you can put. It is better for preventing the mushroom attractor like snakes, some birds, some rats. So you can put the fence also around the mushroom house. Next, if you want to cultivate these mushrooms in commercial purpose, you have to divide this mushroom house into two room. One is a spawn running room, another one is cropping room. Then only we have to supply continuously to the customer for these mushrooms. But the environmental conditions of these two rooms are slightly variable. In spawn running room, we have to maintain 25 to 30 degrees Celsius and no light. The aeration also no need. But in cropping room, I will tell you why these conditions are needed for spawn running room and cropping room later. But in cropping room, we have to maintain 23 to 25 degrees Celsius, but moderate light also need and aeration also need. Right? Next one. The second step is spawn production. So I hope you all are familiar with the spawn. Spawn is nothing but a seed-like structures formed from the mycelium of fungus in the grain-based medium. If you want to do this spawn production, you can do. Otherwise, we want to purchase this spawn from some private agents or Tamil Nadu Agricultural University or IIHR or DMR, right? If you want to do this spawn production, you need some facilities like laminar airflow chamber in the lab, and also some other facilities. Like next one, right? If you want to do this spawn production, you need a fungal tissue. Fungal tissue. That fungal tissues are washed with 70% ethanol and then 0.5% sodium hypochlorite, and again washed with sterile distilled water with the help of a sterile forceps or needle. And then with the help of sterile plate, you have to cut this a small bit of this fungal tissue. Then the tissue was tried with sterile filter paper. After the drying of the fungal tissue, you can transfer this fungal tissue into the potato textose agar medium. After 7 to 10 days of incubation, we can see the development of mycelium from the tissues, right? After development of that mycelium, we have to transfer this mycelium into the half-cooked grain-based medium for mother spawn production. Mother spawn production only. There are two types of spawn are there. One is mother spawn, another one is bed spawn. First of all, we have to produce the mother spawn. This is the method for procedure for mother spawn production. After mycelium development, you can transfer this mycelium, a small portion of mycelium, into the half-cooked grain-based medium in a bottle or polythene bag also. That procedures are done in the sterilized environmental conditions like a lab with laminar airflow chamber. Next one. This is the method of bed spawn production. The mother spawn is the first producing a spawn. The next one is bed spawn production. By using mother spawn, we have to produce the bed spawn production. For bed spawn production, we need some grains like white sorghum grains or maize or wheat. Anything you want are availability. For that bed spawn production, first of all, you have to soak with these grains in the clean and sterile water to remove choppy grains. Then after that, you can boiling these grains in a hot water for 30 minutes. After that, we have to trying these grains in a shady place, sterile environment. Then we have to mix calcium carbonate at the rate of 20 gram per kilogram with these tried grains. For the purpose of adding this calcium carbonate to remove the excess amount of water and to neutralize the pH of the sorghum grains or any other grains, and also to prevent the caking formation in that grains. Next, we have to, after the trying, after the mixing of that calcium carbonate into the grains, we have to transfer these grains into the glucose or glass bottles or autoclavable polythene bag. Then we have to fill these grains at 75% of this bottle or polythene bag. After that, we covered this bottle or polythene bag with the help of adsorbent cotton plug or PVC ring. Then after filling these grains into the bottle, we also sterilize the grains with the help of autoclave or any other autoclave. After autoclave, taking out that bottle or polythene bag with the grains, then we have to add that mushroom spawn, mother spawn into this bottle or polythene bag in the sterile environmental lab or laminar airflow chamber. After inoculation of that mother spawn into this bottle, after 15 days of that inoculation, we can see this mother spawn running into the bottle or plastic polythene bag. That is the bed spawn. That is the bed spawn. Next one. Once the bed spawn was prepared, the third step is preparation of substrate. Substrate means the material which is used for mushroom cultivations, right? Here we can use paddy straw or sugarcane bagaces or culled maize cobs or any other agricultural waste like cotton straw. Whatever you have, you can use that substrate. Here we can use that paddy straw. Take the paddy straw and cut or chopping this paddy straw into the height of 5 to 10 inches. Then that chopping paddy straw undergoes hot water treatment or chemical treatment. If we use chemical treatment, we can use 10% or 20% formaldehyde, or the best method is hot water treatments. After hot water treatment, why we have to do that hot water treatment for if any egg, insect egg or any microbes are found in that paddy straw, we should kill or we should remove that insect egg or any microbes which is found in that paddy straw, so that we have to do that sterilization. After sterilization of the paddy straw, we have to remove the excess amount of moisture content present in the paddy straw. For that purpose, we have to dry that paddy straw in the shady places, not open sunny places. Shady places, that place should be in a sterile environment, right? Next one. This is the this slide shows the one boy trying the paddy straw after sterilize the paddy straw in the shady places. Next one. This slide shows the good and wrong paddy straw trying method. The first one is the good method of preparation of the paddy straw. Another one is the wrong methods of preparation of the paddy straw. If you press that paddy straw after sterilization, a few drops of water should be released. It is good for mushroom bed preparation. If a lot of water leaks out that straw, then it is just too wet. This is not suitable, not good for mushroom bed preparation, right? Next one. Right. The next step is preparation of mushroom bed. First of all, you have to wash your hands thoroughly with any antiseptic and disinfecting agents like Dettol or formaldehyde or anything else. Then take a polythene cover and tie the bottom end with thread or any rubber or any PVC pipe and PVC ring and turn it inwards. Right? Take out a well-grown spread spawn. Already we prepared that spawn in bottle or any polythene bag. That spawn we have to divide and spread thoroughly, because the mycelium mat is formed that grain, so that we have to gently press that grains for spreading and divide into two equal holes. Then we have to fill the paddy straw, sterilize the paddy straw to a height of 2 to 3 inch in the polythene bag. Then take a handful of spawn and sprinkle over the straw and along the sides of the polythene bag. Repeat this process to get the filling of the polythene bag. Then gently press the bed and tie it tightly with the thread. Put ventilation holes with sterile needles for aeration purpose, and also to evaporate heat formed inside the mushroom bed. Next slide. Right, this is the slide shows the mushroom bed preparation methods. Here, some of the students are prepared mushroom bed with the paddy straw. Next one. Right, after bed preparation, you have to keep that mushroom bed in the spawn running room. By using hanging rope method or rack method or cylinder method. Right. Here the hanging rope methods are there, and the cylinder methods are also there. We have to, after bed preparation, we have to keep that mushroom bed inside the spawn running room. After that, we have to watering the inside the that spawn running room. We never watering directly to the mushroom bed. We should only watering on the floor, on the surface of the floor, or the peripheral region of the canny bags only. We have to watering 3 to 4 times per day, depends upon the environmental conditions, right? So that we have to maintain environmental moisture content. Then after next one, previous right, this one. After keeping the mushroom bed in spawn running room, 10 to from the 10 to 15 days, there is a the mother's bed spawn running entire mushroom bed. If you look at the center portion, you can see the dark white colored mushroom bed. That is the 10 to 15 days of inoculation. After 15 days, there is a pinhead size of that mushroom fruiting bodies are developed from that holes. From that holes, already we put that holes. Once the pinhead formations happened, we have to transfer this mushroom bed into the cropping room. Next one. Right, this is the cropping room. The mushroom beds are keeping in the hanging rope system in our college mushroom house. Next one. Right, this is the another method. This method is generally followed in large scale industry. This method is known as a cylinder method. Here the mushroom bed is a little bit higher, the maximum of 10 to 15 feet. Here we have to use the polythene bag at the height of 10 to 15 feet. For standing or supporting purpose, we have to kept PVC pipe with holes in the center of the polythene bag, so that we have to maintain the aeration inside the mushroom bed, and also remove the heat formation inside the mushroom bed. Other methods are same as that hanging rope system methods. These methods are followed in a large scale industry. Next one. Next one. Right. This method is the cultivation of milky mushroom. Right. I already told you one is oyster mushroom. The second one is milky mushroom. There is a slight difference between this milky mushroom cultivation and the oyster mushroom cultivation. Here, an extra step is called casing. After spawn running, casing is nothing but adding of a slight or thin layer on the surface of the mushroom bed after spawn running. That means after 15 days of inoculation, we have to cut the mushroom bed into two equal holes. Then we have to making an extra layer, the formation of the extra layer with the garden soil with the calcium enrichment, enriched calcium garden soil for for the induction of pot formations. Right. This method is known as polythene covered pit tend methods. These methods was followed for milky mushroom. These mushrooms can able to grow 30 to 35 degrees Celsius, but oyster mushrooms needs 20 to 25 or 28 degrees Celsius. Right here, an extra step is known as casing is needed. Casing is nothing but the formation of thin layer of the garden soil on the surface of the mushroom bed. Next one. Right. After 15 to 18 days, there is a first cropping is done. Once the finger stage is formed, after 3 to 3 days, that means 21 days from the first days, we can harvest or craft the first harvesting from the mushroom bed. Depends upon the environmental condition, moisture content, light aeration, and sterile environment. We can harvest 5 to 6 times in a single bed, right? The harvesting should be done in the early morning before watering. Next one. Right, here our boys are collected the oyster mushroom species early morning in our farmhouse. Next one. Next one. Next one, please. Murugesan. Next. Right. These are the photographs. We give next one, previous one, previous one. Previous one, please. Next one. Next, nice. That one. These are the photographs. We give free mushroom cultivation training to textile mills women nearby our college, and also we give mushroom cultivation trainings to the college students. It leads to self-employment of students and also textile mills women. Next one. Next one, please. Thank you. Thank you. Thank you.
Hello, sir.
Tell me, Murugesan.
Thank you, sir. Your presentation is very nice and motivated our students. I hope our students may started initiation to mushroom cultivation also.
Sure.
Yeah, very much thanks.
Sorry for the inconvenience. From the beginning, there is an internet connectivity, so that I can't able to share my screen.
It's okay, sir. No problem, sir.
Okay.
Even though our students are very enthusiastic for listening your lecture, sir, actually.
Thank you.
Okay, sir. Thank you, sir. And participants, the validatory functions will be start at within 5 minutes. Thank you. And also the feedback link will be shared also after the validatory function. Thank you.
Okay.
Nine zero one double 662.
Good evening to one and all present here. Let us start the validatory function. First, I would like to invite Mr. K. Murugeshan, Joint Organizing Secretary, ICPSB-2021 to deliver his welcome address.
Good evening, all. Sunset time. Birds are moving to their homes. Yeah, I welcome you all for the valedictory function of 2 days international conference on innovation in plant science and biotechnology, ICPSB 2021. In this sweet evening, initiation and inspiring is one of the most valuable characters to human being. Yeah, likewise, inspiration person, our founder, Sri V. Chandrasekharan, and chief patron of the program of international conference. I welcome you, sir. And yeah, gardens are very beautiful. The beauty of gardens by a good hedges and edges. Yeah, likewise, our secretary and additional secretary, Mr. R.P. Raji, and our secretary, Mr. R.P. Raji and Mr. P. Venkatachalam, our patron of program of international conference, ICPSB 2021. I welcome both of you, sirs. Every garden maintained by a well-planned manner. Likewise, our SVM college also well maintained and run by our principal, Dr. T. Balasubramanian, sir, and chairman of the program. I welcome you, sir. I extend my warm welcome to Dr. N. Gunashekaran, vice principal and chairman of the program of ICPSB 2021. I welcome you, sir. Every garden have large trees to provide shades like a large shades. Our department head, Mrs. N. Pushpalada ma'am, I welcome you ma'am for the valedictory function. Every garden has small trees, shrubs and herbs. A small emerging tree like convener of our program and assistant professor Dr. R. Vinod Kumar. I welcome you, sir. And shrubs make a beauty of the garden. Yeah, I welcome Dr. V. Chinnadurai, co-convener of the program of ICPSB 2021. I welcome you, sir. Herbs are another one beautiful to make a garden. So I welcome, like a person, I welcome Dr. V. Satish Kumar, organizing secretary of the program of 2 days international conference. I welcome you, sir. The seedlings are next generation producing plants. Like a person, assistant professor, Mrs. Dorani, I welcome you, ma'am. Garden plants are grown by adding a manure and nutrition like our SVM garden, which means our SVM college added to provide a more exposure by eminent person lectures and inviting lectures like that. Yeah, I welcome all the speakers from outside country and outside state. I welcome you all for the valedictory function. And also I welcome Dr. S. Sahaya Satish for the valedictory address of our valedictory function of ICPSB 2021. I welcome you, sir. And also, each and every garden has more beautiful by their flowers and fruits. So I welcome our SVM college students and PG department students for this program, and also the other department students and other department students of SVM college for this valedictory function of ICPSB 2021. And also, I extend warm welcome to other outside country students, professors, staffs, research scholars, research fellows, and outside state are also welcome to the valedictory function of ICPSB 2021. I welcome other department staffs and also technical team for this valedictory function of ICPSB 2021. So once again, I welcome you all for the valedictory function of International Conference on Innovation in Plant Science and Biotechnology of ICPSB 2021. Thank you.
Thank you, Murugesan, for your energetic welcome address. Next, I'd like to invite and request Mr. P. Venkatachalam, Additional Secretary, Sri Vidyamandir Arts and Science College, to deliver his presidential address. Please, sir.
அன்பிற்கினிய கல்லூரியினுடைய முதல்வர், பேராசிரியர் பெருமக்கள், 2 நாள் கருத்தரங்கில் பங்கு பெற்ற எனது அருமை மாணவ கண்மணிகள், இரண்டு நாள் கருத்தரங்கில் மிக சிறப்பான ஒரு கருத்துக்களை வழங்கிய பேராசிரியர் பெருமக்கள் அத்துணை பேருக்கும் மாலை வணக்கத்தினை தெரிவித்துக் கொள்கின்றேன். தாவரவியல் உயிரினங்கள் வாழ்வதற்கு உணவு மிக அவசியம். அந்த உணவை என்ன விதமான புதிய மாற்றங்களை கொண்டு வந்து நாட்டினுடைய உணவு பற்றாக்குறை எவ்வாறு போக்குவது, எவ்வாறு விவசாயத்தில் ஒரு முன்னேற்றமான புதிய நுணுக்கங்களை கொண்டு வருவது என்பதை பற்றி 2 நாள் கருத்தரங்கில் பல்வேறு தலைப்புகளில் பல்வேறு பேராசிரியர் பெருமக்களால் உங்களுக்கு கருத்துக்கள் வழங்கப்பட்டது. இந்த கருத்துக்களை நன்கு கவனித்து எதிர்கால வாழ்வில், எதிர்கால உலகத்தில் விவசாயத்தில் ஒரு புதுமையை கையாள வேண்டும். வளர்ந்த நாடுகள் எல்லாம் இன்று உணவு பற்றாக்குறை இல்லாத நாடுகளாக மேம்பட்டு இருக்கின்றன. அதற்கு காரணம் அந்த நாடுகள் எல்லாம் விவசாயம் முதன்மை தொழிலாக கருதப்பட்டு அதில் புதிய கண்டுபிடிப்புகள் கொண்டு வரப்பட்டு இன்றும் விவசாயத்தில் புதிய வகையான நிலவுகள் செயல்பாடுகள் செய்யப்பட்டிருக்கின்றன. நீர் எந்த அளவிற்கு விவசாயத்திற்கு அவசியம் என்பதை நீர்வள மேம்பாட்டின் மூலமாக எந்த விவசாய பயிர்களை செய்தால் நீர் வளத்தை மேம்படுத்த முடியும் என்றெல்லாம் கருதி அதில் புதிய இக்கால நுணுக்கங்களை பயன்படுத்திக் கொண்டிருக்கின்றார்கள். சாதாரணமாக சொல்ல வேண்டுமானால் ஜப்பான் போன்ற நாடுகள் எல்லாம் தரைத்தளம் அதிகமாக இல்லாத இடங்களில் கூட கடலில் கப்பலை நிறுத்தி அதன் மேல் மண்ணை பரப்பி அதில் விவசாயத்தை செய்து இன்று சாதனைப்படுத்திக் கொண்டிருக்கின்றார்கள். இதற்கு அவர்கள் விவசாயத்தில் புதிய நுணுக்கங்களை மேற்கொண்டிருக்கின்றார்கள். அதுபோல இந்திய நாட்டின் விவசாயம் மேம்பட்டு இந்திய நாட்டில் கூடிய அத்துணை மக்களுக்கும் உணவு பற்றாக்குறை நீங்க வேண்டுமானால், உணவு பற்றாக்குறை நிவர்த்தி செய்யப்பட வேண்டுமானால் நிச்சயம் மற்ற துறைகளில் இருக்கக்கூடிய புதிய நுணுக்கங்களை விட இந்த துறையில் புதிய புதிய கண்டுபிடிப்புகள் வந்தால், இந்த இளைய சமுதாயம் அந்த கண்டுபிடிப்பில் புதுமையை கொண்டு வந்தால் விவசாயம் ஒரு காலத்தில் உயிரான தொழிலாக இந்த உலக மக்களுக்கு ஒரு முன்னுதாரணமாக அமையும் என்பதை கூறி, இதை இளைய சமுதாயம் பயன்படுத்தி எதிர்காலத்தில் உணவு பற்றாக்குறை போக்கக்கூடிய விவசாயத்தை நாம் சிறப்புமாக செய்ய புதிய நுணுக்கங்களை நீங்கள் கண்டறிய வேண்டும். இதில் உங்களுடைய ஆராய்ச்சி மனப்பான்மையை வளர்த்து மேலும் மேலும் புதிய கண்டதில் உங்களுடைய ஆர்வம் மேம்பட வேண்டும் என்று வாய்ப்புக்கு நன்றி கூறுகின்றேன். நன்றி, வணக்கம்.
Thank you for your inspirational presidential address, sir. Next, I request Dr. P. Balasubramanian, Principal, Sri Vidyamangal Arts and Science College, to felicitate the gathering. Welcome, sir.
Honorable Founder, respected Secretary and Joint Secretary, dear eminent personalities and invited speakers, dear staff members of our institution and participants of other institutions, dear students, good evening, one and all present here. It is my immense pleasure to give you the valedictory address before you. I hope this international webinar gave you deep insight regarding plant science and biotechnology. I think you have learned the latest, what are the latest applications and the research being conducted over the world through this webinar. I request the research scholars and postgraduate students to publish your contribution in a reputed journal to reach out your ideas throughout the world. I congratulate the Department of PG and PG Department of Botany staff members for arranging this wonderful webinar. I congratulate the participants also. Thank you for the opportunity given.
Thank you, sir. I request Dr. N. Gunasegaran, Vice Principal, Sri Vidyamangal Arts and Science College, to felicitate the gathering.
The most illustrious founder, most respected secretary, most venerable additional secretary, respected principal, distinguished chief guest and speakers from various nations and within India, heads of various departments, professor participants, scholars, my beloved students. A pleasant good evening to one and all gathered in the webinar. I'm very much delighted that the PG Department of Botany has organized a two-day international virtual conference on innovations and relevance in plant science and biotechnology. Having a minimum strength of students, the department, the HOD with the team of faculty members organizing an international conference for the past 2 days and having succeeded in their efforts, evidenced as they are well planning, well organized, and well executed. On behalf of the management and the principal, and on my own behalf, I convey wholehearted appreciation to the department and the team of faculty members. And one more thing I would like to share here, that out of the conference, the overwhelming response from the participants all over the world and other states, the professors and dignitaries, shows that the speakers' in-depth knowledge and their share of knowledge and the potential of matters relevant to the topic of the conference. And one more thing is proven, that the research and the academic matters have been in deep discussed in the conference. That's why the overwhelming response of the participants have been shown. At the outset, the feedback of the students also proved that the conference has reached the success, almost ultimate success, with the hope of the participants have used and get benefit of the international conference, 2 days international conference, for their betterment in the research and academic affairs. I would like to thank, on behalf of the management and the principal, I would like to thank the speakers from particularly the Korea and Australia and also from other states for giving their fullest cooperation and contribution for making and converting these international conferences into a grand success. And also convey thanks to the participants and professors, scholars, stockholders, and all the other dignitaries for making this international conference a grand success. And once again, I convey thanks to one and all. Thank you.
Thank you, sir, for your encouraging and for your appreciation. Thank you so much, sir. Next, I'd like to invite Dr. S. Sahaya Satish to deliver his valedictory address. Dr. S. Sahaya Satish, Dean, School of Biological Sciences, Director, Center for Cryptogamic Studies, St. Joseph's College, Trichy. He has published more than 70 research articles and he has organized and attended many conferences and seminars. He has submitted one patent. He is a reviewer in many peer-reviewed national and international journals. He is holding many academic positions. He has more than 30 years of research and teaching experience. He has guided more than 10 PhDs and 4 MPhils. With this, I invite Dr. Sahaya Satish, sir, to deliver valedictory address. Welcome, sir.
Hello. May I audible?
Satish sir, is it audible, sir?
Okay, okay, good. Right.
Sir, you're not audible to us, sir.
Yeah, yeah. Honorable patron and founder of Sri Vidya Mandir Education Group of Institution, Thiru V. Chandrasekharan, the secretary of the institution, Thiru R.P. Raj, and additional secretary. May I audible? Kekada? Hello?
Sir, you're audible, sir.
Okay, okay. The respected chairman and the principal, Dr. T. Balasubramaniam, and Vice Principal Gunasekharan, the head of the department, Pushpalada Madam, the convener of this great program, Dr. Vinod Kumar, co-convener Chinnathurai, and organizing secretary, Dr. Sindhil Sadish Kumar, and the speakers, especially my student, Dr. Bhaskaran, and my friend, Dr. Manchu, and other speakers, my student friends, participants, a pleasant good evening to all. First of all, I would like to thank the organizers for giving me this opportunity to deliver the variety address in eventful, nice program, an international conference in a reputed institution like Sri Vidya Mandir. I congratulate the organizers. So wonderfully they organized. They invited many speakers from all over the world and other states with a pluralism, not only from the region, also the topics. So they created a very nice platform for the students, for the young scientists, young researchers to inculcate the recent advances in our biological sciences. So thus they successfully convened the program. So the time is almost 4:30. So for the past 2 days, the participants are hectic with the showers of information from various speakers. So you are all tired, but I am going to give the variety address. I know what will be the response. Usually we used to say in our institution, யாரையாவது நமக்கு பிடிக்காத ஆளை என்ன பண்ணோம்னா வெரைட்டி அட்ரஸுக்கு கூப்பிட்டுருவோம். நான் அந்த அளவுக்கு ஒரு வெறுப்பை ஏற்படுத்தக்கூடிய ஒரு நபராக இருப்பாருன்னு சொல்லுவோம். ஏன்னா 4:30 ஆயிடுச்சு. Anyhow, I want to confine to the time. So I will speak something what I have prepared. In a nutshell, I am not going to take much time because of the short of time. Already in the first speaker spoke on nanotechnology. Since I am indulging in nanotechnology in my field, biology, so amalgamation of bryophytes with the nanoscience, I have the experience in this field. So I want to share something about from my findings. May you see will you see the slide? The organizers, please.
Sir, the slides are visible, sir.
Okay. So some glimpses I have just introduced the nanotechnology. So this concept was first introduced by the physicist Richard Feynman. Will he deliver a talk on the topic, there is plenty of room at the bottom. So after this, in the history of the nanoscience, the scanning tunneling microscope in 1981 and the discovery of fullerene, that is C60 in 85. that lead the emergence in nanotechnology. And this term, nanotechnology, is first coined by Norio Taniguchi in 1974. So Richard Feynman is the person who gave or emerged the idea and concept behind the nanoscience and nanotechnology. In his, he described that this nanotechnology, by nanotechnology, one can manipulate and control the individual atoms and molecules. So nowadays, in the past decade, in this present decade, the nanoscience and nanotechnology are the study of the application of extremely small things can be used across all the other field of science, chemistry, physics, biology, material science, and even engineering, and even civil engineering, mechanical engineering. So many fields. So this nanotechnology have the ability to create many new material and devices with a vast range of application, specifically in medicine, electronics, biomaterials, energy production. This scope is going on. In a nutshell, we can define this nanotechnology term as manipulating the matter on an atomic scale, and also it refers to the constructing and engineering of the functional system at a very micro level. We can say at atomic level. As you know, what is nanometer? It's a 1 billionth of a meter. 1 billionth of a meter. So roughly, we can compare with the width of 4 atoms. That's a nanometer. Our human hair is around 25,000 nanometer wide. So that much small concept. So nano plus technology. So nano is a unit of the length in the metric system. We will explain what is nanometer. We know technology, that is a broad, with the broad sense that making usage and all the tools and machine and techniques in the overall problem of the persisting to the specific function. So anyone can very hard to imagine just how small nanotechnology is. You know, 1 nanometer is a billionth of a meter, or 10 to the power of -9 of a meter. So in the world or in the universe, we can categorize the whole system or the particles or everything, categorize it into 3 groups on the basis of their sizes: macro, micro, and nano. The objects or the particles are the substances above 1 millimeter. 1 millimeter in the scale we can see, in a small scale, we can see 1 centimeter divided by having 10 millimeter. Isn't it? So 10 millimeter, 1 millimeter we can see. So above 1 millimeter, we can categorize under the macro particles. And below 1 millimeter to 100 nanometer, above 100 nanometer and 1 millimeter, we can categorize under the category of micro particles. So the macro, and human being, the apple, and these are all the coming under that category. In micro, a diameter of a human hair is a micro object. The smallest we can see is the human hair. And Escherichia coli is a microorganism, the bacteria, that is coming under 2000 nanometer. That is coming under microorganisms. Nano are those things, those objects are less than 100 nanometer. Less than 100 nanometer. For example, the DNA molecule, polypeptide chain, a buckyball, a diameter of a carbon nanotube, that is coming under 1.3 nanometer. So those substances, those molecules, those matters less than 100 nanometer are called, we can say it as nanoparticles. Others are microparticles and macroparticles. So this is the concept. The nanoparticles should less than 100 nanometer. This is a theoretical matter. Here 1 centimeter can be divided by 10 millimeter, and 1 millimeter we can divide it into 1000 micrometer. Can you see? And 1 micrometer, micron, we can divide it by 100 nanometers, 1 micrometer. See, in the 1 nanometer, we can divide it into 10 Armstrong meters. So 100 nanometer, that much small, that is small, just nanometer. So some of the glimpses in the nanotechnology we can see, and I'm not expertise in these aspects, the physics, the medical science, isn't it? My specialization is antioxidant and antimicrobial, that we can see in the next occasion. But some glimpses, this is nanorods, or one of the morphology of a nanoscale object that dimension from 1 to 100 nanometer. They are synthesized from metals or semiconducting materials. This is how these are produced. This is a combination of ligands and as a shape control agents and bond to the different facets of nanorod with the different strength. So this allows the different phases of the nanorod to grow at different rates, producing an elongated object. What's the use? What's the purpose? The ligands amalgamated or combined with these facets. It display technologies because the reflectivity of the rod can be changed by changing their orientation with an applied electric field. So the MEMS, it can be used in cancer therapies. So the protection, how the nanoparticles can be produced. The two approaches are there. One is bottom up, another one is top. Bottom up. This is assemblage. We can create nanoparticles by assembling the molecules. So in the bottom up approach, different materials and devices are constructed from molecular component of their own. They're chemically assemble themselves by recognizing the molecule of their own breed. They will recognize those have the affinity. Any metabolite, they have the affinity with the silver ion, that will amalgamate. So assembling of this molecule. One example, you know how the DNA molecule assemble the amino acids and nitrogenous bases and sugar molecules, everything, and converted into a long polypeptide chain. So what synthetic base pairing is one of the example of the assemblage. Another one, top down. In top down approach, nano objects or materials are created by larger entities without bouncing in atomic reactions. Usually top down approach in practice less compared to the top downs are very less. Only in the physics they will produce in physics. Solid state techniques can be used to create devices known as nano electromechanical systems, that is NEMS, which are related to the micro electromechanical systems. Okay, these are coming under the electrons. So this is one of the devices having characteristics. So this is the application of nanotechnology. So as we discussed earlier, the innovative application of the nanotechnology to various fields, starting from the medicine and drugs, nano devices, nanobiotechnology, optical engineering, defense and security, bioengineering, cosmetics, nanofabrics, energy. So the nanotechnology have the application in all the fields, Why, how, we will discuss. Some of the implication we not discuss. Okay, I'll just skip. The early 2000s, some of the beginning of commercial application and nanotech technology, although there was limited to bulk application of nanomaterial, but many researchers, many industries, they started many research stations and centers, they started doing silver nano platform for using silver nanoparticles as an antibacterial agent, and these nanoparticle based transparent sunscreens and carbon nanotubes for stain resistant textiles. So in the textiles, in the medicine, so in many field, the silver nano particles came to it. This is one of the example, the modern nanotechnology, antimicrobial fabric. The Nano Horizons, a company, Nano Horizons, in Pennsylvania, has started producing a silver nanoparticle material as both a dye and in polyester and nylon. In the picture, you can see some different colored socks. This is called nano socks. You know what is socks? When I was working at St. James College, Panangotai, before I joined the St. Joseph's, my brother was an advocate. He was at Bangalore at little time in 96-7. He brought me these nano socks. What is the specialty of these nano socks? Usually in the hostel or in the home, college students, the boys, as soon as they came inside, they will remove the socks and throw it on the corner. What will happen? The members in the home, they will feel like some rodents died somewhere. ஏதோ எலி செத்த மாதிரி ஸ்மெல் வருது அப்படின்னு பார்ப்பாங்க. அப்படி எக்ஸ்பீரியன்ஸ் ஆயிருக்கீங்களா? இல்லை கிளாஸ்ல கூட ஒரு பையன் ஒரு சாக்ஸை ரிமூவ் பண்ணான். A bad odor will create from the socks. But when you use these socks, that won't come. How the odor is come? Because of the microbes, the colonies. Usually these boys with the wet legs, with the wet legs, they will wear the socks. No, what will happen? The microbe will colonize and that will create some and the decayed matter that will create a bad odor. If you use this silver nanoparticle socks, these silver are toxic. That's why we are using silver plate. Silver are toxic to the microbes. It won't allow the colonization of microbes in the socks. And what will happen? You can wear it for 2 to 3 days without washing. No water will come out of this. So this is one of the applications. And in the fabrics, nanotechnology in fabrics, the properties of familiar materials are being changed by manufacturers who are adding nanosystem components to conventional material to improve the performance. So to increase the performance, so the nanosystem components are in fabrics also. So in the picture you can see, for example, some clothing manufacturers are making water and stain repellent clothing. Water repellent and stain repellent clothing using nano-sized whiskers in the fabric that cause water to bead up the surface. So the second photograph, you can see the water is bead up. It won't absorb by the so it's helping in the manufacturing, even in bulletproof jacket also. That is using the nano-sized components. So making spill and dirt resistant, antimicrobial and antibacterial fabrics, the nanotechnology supply. We address nanotechnology end of life issues. So little known about the effects on nanomaterials and nano based on human health and the environment. So little knowledge at present. And nanomaterials may have differently in the environment than bulk material. Nanomaterials are already in the commerce and the waste stream. No law deals specifically with the nanotechnology because there is such institution not aware of this. So in the medicine also, the nanomedicine is the medical application of the nanotechnology. So nanomedicine ranges from the medical application of nanomaterial to nanoelectronic biosensors. And even possible future application of molecular biotechnology also. So introduction to the biological system, we can so quickly. This is one of the example. Nanoparticle biosensors. This information I collected from the various sources, but now I'm, as I said earlier, I'm not much experienced in expertise in this. So this is some probable future endeavor in the nanotechnology. So antibodies can be anchored to nanoparticle surface to collect specific proteins. It's a possibility. So target protein capture with MMB probe. So the antibodies can anchor to nanoparticle with a surface. So in this example, the nanoparticle is magnetic, allowing it to separate from and analyzed. So the nanomedicine is a monitoring, repair, reconstruction, and control of human body system at the molecular level using engineered nano devices and nanostructures. Currently, several university and industrial research groups are developing medical application from nanotechnology. So you see here, to study the schematic pathway, starting from the initial and the intermediate compound, initial compound, intermediate compound, and the final compound. The pathway is studied previously by isotopes, isn't it? But the nanocrystals, as fluorescent as a biological labels, we can significantly use this for the, instead of the conventional dyes, we can use this. It reduces the photo bleaching, multicolor labeling, parallel screening, so infrared labels, the blood diagnostics, so molecular size nanocrystals and biomolecule compatible with the many other applications. This is the 3.5 nanometer crystals bound to cell nucleus with the fluorescent. So we can easily identify. This is dendrimers, drug delivery techniques. Is not come to practice. Dendrimers, what is dendrimers? Carrying different materials on their branches can do several things at one time. That is, such as recognizing disease states, diagnosing disease states, drug delivery, reporting location, and reporting outcome of the therapy. Is a inclusive of many functions in a single object. This is the respirocytes. This is nothing but a mechanical artificial red blood cell. Mechanical, because nowadays many patients are transferring the blood every week, isn't it? Like that, the respirocytes, we can RBC also we can substitute by a respirocytes. It's a mechanical artificial red blood cell. So these blood-borne are spherical in shape, and as in the picture, and 1 micron diamondoid, 1000 pressure vessel, active pumping powered by endogenous serum glucose, able to deliver 236 times more oxygen to the tissues per unit. Normal red blood cell, it is delivered 336 times more than the normal RBC, natural red blood cells, and manage the carbonic acidity also. This is about the nanobots, fixing damaged blood cells, other applications. The nanotechnology in drugs, there's a cancer, providing new options for drug delivery and drug therapies. What is the function here? Use of this application or significance? These nano drug to be delivered to precisely the right location in the body and release drug doses on predetermined schedule for optimal treatment. So predetermined schedule, we can schedule the time, and also we can fix the right location also. So attach the drug to nanocyst carrier, they become localized at the disease site as a cancer tumor. Then they release the medicine that kills the tumor without damaging the other tissues. Current treatments through radiotherapy and chemotherapy, as you know, when a patient is treated with radiotherapy chemotherapy, the whole body will get spoiled. It will completely affected because of side effect. But this nano, they using the applied nanotechnology, we can rightly locate it, the exact place, the affected part. So these nanobots can clear the blockage in arteries also. So we can say it as nanobots. They can select or choose the right location and release the drug as per the scheduled dose with the artery. So this is a application of the nanotechnology in medicine also, and the therapy of the cancer. This modern technology is not the fabrication of tiny mechanical devices. They can also will enter the human system to repair the damage also. This is nanobots. And this is the application is going on like this. How it happened? Nanotechnology. What's the speciality in the nano-sized particles? Yeah, nanotechnology in the application. when nanotechnology applied, the ratio of surface area volume increases. You know, in the mitochondrion, if you see the internal membrane, the outer membrane is just one, another wide one, but the internal membrane will enfold many times to form number of cristae. What's the purpose? The enfolding, many enfolding, to increase the surface volume. So that concept is here. These nanoparticles, the size is reduced. The size is reduced, but the surface is large. And if we take the ratio, the surface area to volume increases, what total volume, but surface is increases. So what will happen? More active site will be there, isn't it? They have this ability to improve material strength and intelligence while enabling product to be smaller, lightweight, more flexible, more efficient, and cleaner. We can say always, now adikadi solluven, small is beautiful. I correlated this to my life also. I am little short. I am little smaller than other people, other person. If I traveling in a bus, I feel very comfortable because my leg won't hit the front seat. But the huge people, they will find very difficult to travel. But the function is the same. Like that. If you want to throw a person with a big stone, you will struggle with the hold and throw. But if you make it into different small pieces, within half or the quarter of the total substance, you can attack. So this is an application. One is the increase the surface area volume and lesser in light and weight. And this material, when reduced to a nano size, can show very different properties than on a macro scale. That is, substances as aluminum and plastic become combustible, also solid gold become a liquid. This is another application. One is the volume. Another one is, if you reduce the size, the properties is differing. Property. The size determines the property sometimes. For example, the plastic can conduct electricity very well on nanoscale. Aluminum and become combustible. Solid gold become liquid. So the changing the size, giving new favorable property character. Good. But so to conclude, a variety of researches is being performed in nanomedicine, nanomedical sciences. Few industries products exist right now. So there's a possibility endless, but will take time to develop. I here with this is my work. So the green synthesis, and here with the plant, bryophyte plant, biomedianum, because my PhD specialization, my major research project, which project is in bryophyte, enumeration of bryophyte from the eastern Ghats of Tamil Nadu. So in the bryophyte, a plant called biomedianum, I produced the nanoparticles by amalgamating with the plant extract with the silver nitrate solution. So successfully I produced, and with my PhD scholars produced the nano synthesis there. And the physical parameters I have done, I'm not going to show because of the short of time, I'm going to finish. I done all the physical parameters, starting from the UV visible spectrum. First the color indication, then UV visible, then FTIR, like that, up to the XRD and up to. So by the characteristics, I confirmed the synthesis of the nanoparticle, and I confirmed the presence of silver particles in that nano synthesized solution. Then I done the antimicrobial, antioxidant studies, and even anti-cancer studies also. And successfully I found the nanoparticle synthesized from this bryophyte biomedianum more efficient than the raw or pure plant extract. So this is the my study. So if I have any chance, we will discuss later. So here I conclude myself, and I thank the organizers for giving me this wonderful opportunity to give the valedictory address to your reputed institution. I thank Dr. Vinod Kumar and Satish Kumar. I should especially mention their cooperation in contacting me over the phone and gave me this opportunity. Thank you one and all. Wish you all the best. Thank you.
Thank you so much, sir. Thank you for your informative lecture, sir. Next, I'd like to invite Dr. B. Satish Kumar, Organizing Secretary, ICPSB 2021, Sri Vidyamandir Arts and Science College, to give a report about the conference.
Thank you, sir. Sir, Sahai Satish, sir. Thank you for your wonderful valedictory address.
Thank you, sir. Thank you.
I should thank you for your sparing valuable time with us, actually. And thank you very much. Okay. Sir, shall we wind up your session, sir?
Okay, sir.
Okay, sir. Thank you, sir. So participants, if you have any ladies and gentlemen, good evening to one and all. As an organizing secretary, I am pleased to sum up the conference report. Before summing up, I would like to thank formally the management, principal, vice principal, advisors, head faculty members of the PG Department of Botany for giving me an opportunity to conduct this conference. Due to the pandemic situation, the conference was organized in virtual mode using Zoom platform and also live streamed in YouTube. The two-day international conference entitled Innovation and Relevance in Plant Science and Biotechnology 2021 began with the Tamil Thai Valthu at Sri Vidyamandir Arts and Science College, Uthangarai on Monday, that is 15/3/2021. The ICPSB 2021, organized by the PG Department of Botany, was the first of its kind from our department. Around 300 scientists, young researchers, academicians, experts, students from different parts of the world are registered and also taking part in this conference. Actually, I am very surprised because we have intimated to the participants within a short period of notice, but still we have received a good response from the participants. Mrs. N. Pushpalata, Chairperson, ICPSB 2021, Head PG Department of Botany, welcomed the gathering at the inaugural address of the conference held at IQAC Auditorium at Sri Vidyamandir Arts and Science College. Assistant Professor Dr. R. Vinod Kumar, event coordinator, convener, outlined the focal theme of the conference, in which he emphasized the objective of the conference. The ICPSB 2021 was inaugurated by Mr. R.P. Raji, Patron, ICPSB 2021, Secretary, Sri Vidyamandir Arts and Science College. In his address, he said that he was delighted to inaugurate the international conference ICPSB 2021, followed by Dr. T. Balasubramaniam, Chairman, ICPSB 2021, Principal, Sri Vidyamandir Arts and Science College. in his address while presiding over the conference, said that our institution accorded top priority to research to prepare students for a career and a life ahead of them in the area of research. The faculty at Sri Vidya Mandir Arts and Science College trained students here in research. Dr. Balasubramaniam also briefed out the importance of plant science with special reference to agricultural and farm production. Dr. V. Chinnadurai, co-convener ICPSB 2021, concluded the inaugural function by oath of thanking. After inaugural function, the day one was started. Actually, totally 10 invited lectures was given. We have scheduled 5 lectures for the day one and another 5 lectures for day two. Accordingly, day one, totally we had 5 speakers, in which forenoon session, we had 2 speakers from South Korea. That is Dr. C. Ramachandran, postdoctoral fellow from South Korea, delivered his lecture on topic development of value-added symbiotic enriched feeds and foods, followed by Dr. Satyaseelan Anbaragan, postdoctoral researcher from Department of Medical Biotechnology, College of Biomedical Sciences, Gangwon National University, South Korea, delivered his lecture on topic application of nanomaterials in biotechnology and biomedicine, an overview, followed by Dr. Vishnu Kiran Manam, delivered his lecture from Andhra Pradesh on topic isolation maintenance of microalgal cultures and their applications. At forenoon, after the forenoon session, in the afternoon session, Dr. Vishnu Kiran started his invited talk, followed by Dr. S. Maladi, Assistant Professor, Government Arts College for Men, Nandanam, Chennai, delivered his lecture on topic carbon footprint and climate change. The last session of the day one was delivered by Dr. Bhaskaran Ponnuswamy from Australia, the principal scientist and head in biotechnology, Dummy Oil Palm Research Station, Australia, delivered his lecture on topic innovative biotechnological research on industrial production of cloning of oil palms. That was the day one. And again, that is today we have started, day two was started by speaker from South Korea, Dr. S. Arogya Raj, who is an assistant professor from the Department of Food Science and Biotechnology, Sejong University, Seoul, South Korea. He delivered his lecture on topic impact of herbal medicines during COVID-19, followed by Dr. Matthew Dan, the principal scientist and head Plant Genetic Resource Division, Jawaharlal Nehru Tropical Botanic Garden and Research Institute, Palod, Trivandrum, Kerala, delivered his lecture on topic a progression from classification, taxonomy to systematics. After that, Dr. V.S. Ramachandran, the retired professor from the Department of Botany, Bardiyar University, Coimbatore, Tamil Nadu, delivered his lecture on topic relevance of biodiversity conservation with reference to climate change. The afternoon session was started by Dr. Manju C. Nair, Assistant Professor, Department of Botany, the Zamorians, Guruvayurappan College, Kozhikode, Kerala, delivered her lecture on topic diversity and distribution of bryophytes in Kerala and its major challenges in conservation. The last speaker of the two-day international conference was Dr. V. Ramesh, Assistant Professor and Head in Charge, Department of Botany, Vivekananda College, Madurai, Tamil Nadu, delivered his
lecture on topic glimpses of mushroom cultivation techniques. At the end of the session, we have started a valedictory function in which principal, the management persons, secretary, additional secretary, and joint secretary delivered his welcome address, followed by the valedictory address was given by Dr. S. Sahaya Satish, the Dean, School of Biological Sciences and Director, Center for Cryptogamic Studies, St. Joseph's College, Trichy. I once again thank you all the participants for their active participation. And here I would like to thank one of my organizing committee member, Dr. V. Dharani, for her tireless effort and also for her beautiful comparing. Since we have thanked everyone except Dharani, ma'am, so as an organizing secretary, I'm thankful for my team members, all of them, starting from my convener, co-convener, joint organizing secretaries, Dr. yet to be Dr. Murugeshan, Dharani, and our head of the department. With this, sorry for the inconvenience. I would like to thank our technical team, especially Ari sir, Sriram sir, as well as Thirumalai sir. I hope in the vote of thanks, which is going to be given by Mrs. V. Dharani, in which she will elaborately thank all of the participants as well as the committee members. Thanks once again for giving me an opportunity. Thank you all.
Thank you so much, sir. A warm greetings to one and all present here. Just as one candle lights another and can light thousands of other candles, so one heart illuminates another heart and can illuminate thousands of other hearts, Leo Tolstoy. At the outset, I feel immense pleasure to deliver vote of thanks. I would like to express my gratitude to our founder, Sri Chandrasekharan, Sri Vidyamandir Groups of Educational Institution, for encouraging us in conducting the international seminar. Thank you, sir. I extend my deep word of gratitude to Mr. R.P. Raji, Secretary, SPMC Arts and Science College, for extending his help during the conference. Thank you, sir. I would like to thank Mr. P. Venkadachalam, Additional Secretary, Sri Vidyamandir Arts and Science College, for supporting us during the conference. Thank you, sir. I extend my thanks to Dr. T. Balasubramanian, Principal, Sri Vidyamandir Arts and Science College, for providing timely help and guidance during the conference. Thank you, sir. I would like to thank Dr. N. Gunasekaran, Vice Principal, Sri Vidyamandir Arts and Science College, for helping us during the conference. Thank you, sir. I extend my deep words of gratitude to our advisors, Dr. S. Suresh, Controller of Examination, and Dr. C. Raghavan, R&D Coordinator, Sri Vidyamandir Arts and Science College, for their guidance. Thank you, sir. I extend my thanks to all the speakers for sparing their valuable time amidst their busy schedule. I would like to thank Dr. C. Ramachandran, Postdoctoral Fellow, Kangwon University, South Korea, for his innovative lecture. Thank you, sir. I thank Dr. Satyasilan Anbalagan, Postdoctoral Researcher, Department of Medical Biotechnology, College of Biomedical Sciences, Kangwon National University, South Korea, for delivering an informative lecture. Thank you, sir. I would like to thank Dr. Vishnukiran Manam, co-founder and head R&D, Dr. Ella Pragada Life Sciences, Andhra Pradesh, for his interactive lecture. Thank you, sir. I extend my gratitude to Dr. S. Malati, Assistant Professor, Government Arts College for Men, Nandanam, Chennai, for delivering a lively lecture. Thank you, ma'am. I would like to thank Dr. Bhaskaran Ponusamy, Principal Scientist and Head in Biotechnology, Dami Oil Palm Research Station, Australia, for his enthusiastic lecture. Thank you, sir. I would like to thank Dr. S. Arokyaraj, Assistant Professor, Department of Food Science and Biotechnology, Sejong University, Seoul, South Korea, for his motivating lecture. Thank you, sir. I would like to extend my gratitude to Dr. Matthew Dan, Principal Scientist, Head, Plant Genetic Resources Division, Jawaharlal Nehru Tropical Botanic Garden and Research Institute, Palur, Thiruvananthapuram, Kerala, for delivering a wonderful lecture on classification, taxonomy, and systematics. Thank you, sir. I would like to thank Dr. V.S. Ramachandran, Professor Retired, Department of Botany, Bharatiya University, Coimbatore, for delivering an informative lecture. Thank you, sir. I extend my thanks to Dr. Manju C. Nair, Assistant Professor, Department of Botany, the Zamorans Guruvayurappan College, Kozhikode, Kerala, for delivering an inspirational lecture. Thank you, ma'am. I would like to thank Dr. V. Ramesh, Assistant Professor and Head of Department of Botany, Vivekananda College, Madurai, for delivering an informative lecture. Thank you, sir. I would like to thank Dr. S. Sahai Satish, Dean, School of Biological Sciences, St. Joseph's College, Trichy, for your valuable presence and inspiring valedictory address. Thank you, sir. I would like to thank college technical team for their tireless and timely help. Thank you, Dr. Harikrishnan, Dr. Sriram, and Dr. Tirmalai. Thank you so much, sir. I feel happy to thank all our energetic and active participants for their kind cooperation and patience throughout these sessions. Thank you all. Last but not least, I would like to thank our department staff members and organizers of this international conference, Mrs. N. Pushpalata, Dr. R. Vinod Kumar, Dr. V. Chinnadurai, Dr. B. Satish Kumar, and Dr. K. Murugeshan for making this conference a great success. I also extend my thanks to all our buds and blossoms, our students of Botany Department, for their kind cooperation and support. Thank you all. Thank you once again for your gracious presence. Let us end the international conference. I hope it will be a great success. Let us raise for the national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Once again, I thank you all for your kind cooperation and gracious presence. Thank you all. With this, we conclude the 2 days of international conference. We have forwarded you the feedback link. You will be getting the certificate shortly. Thank you.